Hello everyone, a very good morning to all assembled here. I, Akshata Geet, welcome you all to Second Student Research Congress 2021 organized by SVKMs, Dr. Bhanubai Nanavati College of Pharmacy, co-hosted by University of Mumbai. I would like to start the session with a quote that says, innovation is taking two things that already exist and putting them together in a new way. And that is our theme for Second Student Research Congress event, Innovations in Healthcare. This is the day 2 of our 3-day online events. Now, before briefing you all about today's schedule, let me give you a quick recap of our day 1 event. We heard the keynote speaker of event, Dr. Padma Devarajan, and her views about the research. And then we started with our oral presentation competition, PhD and PG category. And I must say that yesterday we got a very good response from all the participants and our audience. And we hope we get the same support and enthusiasm today as well. I may now like to welcome our judges for today's event. Our very first judge for PG PhD category is Dr. Swati Mittal. She has over 13 years of teaching experience in reputed educational institutes. She is currently employed at Al-Amin College of Pharmacy, Bangalore, as a professor and HOD of Department of Pharmacology. She has worked at VES College of Pharmacy, SPP, SPTM, and CU Shah College of Pharmacy. She completed her PhD from CU Shah College of Pharmacy in the year 2008. She completed her M.Pharm and B.Pharm from VL College of Pharmacy. She has published about 13 papers in different journals. She has obtained 4 research grants. So, to name a few, grant worth ₹5 lakh from Themsis Medicare Limited for the formulation development of controlled release oral suspension. She has guided about 30 M.Pharm projects. We would like to welcome you, ma'am, with a bouquet of flowers.
Thank you so much, everyone.
Moving on, I would now like to introduce our next judge, Dr. Sarita Shetty. She is currently working as Associate Professor of Pharmaceutics in SPP SPTM. Before joining academics, she served as scientist at Syngene International. During this tenure, she has completed projects of major clients like Baxter for analytical method development and validation using HPLC and GC. She has pursued her PhD from SVKMs NAMES and M.Pharm degree from Rajiv Gandhi University, Bangalore. In the year 2015, she has been invited as speaker amongst various eminent scientists worldwide at chromatographic societies on analytical challenges and solutions for difficult pharmaceutical molecules. She is recipient of Oral Presentation Award during Iconica 2020 held at Punjab University. She is recipient as co-PI for an international grant under Grand Challenges Canada. She is a life member of IPA, APTI, IWSA. We would like to welcome you with a bouquet of flowers.
Nice gesture, huh? Thanks.
Our third judge for this event is Dr. Manisha Ozama. She has completed her bachelor's of Pharmacy from LM College of Pharmacy and Master of Pharmacy from Sardar Patel University, Gujarat. She has obtained her PhD from NIMS University, Mumbai. She is currently working as Assistant Professor at SVKMs Dr. Bhanubai Nanavati College of Pharmacy since 2009. She is involved in research related with natural products and phytopharmacology since 14 years. Her area of research mainly focuses on effect of natural products in diabetes and its complications. She has published more than 15 research and review papers in peer-reviewed journals to her credit. She has authored 8 book chapters. Dr. Manisha ma'am has more than 20 national and international presentations to her credit. She has received a travel grant from the Department of Science and Technology, Government of India, to present a poster at International Conference, Japan. We would like to welcome you with a bouquet of flowers. Today, I am extremely grateful to receive this opportunity to give the glimpse of all the judges' achievement to the audience we extend a warm welcome to all our judges. I'm quite sure all the participants must have been waiting for this moment eagerly. So without waiting for a minute, we'll now move forward for poster presentation competition. The event will take place in two categories, PG/PhD category in the first half and UG category in second half. Each team will be allotted 5 minutes, 2 minutes for presenting the poster and 3 minutes for Q&A session. The presenting author of the team will be allowed to present answer to the Q&A session. Presenting authors for all teams are requested to remain alert when their team code is called out. Giving personal information like name, affiliation, etc. are strictly prohibited during the presentation. Before moving forward, I request the participant, the presenting author, to please rename themselves according to their team code if they have not done it, and only the presenting author will rename themselves. Please maintain the time limit strictly. We will now begin with the poster presentation event. Judges, if you are ready, we can call the first presenter. I would call upon PhD1.
Yes. Good morning, everyone. Just a request, shall I share my presentation now? Can I share?
Yes.
Yes.
Thank you. Good morning, all of you. My poster number is PhD D1. Topic of the project is comparative binding mode analysis for synaptic and extrasynaptic GABA receptors. Introduction part. This is regarding PCOS. As I have selected PCOS for this selecting extrasynaptic and synaptic GABA receptor. So polycystic ovarian syndrome, it is one of the most common endocrine disorder among women of reproductive age with the association of hyperandrogenism and chronic anovulation in its most typical form. Polycystic ovaries are defined on ultrasound by the presence of 8 or more than 8 subcapsular follicle cysts of size 10 millimeter and increase ovarian stroma. The disease further generates a constellation of different metabolic abnormalities, worsening the situation more like the increased levels of luteinizing hormone and decreased level of follicle stimulating hormone, which give rise to further abnormalities. The aim and objective behind the study is to identify a suitable target for the PCOS, to design a molecule suitably selective for the target, to design a synthetic scheme in order to synthesize design molecule, synthesis of design lead molecule, and characterization of the synthesized compound by spectroscopic techniques. The rationale behind the work is PCOS is found in 1 in 15 women globally. Expensive symptomatic treatment is available, and it is thus an area requiring extensive research for the development of a treatment that proves
PhD1, your time is up. Time for questions.
My time is up. I have not presented.
The time is only 2 minutes are only there for presenting and 3 minutes are there for Q&A session. Please maintain the limit.
Ma'am, I request you, I can go a little bit fast.
Highlight.
Only 30 seconds and please present the highlights.
Okay, fine. So I'll go further. The experimental overview which was done as sequence alignment, homology modeling, ligand preparation, protein preparation, energy minimization, ligand docking, molecular dynamics, and validation of the homology model. For experimental section, the homology modeling of the alpha-4 beta-3 delta isoform of the GABA receptor, it was done at Swiss model portal, and we have selected PDB-HUO as a reference for preparing alpha-4 beta-3 delta. Sequence alignment was done and the sequences was found at the UniProt on Swiss portal model. This is the sequence alignment for the alpha-4 beta-3 delta. Molecular dynamics and energy minimization. It was done.
Sorry to interrupt again, but the time is over. This is an poster presentation, not a presentation competition. Maintain the time limit.
Okay, ma'am. So this is the
judges. You can now ask the questions, please.
Can you show your first slide where you have compiled all your
yes, ma'am.
data? Yeah.
Yes, ma'am. Thank you very much. Is it visible, ma'am?
Yeah. Slightly, can you zoom the results and discussion section?
Okay, ma'am. Ma'am, I have to go back because as the font size is little bit small, so
Okay.
I have to go by each and every point. So I'll go directly to result and discussion. Yes, ma'am. Can you see?
Yes.
Yes.
Can you go to the next slide?
Yes, ma'am. This was regarding docking studies. The docking studies was performed on Glide, which we have received from the Schrodinger software. The docking studies for coumarins and benzodiazepines were compared. Out of that, flumazenil gave us little bit good docking, but coumarins that we are proposing for a treatment for PCOS, which is giving us a very good docking scope.
Fine, fine. I just wanted to see the results of this. Fine.
Ma'am, can I show you the docking score also?
Fine, fine. No problem. Yeah. Thank you.
Yes, welcome.
So what have you concluded?
Ma'am, your voice is breaking. I'm not able to understand the question. Hello, am I audible? Hello, am I audible?
Yes, you are audible.
Yes, the conclusion is we have designed alpha-4 beta-3 delta isoform, and that we have designed with taking a reference of PDB-HUO, and from that we are going to check the interaction of the coumarins, how well they are getting fitted in the pocket for showing the greater interaction. So we are proposing coumarins as one of the treatment for PCOS. Because coumarins are easily synthesized, they are a natural molecule, so extraction can be done. For this, we have selected coumarins.
Okay, so
And
what is the rationale behind selecting coumarins? Is there any structural similarities which is supporting in this binding with the receptors?
Ma'am, it is showing a greater interaction. Coumarins we have selected because till now no molecular level study is done. This is the first study that we are going on the receptor level, and that too, it is the synthesis of the coumarins are little bit easy. But if we further skip the synthesis, we can just go and take herbal extraction, that is natural molecule. So extraction can be done, and it is more hydrophobic than benzodiazepines, which is serving our purpose of crossing the blood-brain barrier, as we are targeting GABA receptor.
Okay. Okay, fine.
PPSG1, please stop sharing your screen.
Okay. Okay. Thank you, ma'am.
Yeah, now I would like to call upon PPSG2. And before starting with PPSG2, I would like to tell everyone that please maintain the time limit. This is a poster presentation competition. So 2 minutes, in the 2 minutes, just present your highlights and don't go much deep. And also there are 3 minutes. 3 minutes are going to be there for Q&A session. So please maintain the time limit strictly, or else they will get disqualified. PPSG2 can start.
Very good morning, one and all. Myself, Atul. I am the dissertation. Today's presentation is a long-acting nanofiber of lutein for age-related macular degeneration. The age-related macular degeneration is a chronic disease. It is characterized by abnormal growth of new blood vessels. That means the angiogenesis in the retinal pigment epithelium. It causes the leakage of the blood vessels and disturb the retinal vision. Central visions. We can see that this is normal pictures, that normal vision, and in age-related macular degeneration, central vision is lost. This diagram indicates that the destruction of rod and cones, and this is a complete eye structures. For this, I have developed the nanofibers by using the electrospinning. Here is the experimental methods, the polymers and the drug dissolved into the solvent, then electrospun on the by using the electrospinning machines. Then the seed is cut into the appropriate size. And here we have I have discovered the new techniques, that is the nanofiber seed injected into the intravitreally through the 21st gauge needles into the directly into the vitreous chamber, that will be directly available into the vitreous chamber. This bypasses the all barriers present into the retinal regions. I have characterized for the scanning electron microscopy. This is found the 200 to 400 micrometer in diameter. Then drug DSC. Here DSC we can see the plain drug peak, then the polymer peak, and in case of nanofiber, the plain drug peak is disappear. Simultaneously, I have done the PXRD study. Then the FTIR study, the PXRD and FTIR study indicate that the drug that lutein encapsulated into the formulations, and we got the lutein loaded nanofibers. Here I have done the in vitro drug release study. It indicates that the plain drugs have, as compared to the plain drugs, the lutein nanofiber shows the sustained release for prolonged period. In conclusion, I can say that the fabricated lutein-loaded nanofiber will sustain the drug release for a long period in the eye, and it will be effective therapy for treatment of age-related macular degeneration. Here is the some references.
PPSG2, the time is up.
Yes.
We can start with the Q&A session, ma'am.
Yes, can I ask you questions here?
Yes, ma'am.
Yeah. How did you calculate the dose here?
Ma'am, actually, those calculations was done by the amount of required for the posterior segment. Here is the lutein is the carotenoid, which generally available into the back side of the eye. I have calculated the dose, calculated the drug loading efficiency, and after that amount of drug loading efficiency, from this I have dissolved that total nanofiber into the organic solvent and analyzed by the UV, and then I get the amount of drug present into the seed. On the basis of the total amount of drug present into the particular area of the seed, I have calculated the dose required for the for one dose, and then I have cut the nanofiber sheet accordingly.
So what was your polymer you have used here?
Here I have used the PCL 80,000 polycaprolactone having a molecular weight 80,000.
Okay, and what was your viscosity, the solution viscosity?
Ma'am, it's the solution viscosity is not actually measured, but it's easily electrospun.
Okay, so you have not done any process variables during your development trials?
Yes, I have done, but actually I have prepared the 20% PCL solutions. That's 20 gram of the PCLs into the 20 ml of DCM.
20 grams of PCL.
Oh, sorry, 2 gram of PCL.
Okay. Okay, and how much was the drug content uniformity what you have obtained?
Ma'am, I have got the 2 mg of the drug into the 4.5 mm of sheet. That's square.
Okay, and what was the thickness of your fiber?
Ma'am, I
sheet?
have did the it's in 200 to 400 nanometer in size.
Okay, and the thickness of the sheet, have you measured the thickness of the sheet, nanofiber sheet?
Yes, ma'am. It's 0.5 5 to 0.6 mm.
Okay. Okay, and what were the challenges you faced when you were preparing these nanofibers?
Hello.
What were the challenges you faced?
Ma'am, actually, this is um, uh, PCL having the also the good strength, but in case of the very thin layer, we need to the very proper handling for the nanofibers. We, the handling procedure, and I have mentioned there, I have inserted into the needle this insertions of nanofiber into the needle. It's not easier for us. We need to the develop the specific injector for this.
Okay. So your end product, how did you use it? Did you do any animal studies here?
Not yet, but I'm planning to do in next 1 or 2 months.
Okay. That's it. Thank you.
Thank you, ma'am.
Okay, I had one more question. Are there any regulatory requirements for the particle size for the ocular drug delivery systems?
Yes,
And are
ma'am.
you, yeah,
Yes, ma'am. But now, still date, there is a not specific regulatory authorities or regulatory specifications set for the nano formulations. We need to, as per guideline, we need to the give the some specific range for the or we need to the decide the specifications for the nano formulations.
and how will you decide what should be your optimum particle size range for the ocular drug delivery so that it doesn't inhibit the vision?
Ma'am, actually, the final formulation is injectable, like injectable implant, and it will be inserted through the intravitreal injection, as like to the currently marketed Vijay drug delivery system. I am trying to the follow the guidelines as per the intravitreal injection.
Okay, so yeah, what that is what. How will you decide what particle size you will consider as optimum? And I mean, anything that goes into the vitreous chamber should not inhibit your vision, right?
Yes, ma'am, but I'm targeting the less than 200 micrometer, sorry, nanometer particle size, and the devices will or the that nanofiber sits with having the small spore structure, which will allow the drug release with constant rate, and it not be release the burst or the show the burst release.
Okay. Thank you so much.
Thank you.
Okay, Atul, I have a one question. In your conclusion, you have mentioned that it will remain for longer period in the eye, the nanofiber which you have created. Okay, so can you just specify what duration will be there? The longer period means how many minutes or how many hours, like that?
No, no. Actually, ma'am, this age-related macular degeneration is a chronic disease. It requires the treatment throughout the life. The patient is not easily treatable or the curable. For particularly AMD, we need to the treatment up to the patient life. Hence, I am trying to target the this lutein in posterior segment of eye for the more as much as more, but still date the formulations available in the market is required monthly injections. I am trying to convert that monthly injections by developing such type of therapy up to the at least 3 months to 6 months.
Okay. Okay, fine.
Okay, thank you, ma'am.
Please call upon PPHD 3 now. PPHD 3.
Good morning, everyone. Am I audible?
Yes.
First of all, I would like to thank the institute, BNCP, for giving me this platform.
Your voice is breaking. PPHD 3, your voice is breaking.
Am I audible, ma'am? Hello.
Yes. Yes.
Video is there, ma'am? Video is visible, ma'am?
Yeah, you are visible.
Okay, okay. So I start with the topic that is nanobiotics, that is era of new antibiotics. Now we already have 100 of antibiotics on the desk, but why we need a new antibiotic, that is a nanobiotic in hand? Reason is that we have developed a resistance against the existing antibiotics, and the resistance has been developed because bacteria have acquired through evolution they have acquired this resistance, and because of the non-appropriate use of antibiotics, every third prescription we see there is an antibiotic prescribed, so that has resulting in the antimicrobial resistance enhancement, and therefore the lag of the discovery of new antibiotics also pushed this, that we should move forward towards the discovery of something else, that is a nanobiotic. So nanobiotic is a promoting technique, that is transformational therapeutic agents themselves into the nano-sized assemblies that can be done. That is, we develop a delivery system that is carrier free. And this review is here to provide the concise description of the characteristic what a nanoparticle should possess, so that it must be considered as a suitable antibacterial delivering agent. So starting with the left-hand side, there is a statistical data which is showing that how with years from 1985 to 2016, we find that the resistance is continuously increasing and the antibiotic discovery is continuously falling. One of the reasons of the fall in the discovery is that pharmaceutical companies do find an incentive or momentary benefit to discover a drug that is
Can't hear your voice. I can't hear you. No, your voice is not audible.
Hello.
Yeah.
I can hear you, but ma'am, all the time. Now I am audible, ma'am?
Yes, yes.
Okay,
Only
we are
15 seconds. Only 15 seconds remaining for you.
Okay, ma'am, here I have shown that how there is a tuning of antibiotics is possible because they have possessing a high degree of freedom in terms of shape, size, and charge. And moreover, talking about the mode of action, there are a number of mode of action. They can show the effect. They can cause membrane damage by release of ROS, that is reactive oxygen species, then RNS, that is reactive nitrogen species, nitric oxide species. Then they find applications in nano-X-ray therapy.
Your time is up. Your time is up. Now time for questions. Judges, you can ask the question.
Yeah. Can you quickly tell us the relation with regard to the resistance which would be provided with this new concept?
Actually, these are designed in such a way that the bacteria has to undergo multiple evolutions to develop resistance against that. Right. So therefore, they are we can say they are resistant to resistance that is being developed in the bacteria, which is not seen in conventional therapy.
Okay. Any toxicity data you have searched on and anything
yes,
you have found?
Yes, ma'am. I have already searched various journals. I have seen that already silver-linked nanoparticles, which have antimicrobial activity and which are synthesized to biological route. That is, they are inducing toxicity even in a single dose. They are there. So that's one of the challenge that we have to manage while we are making them on a larger scale. I have shown in the last portion of the slide that no doubt antibiotic providing promising future, but yet challenges are, for example, biocompatibility and safety. This is one of the second important challenge that we can say when we preclinical evaluation for the toxicity studies, because much more study in the regard to pharmacodynamic and pharmacokinetic is yet to be done. We do not know much about the stability, physical and chemical, much about the you can say the how it is interacting with the tissues, because it is targeting the tumor cells, so it is accumulating there, no doubt. But there are chances that it can also accumulate into it can also go into the surrounding tissues, and once it goes, it will definitely be fatal. So that's why much more research has to be done into this regard. And I have also shown here the clinical trials which are in pipeline. Right, this is the data from the latest journal, that is 2020 I have got, and there are some drugs, anti-ciprofloxacin, amikacin is there. So these are in the phase 3, phase 2 clinical trials. And moreover, we can say that here the studies in the amikacin phase 3 clinical trials, 36 patients have been used, and in phase 2, 30 patients were used. They are still on, you can say, more exploration is required, and there are 3 parameters we can say. First is the instability issues, biostability issues, biocompatibility issues. Second is toxicological parameter, which is a challenge. Third major challenge is, ma'am, we have no regulatory guidelines. No regulatory guideline is there, and no protocol is there to ensure the GMP and characterization studies of them. So that is much more needed to be explored.
Yeah, my concern was also the same. You're talking about all amikacin and everything which is being inhaled. So these are all very large molecules. What about
Yes,
the clearance of these from the lungs?
Yes,
so
ma'am.
precisely what is I was concerned about, the toxicity.
Yes,
and
that is the basic thing, ma'am. That is the basic thing. Much more need to be done in this regard. Hello, am I audible? Hello.
Yes, you are audible. Yes, you are audible. Good presentation.
Thank you so much, ma'am.
Good.
Thank you so much, ma'am. Anything else, ma'am? I should explain. Hello.
We would start with the next participant now.
Thank you so much.
PPG1.
Yes, am I audible, ma'am?
Yes. You can start now.
Okay, yes, ma'am. Screen visible? Excuse me. Hello.
Yeah.
Is my screen visible?
You don't have to share the screen. It is visible. It is visible.
Okay. Yes. So, good morning, everyone. My title of the poster is Assessment of Effect of Food on Dissolution Profile of Paracetamol Tablet by In Vitro Method. So, delivering a drug by oral route it is considered to be one of the most favored route of drug administration, and people often consume the OTC drug at any time before and after the breakfast with water or any other available hot or cold beverages. So however this breakfast interact with the drug which affect the release of drug, and taking this into the consideration, this current study was planned to assess the effect of some most common Indian breakfast on the release of drug. So by using USP type 2 paddle dissolution apparatus in which dissolution media is used, was modified phosphate buffer along with its pH maintained at 5.8. So during the research and development stage, a disintegration and dissolution of the tablet and the dosage type is studied in either water or stimulated gastric medium, with the stimulated gas intestinal medium which is being used occasionally. The evolutionary parameters of a tablet is also studied, that is the weight variation, fiability test, disintegration and hardness test. Then there is a preparation of the phosphate buffer. Phosphate buffer is prepared by using potassium dihydrogen phosphate and NaOH, to get the approximate pH of 5.8, and this phosphate buffer is used as a dissolution media for the study. Then the calibrations curve, the standard calibration standard solution was prepared by the paracetamol drug, which was dissolved in 50 ml of distilled water, and dilutions were taken as per the requirement. So the dissolution study was done by using rotating paddle apparatus in which the phosphate buffer of pH 5.8 was taken along with the said dosa, which was
Given your
taken
time
and make
is
a
up.
press using.
PPG, when your time is up. Time for question and answers, ma'am. Judges, please.
Yeah, why you have selected, have you selected any particular breakfast for your study and how you have done it?
Yes, ma'am. We have breakfast like poha, upma, shira, idli, sambar. Then dosa were used for this study, and dissolution study was done using this breakfast and along with the phosphate buffer, which was used as a media of pH 5.8. And the samples were then samples from the dissolution were analyzed under HPLC and UV.
Okay, so have you included this breakfast in your study and what is the process?
Yes. Come again, ma'am.
How you have included this breakfast in your dissolution study and what is its effect?
Actually, dissolution study, it is like we make a paste of particular breakfast and taken as a, suppose 250 ml of fresh dosa paste as a sample, which was added in 650 ml of phosphate buffer, and make up the volume up to 900 ml. Then temperature is maintained at a 37 degrees Celsius, and the RPM is set up to 50 RPM. Then this way we have analyzed its results under UV and HPLC.
Okay, so what is the benefit of doing this type of study? Because paracetamol is a very common drug. Is it not good to change the time rather than taking morning? We can take it in the evening or afternoon. No? And this breakfast is not limited. There are multiple options for breakfast. So what are the benefit of doing this type of study?
And usually this area is very neglected, and people broadly consumes the OTC drug before or after the breakfast. So the main intention
We've
of
lost you.
this study. Hello, am I audible?
Yes, yes.
The main intention of this study is to determine the precise interaction between the contents present in the breakfast and the paracetamol.
See, you have mentioned over here something about beverages also. You've studied only breakfast or beverages also you
Yeah,
have studied?
we have also studied beverages, which includes along with water, milk, tea, then hot and cold coffee, and also some cold drinks we have studied.
Okay, so what have you concluded finally with this study? What did you arrive at?
Yeah, we concluded that usually this drug paracetamol is broadly used as a OTC drug, and it should be taken with water instead of any other breakfast to get a better therapeutic effect. And paracetamol might have any chemical interaction with content which are present in these breakfast and other beverages which usually taken by the people. And it is also observed that when paracetamol is taken with water, maximum drug dissolution takes place as compared to other breakfasts, and the minimum dissolution of drug is obtained in say upma if they are compared with each other.
See, but it is also an NSAID, right? You can't take it on an empty stomach with plain water, right? You need to have breakfast with it.
Yes. So for that, we in our study we have concluded that the drug releasing poha was observed at observed in high percentage, that is 104% at a 120 minute after taking breakfast. So the dissolution hello.
All right.
Yes, ma'am. Anything else?
Yeah, can I ask here? Yeah, the same question.
Yes, ma'am.
What was your main objective of taking paracetamol as the drug? Because there are a lot many studies which have been explored, simulating all these conditions in presence of dietary fibers, various type of fats. So, what is that new thing you wanted to explore in your studies?
Ma'am, basically, as I mentioned earlier, paracetamol is broadly used and people consume it for the various reasons, like as a painkiller, antipyretic, analgesic. So it is most commonly used drug. So that's the reason I have taken paracetamol as a drug for my study.
So when you were doing your literature search, you could not find any data with regard to paracetamol and the effect of food after the consumptions.
Actually, our intention is to study the drug paracetamol. We have taken the brand crocin, which is most commonly used.
Okay.
So drug was paracetamol itself.
Got it. Yeah. Thank you so much. Yeah.
Thank you, ma'am. Thank you.
Okay, thank you, ma'am. We'll start with PPG 2.
Hello.
Yeah, you're audible.
Are you sharing or may I share my screen?
We'll share.
Okay. So good morning to one and all. My topic for today is analytical method development and validation by HPLC. So coming on to objective, the objective of the study was to develop an simple, accurate and stability indicating reverse phase method for the drug A and its impurities, and to validate it afterwards by following the ICH guidelines. Rationale of the study was there was no official method for this compound A or drug A, so there was a need to develop an method for such. So coming on to system, the analysis was performed on water system with the 2695 separation module and a PDA detector. The wavelength was determined by spiking the sample by using a sample, and spikes was injected. From the chromatogram obtained, it was observed that 286 nanometer is optimum for the determination of all impurities as well as compound A. Then for the first trial, we used the mobile phase was OPA 88% of OPA with the pH 7. And the mobile phase B was used a mixture of acetonitrile and water in the ratio of 80 is to 20. The observation was no separation was observed between the peak and unknown impurities. Tailing in the main peak and the main peak retention time shifted to 30 minutes. Then for the second trial, we used the same buffer, but we changed the pH to 7.23.
Your time is up. Judges, please ask questions.
I just have one suggestion. When you are presenting such type of posters, rather than putting tables, because it is not readable only.
Actually, ma'am,
chromatograph and all that.
I shared the other poster also, but they have not like put my original poster here. I have put the chromatograms.
Okay.
Can I share it for a second? Hello?
Yes.
Yeah, share it.
Okay. Is it visible, ma'am?
Still, the content is too much on the poster. But yeah,
It's
yeah, it's very difficult to read.
very difficult to interpret what you have tried to portray.
Okay.
Yeah, so because it's not readable, as ma'am is saying, no, it's very tiny. We can't read a word what you have written. Yeah. So here, what are the validation parameters that you have done? All of them.
Specificity, precision, accuracy, linearity, stability, and analytical solution. And also force regression studies were done for the same.
Okay. Force degradation studies were done with
Thermal, photolytic, oxidative alkali, and acid degradation.
and how did you perform the photolytic degradation studies?
It was exposed under UV at 254 nanometer for 24 hours, and the first degradation was done like this, and the second was done like exposed between 400 nanometer to 800 nanometer in sun test apparatus at 55 kilolux for 24 hours.
Okay.
Should I stop sharing?
Yes.
Okay, thank you.
Yeah, I have one question here.
Yes, ma'am.
Yeah. With regard to the choice of buffer, okay,
Yes.
how did you understand that you have to start working with pH 6.8 or 7? I could not really read your.
We started with 7 pH, that is neutral, so that we can determine the what effect it gives. Then we started shifting it towards basic and then acidic. According to the chromatograms we get.
So when you develop any analytical method, is that the rule that you have to always start with pH 7?
No, it's not necessary, but it's according to the compound pH, whether it is acidic or basic.
So what property of the drug will you look into? What is that property? Okay, no problem. And in few of your initial chromatograms, you had not spiked impurity, is it?
Ma'am, they are spiked. They are known and unknown impurities both.
So even the initial chromatograms had the spiked impurities?
Yes.
Okay. Because the intensity, the absorbance were not that sharp, whereas
Yes, the response was very low, and because of the pH and the flow rate and also other parameters, the peak shape was not good, and unknown impurities were not separated from each other.
okay, okay, fine. Thank you.
Yeah, thank you, ma'am.
Yeah, thank you. PPG3, we'll now start with PPG3. You can start now.
Yeah. Am I audible?
Yes, you are.
Thank you. A very good morning to one and all present here. The topic of my presentation is anti-inflammatory polymeric nasal film to treat COVID-19 early stage infection. As we all are very well aware that COVID-19 infection is caused by SARS-CoV-2 virus and the infection starts in the nasal cavity. The coronavirus contains big spike protein and RBD protein for cellular attachment and subsequent growth. The virus growth triggers strong inflammation and nasal mucosal destruction with the release of huge cytokines which enter the circulation, and it causes immune stress. With the innovation of glycerol-based osmotic filmogen solution, that is COVID spray, early stage of COVID-19 can be prevented as it is an antiviral barrier containing glycerol, gelifying agents, and an association of plant polymers. When COVID spray is applied on the nasal mucosa, it forms instantly a stable osmotic film. The gelifying agents in the film swells when it comes in contact with water, and it attracts the hypotonic liquid from the nasal cavity, and thus it will drain all the contaminants present in it, and it will neutralize the spike S1 and RBD protein, and therefore it will not infect new healthy cells. The clinical study of COVID spray was carried out for checking its efficacy, and it was seen that the symptoms were stabilized just after the first application and improved progressively. The results clearly show that COVID spray can stop the development of severe respiratory symptoms without any undesired effects, provided that the treatment is taken as preventive treatment. So the main objective behind this innovation was to minimize the nasal surface concentration of pro-inflammatory cytokines, and it was found to be instant, simple, safe approach that helped to attenuate COVID-19 induced systemic inflammation at an early stage. Thank you.
Jay, over to you.
Can you comment on the dose? Design of the device and the dose calculation? We cannot hear you.
PPG3, you are not audible.
Hello, ma'am.
Yes, now.
Okay. So it is a 15 ml bottle, and 3 to 4 times a day the patient can spray.
So can you comment on the spray rate and the volume? How will the dose be calculated? The pattern? Any idea with regard, say, suppose you want to develop such kind of sprays in your lab, how would you study on that?
Ma'am, actually, it's a review poster, so they have not mentioned the spray rate in that.
All right.
No problem.
Okay.
Thank you.
So, and what do you think if you're going to use it for a preventive this thing? How long should I use it? Lifelong, or in certain seasons, or how is it going to be? You're saying it's for a preventive
Ma'am,
measure,
because
right?
yes, ma'am. So as a preventive measure, so as soon as the clinical symptoms appear, so we can start using this nasal spray. So it's not a lifelong thing, because if we are using the spray for a particular say for 3 days, so this particular one spray has a long lasting life of 4 to 6 hours. So once we come to know that the symptoms have reduced because of this, then we can stop using it.
But once the clinical symptoms develop, that means the person is already infected.
Yes, ma'am, but it will not severe the condition of the patient. We will come to know in the initial stage itself, and it will not invade the lungs and then make it more severe. We can prevent the severity of the infection.
Okay, so you mean to say it is not for the prophylactic purpose. Once we are getting the symptoms, early symptoms, then it is used, right?
Yes, ma'am.
Okay, so how many time that spraying is done? In that study, they have mentioned anything about the durations, how many times a day?
That to use it for 3 to 4 times a day.
And they have mentioned one spray, how much amount of the drug?
A single spray 3 to 4 times a day.
No,
They
no,
have not.
the dose of the drug, the anti-inflammatory drug in a one spray. How much amount will?
No, ma'am.
Okay. Okay.
Thank you.
Thank you.
Yeah, we'll move ahead. PPG 4. PPG 4.
Yes, ma'am.
Yeah. You can start. Yeah, you can start.
Yes. Good morning, one and all. The topic of my poster is software-based design, development, characterization, and preclinical evaluation of nelfinavir-loaded nanostructured lipid carriers for oral delivery. So in this, the nelfinavir is the non-peptide protease inhibitor, which is used in treatment of HIV/AIDS, along with the other other drugs like NRTIs like zidovudin, lamivudin. So the disadvantage of the nelfinavir it is that it has low solubility and it undergoes a significant first pass metabolism. Thus, to improve its oral bioavailability, we have converted it into a nanostructured lipid carriers to increase the oral bioavailability. So in this, the solid lipid and liquid lipid used is oleic acid and GMS. It is melted and the drug is added to it. And the dispersion is then dispersed in the aqueous phase, which is then introduced into the hot high pressurized homogenizer to form the NFB loaded NLC dispersion. Then it is evaluated for the mean particle size, polydispersibility index, and further it is underwent through preclinical trials using male booster rats. And the results are obtained. The optimized batch was found to be F4 batch among the 9 batches. And the design of expert software version 8 was used for optimization, and the results were obtained. And it was concluded from the in vivo studies that the results developed optimal NFB NLCs, produced extensively improved oral viability, and which was found to be almost 2.5 times than the available nelfinavir tablets.
Your time is up.
And also, okay, thank you.
Yeah, forward to you, judges.
What was your yield?
Yes, ma'am.
Yield.
It was obtained about 10 milliliters. It was not actually the specific amount taken, so it was evaluated for up to 10 milliliters.
Okay, and what was your main objective of applying this DOE, and what was your conclusion?
We applied the DOE to optimize the batch of formulating the nanostructured lipid carriers. It was concluded that the batch 4, that is the F4 batch from the DOE, uh, it it had the mean the optimum PDI zeta potential and entrapment efficiency was also higher, and also the in vivo studies and drug release also was found to be optimum in the F4 batch.
So what was the variables you had applied?
Yes.
Variables were the mean particle size, zeta potential, and entrapment efficiency.
What are the reason behind you selecting Polaxomer 407?
Polaxomer is used as the surfactant.
Oh, thank you.
Okay, so when you say software-based design, it means use of DOE.
Yes, ma'am.
Is that what you mean? Okay, and so you have applied this design of experiments before you started the experiment, or after conducting a few trial and errors, and after that you have applied DOE, and then
Actually, it was done side by side. Like optimization was also carried out, and the evaluations also were carried out.
no, you didn't get my point. So when you apply design of experiments, do you do it before you start with your experiments, or you do it in between after you have performed a few experiments, then you plan your remaining experiments based on the design of experiments?
Uh, it was done. It was started before the evaluation and preparation, and then after each and every step, it was again considered to it was again applied. So to get optimized batch.
So you have okay. All right, and I just, as Madam said, what were the variables you said? Your variables were
Mean particle size, zeta potential, and entrapment efficiency.
entrancement efficiency. Okay, and just let me have a look at the results. Zeta potential.
Can you name the software?
Yeah.
Design of experiment.
No, name of software, like, is was it Design Expert, or was it some
Yes,
other?
design expert version 8.
Okay, we'll move ahead. Thank you, ma'am. PPG5.
Hello, ma'am. Am I audible?
Yes, you are audible.
So my topic for the day is advancements in ionic liquids in pharmaceutical industry. So basically, IS are molten salts of cationic organic part and anionic inorganic or organic part. Uh, they are in liquid state and having the melting point below 100 degrees Celsius. They are having high thermal chemical stability and uh, high solution ability. They have varied applications in API synthesis, in crystallization, in biological activity. Also helps in drug delivery system and drug formulations. So their characteristics are that they are non-toxic. They have no vapor pressure. They have higher stability and they are non-volatile in nature. They can be reused and recycled, which makes it a green method and a sustainable option. Hence, name as green solvent or green chemical. It has application in synthesis in the form of catalyst, reagents, or reaction media. Its application is being used in various syntheses. For example, paracetamol, naproxen, ibuprofen have been developed by this method. The reactions are faster. Biological activity due to its structure, uh, cation and anionic structure, they have and a long alkyl chain. They have its activity as antimicrobial, anti-tumor, and in antioxidants. Due to its high solution ability and uh, its permeation enhancing technique. It is having a variety of application as drug delivery system, for example, ionogels, membranes, nanofibers,
PPG5, your
vesicles.
time is up. Over to you, judges.
Okay, did you find any papers which spoke about the biomedical applications? Or because this has a good property of high thermal chemical stability. So did you find any papers which spoke about its biomedical application?
So I found various papers regarding its application as antibacterial and anti-tumor. For example, majorly I found anti-tumor because it is anionic in nature. Ionic in nature, it can be combined with PDT and heat using ultraviolet rays. So it has anti-tumor activity. That was a paper I found up.
Okay, in biomedical or medical devices, any papers?
It has uh, cells have been developed
Okay, okay,
in
okay. Thank
medical
you.
devices.
So you mean to say it is still at the study level, right?
Yes, ma'am. So green chemistry is prevalent. It has been used commercially. The crystallization is also used. For example, I have seen papers for uh, selective crystallization of gabapentalin. Also, synthesis of paracetamol and ibuprofen have been studied. Uh, Drug delivery and its biological activities, it is in the nascent stages.
Okay, okay, good.
Yes, ma'am.
Okay, I have no questions.
Thank you, ma'am.
Yeah, thank you. We'll go ahead. PPG6.
Yes, I'm here. Hello, am I audible?
Yes, you're audible. Just a minute.
Should I share the screen?
No, we'll share. Just a minute.
Okay, sure. Good afternoon, everyone present here. My topic for today's presentation is brain-targeted drug delivery system for Parkinson disorder, development of levodopa niacin co-loaded with antioxidant. As we know, Parkinson disease is a progressive neurological disorder, and it's the second most common neurodegenerative disease. Levodopa is the golden standard for medication for Parkinson disease, but it has some side effects. After long-term use of levodopa, it has on and off effect. To minimize these side effects, we propose a combination approach for levodopa niacin co-loaded with antioxidant. As represented in table 1, any antioxidant known to have a neuroprotective effect in vivo model, but they are failing to reproduce this effect in clinical trial. Therefore, we propose to give this combination niacin in intranasal drug delivery system. The direct transport of drug into the nose to the brain through olfactory and trigeminal nerve system. Through nose, they can bypass the blood-brain barrier and directly target to the brain. This is an excellent approach to avoid the conventional dosage form. In the further flowchart, we have explained the different types of niacin preparation and its characterization. Our rationale for this project is to effective control of wearing off effect of levodopa. This combination will help in slow down the neurodegeneration as it has an antioxidant effect, so it will give alternative neuroprotective effect. The effect of brain targeting via nasal spray, which will avail the antioxidant rich in the brain itself. The direct brain target can eliminate the use of carbidopa as well, since we are targeting brain levodopa directly into the brain. Thank you.
Yes, over to you, judges.
Okay, you're talking about levodopa directly to the brain, and what antioxidant have you co-loaded it with?
Yes, ma'am. Combination of levodopa and antioxidant in a niacin formulation and converting into a nasal drug delivery system.
What was the antioxidant that you have used?
We haven't used any antioxidant. In a literature survey, we come across there are many antioxidants which are showing neuroprotective effect in Parkinson, but the effects are not reproducible. In clinical trial, they are failing. So, in the literature survey, it has mentioned that brain targeting could enhance the antioxidant level in the brain and give the positive results for anti-Parkinson.
Okay, one question from my side. Can you differentiate
Yes, ma'am.
what are those data you will obtain when you measure the size parameter through SEM and AFM? What are the additional measurement or characterization will you obtain?
Ma'am, for characterization, we will use particle size of niosome, their vesicle size.
Correct. So how do I make a decision? Do I go with SEM or AFM? Are there any additional information I can obtain from these two different instruments?
No, ma'am, we are going for SEM and AFM only.
But why?
And actually, this a proposal. We haven't started any work on it. This a proposal that we can do it.
Okay, fine.
Yes. Thank
So,
you very much, ma'am.
Yeah, so you are going to work on this. You haven't started this work, right?
Yes, ma'am. This is a literature survey we come across, and we found that antioxidants are showing very good effect in vivo in preclinical study, and they are failing in clinical trials. So we are just proposing this can be effective.
So you are going to include antioxidants in your study.
Yes.
Along with your levodopa.
Yes, ma'am.
Okay.
Okay, we'll go ahead. If no questions,
you.
then we'll go ahead. PPG 7. PPG 7. PPG 7, are you there? PPG 8, we'll go ahead with PPG 8. PPG 8, are you ready? PPG 9, are you ready?
Yes, ma'am, yes.
Okay, we'll start with 9.
Sure, ma'am.
Just a minute. Yeah, PPG 9, you can start.
Okay, ma'am. Okay. Hi, I'm Abhijit Thorat and I'm presenting my poster on preparation and evaluation for polyacromer TPGS loaded micelles of sorafenib to treat the advanced labial solar carcinoma. Generally, micelles are the unique self-assembled core shell structure which enables the incorporation of a poorly water-soluble drug into an inner hydrophobic core, thus improving the stability, solubility, and viability. Mixed micelles are of growing interest of formulation as a yield good drug stabilization, high drug loading, small cellular carriers, high stability, extended circulation, and better tissue targeting. P-glycoprotein inhibition, extended drug release, less systemic side effects, hypersensitization towards the multi-drug resistant cells, and subcellular localized properties. Here we can see the micelles can be considered as the promising carrier for solubilizing hydrophobic drugs since nanomicelles are amphiphilic molecules, the hydrophobic core, and accommodate the hydrophobic drug and corona being highly hydrophobic, produce a clear aqueous solution. Mixed micelles could increase the stability, improve the drug loading and encapsulation efficiency compared to the single polymeric micelles. The presence of the lipophilic core increases the solubility of a poor water soluble molecules and offer a possibility to obtain the controlled drug release. The small particle size prolongs the residence time in the blood circulation by passing the liver and spleen filtration and glomerular elimination and enhancing the cellular uptake and ability to cross the epithelial barrier. All these aspects results in increased the drug viability. Thus, the TPGS micelles formulation may also achieve the synergistic effects with the formulated drug. Actually, ma'am, I'm using here a TPGS being a polymer which acts as an anti-cancer drug. It's a newly detected polymer which is showing anti-cancer effect and also acting as a surface active agent. We can see here much more benefits or applications like stimuli response nanocarrier for drug and gene delivery. It is used for the ocular delivery, oral delivery, gene delivery also. And just before this, there are many more works done on micelles, but by using a thin film hydration method. I have used here the ethanol injection method and having a particle size range of
Your time
16
is up. TPG9, your time is up. Over to you, judges.
Okay, PG9.
Yes, ma'am.
Can I know which of collapsomer have you used?
Actually, ma'am, I'm getting your blood voice.
The polymer grade and what was the viscosity?
Good.
Hello?
Hello. Am I audible to all?
Yeah, yes, your voice is breaking. May I know the grade? Your polaxomer grade.
Hello.
Am I audible to you?
Yes, ma'am. Yes, your voice is breaking a little bit.
Yeah. May I know the grade? Polaxomer grade.
Polaxomer grade. Okay. Actually, ma'am, it's Polaxomer 4000. Only for Polaxomer 4000.
4000.
Yes,
what
ma'am.
is that 4000 indicating?
It's the molecular weight of that Polaxomer, ma'am. 4000 is indicated. Molecular.
Okay. Okay. Now, can you comment on your release profile?
Yes, ma'am.
You have given x-axis. I guess it's not visible. Somewhere it looks like 200 minutes.
Actually, ma'am, it releases 300 minutes. I have shown here. It releases 95% within 5 hours. At 5 hours, I can say.
So is it justified with your choice of the polymer?
Yes, ma'am.
Okay, okay, fine. Thank you. Please cross-check the grade of your polymer, okay?
Okay, sure.
मुझे उन्हें ड्यूटी डाला था नहीं मैं प्रिंसिपल से
Acha, you are talking about this TPGS as a polymer or that itself is having anti-cancer activity?
Both activities it's showing, ma'am, and also it is showing the surface active agent activity, solubilizing activity also.
All right.
That's why, ma'am, I have used this. TPGS is recently developed for multi-drug resistance and also having a good polymeric action.
All right. So, and one more thing I wanted to ask you: why have you specifically mentioned advanced hepatocellular carcinoma? Is there something that you are doing to target to the hepatocellular carcinoma?
Sure, ma'am. Sure. Actually, ma'am, I have given my sample for cell line study, but still I don't have got any result. And I have mentioned here the advanced hepatocellular carcinoma just because, ma'am, my drug sorafenib showing the activity as a multi-kinase inhibitor. It's showing the also activity of the anti-angiogenesis. It's showing the activity on apoptosis, cell apoptosis, and also it's showing the activity of anti-proliferative activity. And
So,
the polymer I
tell
have
me,
used,
just a minute. So, can you not say simply carcinoma? Why specifically hepatocellular? Unless you are targeting to the liver, I think it should be in general carcinoma.
What, ma'am? Can you say again?
Are you targeting it to the liver?
Yes, yes.
How?
I'm targeting this. Okay. By tyrosine kinase and RFS kinase, serine and threonine kinase, ma'am. Through this media, cytochrome P450.
Okay. All right.
We'll continue. We'll go ahead. PPG, thank you so much. PPG 8.
Thank you. Thank you so much, ma'am. Thank you.
Ma'am, please note this was PPG 9. We had skipped PPG 7 due to some network issues and also PPG 8. Now we'll go back to PPG 8. This was PPG 9 and now we'll go back to PPG 8. PPG 8. Can't hear you, PPG 8. We'll go ahead. PPG 8, you are not audible. We'll go ahead. We'll go ahead with next PPG 10. Are you there? PPG 10.
Yes, I'm here.
Yeah, okay, we'll start with
Uh, can you give me just 1 minute? I'm trying to switch on my video.
Yeah, sure.
Yeah. I hope I'm visible.
No, yeah, you are visible.
Yeah. Okay, I'll start now. The topic for my poster is development and evaluation of a pulsatile drug release system for management of diabetes mellitus, and my code is PPG10. So the drug which was selected here was vildagliptin. It was selected because of its short half-life, and the conventional drug has to be taken 2 times a day. So our project focused on making a formulation which would be taken only once a day by formulating it as a as a capsule which will contain immediate release granule and a mini tablet which would be coated with a lag time coat and a pH dependent coat. For developing the granules, different batches were optimized with different types of binders. And as we can see in the figure 1 of the results, the batch containing D4, that is PVP K30, was in the amount of 10 mg was selected as it showed the release of 92 to 93% in 5 minutes, which was our desired immediate release time. Then the two different types of capsules, that is gelatin and HPMC, were tested for the immediate release granules. And as we can see in figure 4, the gelatin capsule showed 91% of release in 5 minutes, which was desired for the immediate release time. For the core tablet, which was direct compression, 5 different batches were made using different kinds of disintegrating agents, out of which the 5th batch was selected, which contained cross povidone in the concentration of 3 mg, as it shown the disintegration time of 1.07 minute, which was the lowest as compared to other batches. The lag coat was made of ethyl cellulose 20p, which was the hydrophobic one, and HPMC K4M, which was the hydrophilic one. And as we can see in figure 2, the factors and variables were directly proportional. That is, amount of ethyl cellulose and percent weight gain of the tablet was directly proportional to the increase of lag time. Therefore, out of the 9 batches, L6 and L8 was selected as it shown the time release required. These batches were further taken for the pH coating, which was Utrazid S100 with varying weight gain concentration of the coat, that is 5 and 10%. As we can see in figure 3, F2 was the one which showed the desired release pattern as immediate release of the drug observed
Your
after
time is
8
up.
to
PPG 10, your time is up. Over
10
to judges.
Okay.
Over to you, judges.
Can you quickly tell me the objective of selecting pulsatile and diabetic?
So, ma'am, pulsatile, we wanted to create pulsatile system because diabetic drug vildagliptin. Usually, we know that diabetic drugs we have to take 2 times a day. So, our objective was to create a patient compliance formulation which would be given just once a day, and it would mimic the second dose which was taken after taking our meals. So this would contain immediate release granules which would immediately release after intake of the capsule, and it contains a mini tablet which are given the coat so that when it reaches the colonic system after 8 to 10 hours, it will release the second dose, and it will mimic the second dose which we take conventionally for the second time the day.
Okay, there is something called dawn and dawn effect, which is generally observed in diabetic. Okay, so how do you think that this kind of system will influence it? You know the Down syndrome?
Hello?
Hello? Sorry, you understand the Down phenomena which occurs in diabetics?
No, ma'am, I'll have to look through that. I'll have to see that.
So there are two types of phenomena, the Somogi and Dawn phenomena. Okay, just study that because this is something very, very important when you think about giving it in a pulsatile release.
Okay.
There is a change in the blood sugar level during midnight around 2 to 3 o'clock and early in the morning.
Okay.
Okay. So
yeah.
Study that and see the effect of this drug.
Okay. Okay. Sure, ma'am. I will go through that.
Okay.
Yeah. Thank you so much.
Any more questions, judges? Okay, we'll move ahead.
No, please move ahead.
Okay, thank you.
PPG 8, are you there?
Yes, I'm audible now.
Yes, you are.
Okay.
Judges, please note this is PPG 8.
So good afternoon, everyone. Yes. So good afternoon, everyone. My topic for the poster is antipsychotic effects of metoclopramide in zebrafish via long-term L-methionine exposure. So schizophrenia, it is a severely debilitating lifelong psychiatric disorder which affects approximately 1% of the population, and its pathobiology is also poorly understood. So there's a need for further translational research in this field. So in this research, we have taken zebrafish as the model, and we have investigated the antipsychotic effects of metoclopramide in zebrafish. So for this, the tests which were done, the behavioral tests were novel tank diving test, social interaction test, and T-maze test. And the in vitro studies which were done were estimation of neurotransmitter serotonin using LCMS. So after doing the study, we found out that long-term L-methionine exposure showed the changes in behavioral patterns which causes schizophrenia-like behaviors. And we observed changes in locomotion, anxiety-like behavior, memory, and neurotransmitter levels after long-term exposure to L-methionine. And the metoclopramide treated group showed antipsychotic effects in this novel tank diving test, social interaction test, and the T-maze test. And we also found out that the serotonin level was less in the metoclopramide treated group as compared to the methionine group. So from this study, we conclude that the metoclopramide has antipsychotic effects. And we can also that the antipsychotic properties of metoclopramide has been shown by inhibiting serotonin levels in zebrafish brain at high doses. So this was the gist of my study.
Over to you, judges.
Yeah, so how you decided the dose for the study?
Ma'am, we did the pilot study first before that. So from that, we took highest dose of 4,000 μm micromolar. And from then we came down. So from that, we selected 3 doses, like 750 μm, 1000 μm, and 1250 μm.
Okay.
So we found out that 1250 is more effective.
Okay, so that 4,000 you have started for pilot study. Is there any reference how you have selected 4,000 only? Is there any reference?
No, and there is no dose reference, but we studied, so we took the highest possible dose because in 4,000 it showed side effect, like it showed the symptoms of muscular dystrophy, like it was not for like it were dying and it was not showing a proper effect as we wanted. It was showing the side effects.
Is there any correlation you have checked for human dose and zebrafish dose, like calculation?
Yes, ma'am. Like here we have taken the doses in range of micromolars, and in human dose it is in mg. So we have converted that to zebrafish. The calculations were back calculations were done, and then we came out. We found out we can start with 4,000 μm, and then we came down to 1,750 and 1,250 μm.
Okay.
No questions from my side.
Okay, moving forward. PPG 11.
Hello, am I audible?
Yes, you are audible. Just a minute.
Yes. So good afternoon, one and all. My topic for today is development of biomimetic soft magnetic nanorobots for removal of kidney stones, that is renal calculus. Nanorobotics is an emerging technology field, creating machines or robots whose components are at or near the scale of a nanometer. So more specifically, nanorobotics refers to the technology of engineering discipline that constructs nanoscale or molecular components. The potential uses of nanorobotics in medicines include early diagnosis and targeted drug delivery for cancer, monitoring of diabetes and healthcare. So future medical nanotechnology is expected to employ nanorobots that will be injected into the patient to perform work at a cellular level. Some research work have explored for the soft surgical nanorobots that could navigate inside the human ureter via crawling or rolling mechanism. And it is proposed to provide a novel and minimally invasive method for removal of kidney stones. It has been inspired from the soft body creatures such as jellyfishes. These soft surgical nanorobots are unique since they are able to move easily in the physiological environment inside the human body. So these nanorobots can easily transit from swimming through liquids to moving on solid surfaces without any physical intervention. That's it.
Yeah, no question from my side.
Is something like this there in the market, or this is just a theory which you have come across and presented?
Ma'am, still research is going on.
Okay, so is there anything or nothing is there right now currently? Some such
No,
nanorobots?
No question from my side.
No, yeah, please, we'll move ahead.
Thank you.
Yeah, we'll go ahead. PPG 12.
Yes.
Yeah, we'll go ahead. Just a minute.
Am I audible?
Yes, you are.
So for the topic, my topic is a novel clean chemistry approach, selective halodecarboxylation of vinyl carboxylic acid for the synthesis of beta-bromostyrene by using catalytic amount of calcium nitrate along with the use of potassium bromide as the source of bromine and glacial acetic acid as the solvent media by using microwave as an energy, providing source for the reaction to occur. The product of these was successfully obtained in a good yield. There were several methods reported for selective decarboxylation followed by halogenation by using different reaction conditions. A very well-known named method to synthesize this compound was Hunsdicker reaction.
But as it was a conventional method, there were various disadvantages of these reactions. Whereas in our novel method, by taking into the account of these green chemistry principles, our research applies to many of these principles, such as using a green solvent such as glacial acetic acid, microwave as green source of energy, and KBr as a bromine source. This reaction can also tolerate the electron donating group on the aromatic ring, and also we have less reaction time. For the main reaction, we performed various trial and error method prior and optimized the reaction conditions and monitored this reaction using TLC at different time points. The TLC shown here is of 5 minutes, giving the maximum product. We optimized the procedure as well, which included formation of product, separation of the product, and evaporation of the solvent. And the beta bromostyrene was analyzed by using HNMR. We also obtained a product using various derivatives such as 2-chlorocinemic acid, 2-nitro, and 4-methylcinemic acid. Here you can see the proposed mechanism of reaction, which follows the two pathway. And for the conclusion, the beta bromostyrene, a stable product, was obtained by a regioselective attack using the green chemistry method, and the reaction can further be modified using halogens. Yes, thank you.
Over to you, judges.
Yeah, nice work. May I know what was your batch size?
Ma'am, the yield which we got was, so here like the quantity taken was in 0.5 grams, and the yield we got was 0.8 grams for cinemic acid, and then different yield for the derivatives.
Okay, did you try increasing the batch size or so? Like, what is the possible challenges you will face when you're scaling up this particular batch?
Yes, ma'am. Actually, this reaction can be further happen using different halogens. So we actually optimized the whole process. So we couldn't able to like scale up studies was not done in our project. But during optimization, there were many trial and error methods using different solvents, such as a different solvent using acetonitrile and various solvents with different mole quantities and the different molecular weights. So after that, we came to the conclusion of this main reaction where we are using potassium bromide and calcium nitrate and a greener solvent, that is acetic acid. So we took a TLC at different time points like 20 seconds, 1 minute, 2 minutes, 5 minutes, and 10 minutes, and also 15 minutes. But we observed that the product was degraded in 10 minutes and 15 minutes.
Oh, yeah. Thank you.
Thank you. I wanted to know what is your greener solvent, or what was the aspect that you classify this as green chemistry, and you put it forward.
Yes, ma'am. Ma'am, and also in conventional method, many of the literature papers, they mentioned benzene, NBS, that is N-bromo, succinamide. So all these are toxic solvents which we used in conventional method using Hunsaker reaction. So this was a novel approach using all these reagents.
Okay. Thank you.
Okay, moving forward. PPG 13, are you there?
Yes. Am I audible?
Yes, you're. Yes, you're audible. Let's start PPG 13. You can start.
Okay. So, good afternoon, everyone. The topic of my poster is formulation of sublingual tablet using natural super disintegrants. So we formulated a sublingual tablet of unacetone hydrochloride using natural super disintegrants such as dehydrated banana powder and isabgol husk powder. And we compared the efficiency of this natural super disintegrant with synthetic super disintegrant, that is sodium starch glycolate. So the advantage of using a natural super disintegrant was it was biocompatible, non-toxic, and cost-effective. We did this formulation using 3x2 for full factorial design and optimized the formulation. And we found that the optimized formulation F3 containing isabgol husk powder 8 mg was the most best formulation out of the 9 formulation that we formulated, and the disintegration time was 7 seconds, and the maximum drug release was 106.3% within 30 minutes. So we also found the other evaluation parameters were within the pharmacopoeial limits of IP, and we also
We can't hear you.
Hello.
Yes. Are you done, Arya?
No, 1 second. So we formulated this 9 formulations, and isabgol husk powder was one of the best best formulation we found, and we understood that it was very effective to use natural super disintegrants rather than synthetic super disintegrants because it gave a faster disintegration time, and also it was comparatively low toxic. So it was a better formulation than the conventional formulations which were available in the market. Thank you.
Your time is up.
Okay, Arya, I have one question. Since you are mentioning that isabgol is comparatively a good one as a super disintegrant, but isabgol itself is having a biological effect. Okay, so what is the amount you have included as a disintegrant, which is, and it is having any pharmacological effect or not? Have you checked that?
Ma'am, pharmacological effect was not checked because we were only focusing on the formulation aspect and the disintegration and dissolution aspect.
Because whenever we are selecting the excipient, it should not be having any pharmacological effect, right?
Yes,
that
yes.
is the criteria. And you can see there are many studies which is indicating use of isabgol. So have you used a husk or entire isabgol?
Only the husk powder, ma'am.
The husk itself is having that mucilage in it.
Yes, ma'am. That mucilage was giving the super disintegrant effect.
Okay. Any more questions, judges?
Yes. Can you tell me what do you understand by super disintegrants? Hello.
Yeah.
Hello.
Yeah.
Hello, ma'am. Am I audible?
Yes.
So, ma'am, super disintegrant effect.
We can't hear you, PPG 13.
Hello.
Yeah, you're audible now.
I'm sorry, I'm facing some network issue.
You can answer the question now. You're not audible. PPG 13. Judges, she's not audible, so we'll move ahead.
Okay.
PPG 14, are you there? PPG 14? Yes. Yeah. Yeah.
So can I start?
Yes, you can start.
Hello everyone, good afternoon one and all. So the topic for my today's poster presentation is phytochemical investigation of aerial parts of Staples asper. I'm sorry. Aim for the project is to conduct phytochemical investigation of bioactive extracts. Objective was to conduct bioactive guided fractionation of the potent extract and identification of phytoconstituents, which is responsible for cytotoxic activity. Following is the methodology used. Coming towards result and discussion, the following table represents percent extractive value of successive extracts and following representative solvents. Majorly, steroidal glycosides were present in the DCM extract, which was confirmed using phytochemical tests. All extracts were screened for cytotoxic activity by SRB assay on MCF7 cell lines, which showed that DCM extract had better activity. The confocal images below shows that DCM killed major cells rather than any other cells. With respect to the above results, I proceeded with DCM extract for column chromatography using toluene as solvent to separate major constituents present. Obtained fractions were sent for analysis, which includes HPTLC, HPLC, LCMS. On derivatization of HPTLC plate, steroidal glycosides were confirmed on the major basis. On basis of LCMS data, following are the probable constituents which were identified at mass spectroscopy. So in conclusion, I could say that aerial parts of the plant, Staples asper, were found to be effective parts of this plant, and the toluene soluble parts of DCM extract could show significant anti-cancer activity. From aerial, the probable structures identified by the mass spectroscopy, which were isolated by column chromatography from aerial parts of Staples asper, could be effective in treatment of breast cancer. Thank you.
Yeah, over to you, judges.
What have you concluded from these confocal microscopy studies? I couldn't get you.
Ma'am, the DCM extract was found to be effective rather than petroleum ether, methanol, and water on MCF7 cell lines.
Okay, so you've tried to observe the MCF7 cell lines by confocal microscopy.
Yes, ma'am.
And why do you think the DCM extract was better than petroleum ether extract?
Ma'am, the steroidal glycosides from the literature survey, I could identify that steroidal glycosides were present in major amount. So, and the DCM steroidal glycosides are responsible for anti-cancer activity. So that's why I was keen focused on DCM extract.
And this results in discussion, what does this table in the first column, the last table indicate?
Ma'am, that are the percent extractive values, that is yield which I got from the extraction.
Okay, all right.
Okay, can I ask? Yeah, so with regard to solvent choice for the extraction, what you have picked up,
Yeah.
which category does it belong to? Like you have different classes, the FDA classes.
Ma'am, actually, I started from increasing polarity to the non-polar solvents.
Okay, but did you try to understand the residual solvent which is left behind your extract? Because your DCM, how safe it is?
Yes, ma'am, DCM is not safe for like inhalation. It is carcinogenic. But I can use N-butanol, but did not use N-butanol because it was not available. So I had to go with DCM extract only.
Just a suggestion, maybe once the extracts, you can just check in the residual solvent. Okay, whether it passes your limits or not. Okay?
Okay, ma'am. Okay.
So the solvents themselves may be cytotoxic, isn't it?
Exactly.
Ma'am, but after evaporation, the DCM like amount is very less for the toxicity, I guess.
I guess.
But but this is a cell line study, so whatever the concentration is there, that is enough to kill the cell, no?
Shall we move ahead?
Yes, ma'am. We'll go ahead. PPG 15. PPG 15, are you there? PPG 16. PPG 16, are you there? PPG 16.
Hello. Am I audible?
Yeah, you're audible. Yes, you can start. PPG 16, you can start.
Good afternoon, one and all. My code is PPG 16. The topic is dissolvable hydrogel microneedles loaded with baclofen for local relaxation of muscle stiffness, formulation optimization, and characterization. So basically, it's a proposal for the treatment of muscle stiffness, which is an acute disorder that is characterized by pain and muscle rigidity, which can be disabling for the patient. And baclofen is a drug which is a GABA-B receptor agonist, an efficient drug for treatment of muscle stiffness, majorly administered orally, but it has side effects like dizziness, seizures, insomnia, constipation, etc. So for that, microneedles can be efficient for the topical delivery of baclofen, and microneedles have superior stability when compared to other topical formulations like creams, gels, etc. Thus, the use of baclofen-loaded microneedles can prove to be an efficient drug delivery system in the treatment of muscle stiffness. So the methodology follows microneedle mold is fabricated using polydimethyl siloxane, and PLGA is the matrix forming for the microneedle, and the baclofen and other ingredients are disposed in the DMF and THF mixture and poured into the mold. The microneedle matrix is dried and allowed to solidify before removal from the mold. Characterization, we do visual inspection, fracture test, insertion studies, skin irritation, skin permeation test, in vitro drug release. So the expected outcome will be a novel system that combines a drug with proven clinical benefit in a new delivery system, which is biodegradable. The system will be easy to administer and will be painless. The presence of microneedle will cause excellent permeation with a rapid onset of action with sustained release profile. Thank you.
Yeah, over to you, judges.
Yeah, this is a review, right?
So, ma'am, it's a proposal, research proposal.
It's a proposal. Which grade of PLGA you will be using?
Ma'am, 75 or 25.
Why? What is 75 and what is 25?
Means the PLGA is poly-D-L lactide co-glycolide copolymer. So the lactide is to glycolide ratio is 75 to 25.
Okay. Why can't I go with 25 and 75?
Ma'am, because I have seen articles where their microneedle prepared using these polymers. Means the ratio of the polymer is this, that's why. And also means the lactide is a water, means it is a hydrophobic, and glycolide is hydrophilic. So the ratio of 75 and 25 has proven a good result. Means it has given good results for microneedles. That's why.
Okay, fine. Thank you.
Any more questions, judges? Okay, so we'll, ma'am, yeah, you wanna ask?
No, it's okay. Let it. You can proceed.
Okay, thank you.
Okay, so PPG 15.
Yes, am I audible?
Yes, you're audible.
Okay.
You can start now.
Okay. The topic of my presentation is the phytosomal curcuma modernistic approach for the therapeutics. Introduction will be turmeric. You all know it is a yellow colored spice which is obtained from plant Curcuma longa. Family belongs to Gingiberraceae. Chemical constituents will be curcumin, protein, carbohydrate, mineral, philandrin, cineol, borneol, sabine, gingiberine, sesquiterpene, etc. It is used for various types of cancer. Here we are developing the phytosome preparation of the curcuma longa. It is helps to convert the insoluble lipid insoluble drug into a
lipid water soluble drug into lipid soluble drug so that it can be easily passed through the biological membrane and reach the blood. Thus it will improve the pharmacokinetic pharmacodynamic profiles, and therefore the bioavailability increase and the dosing frequency and dose will be reduced, and therefore there is a patient compliance. Goal here to increase the bioavailability and therapeutic efficacy and evaluation of the final phytosomal formulation. Experimental details, here we have done UV spectrometric analysis. There is a calibration curve, and then there is a preparation of the curcuma longa phytosomes. It is done by solvent injection method. This bow diagram indicates that one is of the phytosomal complex, and another diagram shows this that the phytosomal complex now can easily enter the biological membrane. Here characterization is done by zeta potential and entrapment efficiency. Cytotoxicity studies now carried on human breast adenocarcinoma cell, that is MCF7 cell line, by MPT assays, and IC50 value was recorded. UV value we get as 424 nanometer. Zeta potential value is minus 4.08 millivolt. Entrapment efficiency, we have uses different batches with different concentration, and the batch number 2, that is 20 mg of extract, 20 mg of lipid, and 2 mg of cholesterol gives maximum entrapment efficiency. The cytotoxicity evaluation, here as a drug concentration.
Your time is up. Your time is up.
Okay.
Over to you, judges.
Okay, so why you have selected this MCF7 cell line only?
Because ma'am, we want to check the efficacy of the drug on the breast cancer cell. That is why we have selected MCF7 cell line.
But why you don't want to check in other cell lines? What is the rationale behind selection of this only? Is there any literature?
No, ma'am, it is not like in literature survey. It is shown that it gives a good efficacy with this cell line. That is why I tried this cell line.
It is already reported, right?
Yes.
So what is the novelty in your work?
Ma'am, it is reported, but not with the same formulation. It is for the drug that it is a good in MCF7 cell line.
Which is what even I would like to know, because curcuma is already reported to be cytotoxic. There is so much literature available regarding its use as an anti-cancer agent.
Yes, ma'am.
So
The
what is the
novelty is
novelty?
that here we have prepared the phytosomal formulation of curcuma longa. So this is not reported in any of literatures.
What do you mean by a phytosomal formulation?
Phytosomal formulation is that as a curcumin itself is not easily absorbed by the blood, so we have converted it into a phytosomal formulation. Here it is here it is mixed with the lipid and cholesterol, and it is converted into phytosome. So it forms a film around it, which is lipid soluble, and now it can be easily passed through the biological membrane and thereby in the blood.
So have you chosen the pure curcumin or that curcumin, that fruit drug?
No, ma'am, curcumin.
Curcumin. I doubt, because this is a very common drug. Even in the market, nutraceuticals are available for the cancer therapies as a dietary supplements. So, and the phytosome is a very common formulation. So I doubt that this type of formulation is not available in the market.
Okay,
Very
ma'am.
true.
Yeah, can we go ahead, ma'am? Yeah, so PPG 17, are you there?
Yes.
Yeah, okay.
Can I start?
Yes.
Okay. So good afternoon, everyone. So my topic today for the presentation is novel oral treatment strategies for the management of epilepsy. So epilepsy is a disorder in which a nerve cell activity of the brain is disturbed and it causes seizures. So there are many conventional therapies available, but they have several disadvantages like withdrawal symptoms and some deleterious interaction. So to overcome this,
we formulated an oral disintegrating extended release tablet by using topiramate microspheres. So topiramate is standardized by using HPLC method, and for extended release we use microspheres. So microspheres were prepared by using ethyl cellulose and HPMC by emulsion solvent diffusion technique. So we prepared the 9 trial batches and they were evaluated for the yield, particle size and entrapment efficiency. And the one which have highest entrapment efficiency was selected. Then we done the scanning electron microscopy of the same. Then the further developed microsphere were further compressed by using mannitol as a diluent by direct compression method. And it was further evaluated for its appearance, dimensions, uniformity of the content, disintegration, in vitro dissolution studies. Also in vivo dissolution studies also done by using a rat behavioral test avoidance model. So to conclude, we like to say that this formulation can be used, can help to treat the epilepsy by masking its test and giving a sustained release profile. Thank you.
Over to you, judges. Over to you, judges.
With regard to HPLC chromatogram, okay, what is the unit on y-axis and what was your concentration you have spiked?
PPG 17, please unmute yourself.
Yes. Can you repeat again?
Yeah, in your HPLC chromatogram, okay, what is the unit on y-axis and what was the concentration of the drug you have injected? And you have prepared an ODT.
Yes.
Right?
Yes,
and
ma'am.
why it has been, you have done your HPLC analysis in acid media.
Yes, ma'am. So the reason for using acid is when it disintegrates, mouth has acidic taste.
Your voice is not audible.
Hello, am I audible?
Yes.
The oral disintegrate into the mouth itself. So mouth has pH, acidic pH. So we use the HCl.
Okay, mouth has got acidic pH. Okay, thank you.
Thank you.
You prepared microspheres and then you have compressed them into a tablet. Have you checked for the morphology of your microspheres after being compressed into tablets?
Yes, ma'am.
Yes. Okay. And have you added something in your formulation as something to, as a, what do you say?
Mannitol as a diluent, so it is used as also diluent, and also it has sweet test, so it will mask the test of topiramate, which is slightly bitter, so it will be more compliance with the like patient compliance.
All right.
Okay, we'll go ahead. PPG 18, are you there?
Hello.
Yeah, you're audible.
Yes.
Yeah, you can start now.
Ma'am, so our topic, novel topic topical emulgel as non-comedogenic sunscreen formulation. Acne can lead to anxiety and depression. UVB rays from sunlight can damage DNA and lead to malignant transformation. The conventional products for acne can cause itching, dryness, and make our skin more prone to sunlight. The emulgel preparation is done by mixing emulgel, ultra gel, and oil in water immersion. Table number 1 is a prototype formulation, and it was found that the prototype formulation 4, which does not contain zinc oxide and capric triglyceride, was found to be more stable. So these were it was found that stearic acid and montano 82, which is surfactant, affects the formulation. So these were optimized by using 3 raised to 2 factorial design. Table number 2 is a trial batch for screening the 3D surface response. As the concentration of stearic acid montano 82 increases, it was found that the viscosity of the formulation also increases. And as the concentration of stearic acid increases, the spreadability of the formulation goes to decreasing. The evaluation of trial batches were done for viscosity, spreadability, pH, centrifugation stability, and freeze law. And it was found that the formulation third was shows the optimal parameters. Determination of SVOs were done by using a UV spectrometer, and it was found that the formulation shows the highest SVO. Results and design. In vitro drug diffusion, XVO drug concentration was found to be less,
Your time is
and
up.
percent skin retention
PPG
was to
18,
be
your time is
good
up.
as okay.
Over to you, judges.
Yeah, nice work. How do you perform the experiment study?
Yes, ma'am.
How did you perform the ex vivo study?
By using, ma'am, PIGS EA.
Okay, what were the challenges you faced there?
So, ma'am, it was found that the zinc oxide does not produce the desired effect on sebaceous glands.
Pardon?
It was found that the zinc oxide, which we evaluated, does not lower the sebum production. Did not.
Okay,
No,
and
that is a
why?
known fact.
Sorry.
Yeah, that is a known fact, no? Zinc oxide is just an astringent and doesn't reduce your sebum production. And you seem to have
Yes,
performed some clinical
ma'am.
trials as well. You have, first thing, you have put up a lot of matter on the slide. Nothing is readable again. And you seem to have performed some clinical trials as well. So, what
Yes,
have
ma'am.
you compared with, or I mean, was there any comparison that you have done? Or, I mean, when you did,
In
tested your formulation, you
in
would
a
have
trial.
used some control groups, right?
Yes, ma'am. Yes, ma'am. Ma'am, it was found that the no more acne was formed on the uh, acne-prone skin after the usage of up to 1 month.
Okay.
Can we go ahead, ma'am?
Yes, please.
Yeah. PPG 19, are you there?
Hello.
Yes, you're audible. PPG 19, you can start.
Yeah. So my topic is lopinavir loaded SLN for HPV-induced cervical cancer. Introduction: the cervical cancer caused by human papilloma virus is the second most cancer caused in the woman around worldwide. So for that, treatment needs to be developed, and co-delivery of the agents by using nanocarriers was developed. The aim of my study was to develop and evaluate lopinavir loaded SLN cream for cervical cancer to be applied topically locally. Rational, the lopinavir oral formulation showed less solubility, extensive pre-systemic metabolism. Hence, localized action was preferred. Local administration will provide localized action, thereby decreasing the dose. SLN were selected because it increases the drug permeability, absorption, concentration at the target site. SLN were prepared by using solid lipid and lopinavir were melt together, and then in another beaker, surfactants and water was taken, and they were pre-emulsified and then cooled at room temperature, and then they were further subjected to high pressure homogenization for size reduction. The optimized formulation composition was lopinavir, compritol, 888 ATO, poloxamir, 2 and the homogenization parameters were pressure was 10,000 psi, time was 15 minutes. Evaluation done for SLN were particle size, PDI, zeta potential, and entrapment efficiency, which showed 69.85%. The cream and then further cream was prepared using emulsifying wax, glycerin, IPM, HPMC K15, methyl propyl parabene, lactic acid, and the evaluation for the cream was pH, viscosity, spreadability, drug content, and bioadhesive strength. Bioadhesive strength was done using goat vaginal mucosa, and the drug content showed was 98.78%. Then in vitro drug release was performed using nylon membrane, and it showed for plain cream it showed 65.39%, and for SLN beige cream 89.63%. And ex vivo studies was performed using goat vaginal mucosa. Plain cream showed 42.56%, and SLN loaded cream showed 82.25%. The conclusion for this project was lopinavir SLN vaginal cream was found to be controlled release as compared to plain, and it showed good results. Vaginal cream showed good potential to target HPV virus induced cervical cancer as compared to oral route.
Your time is up.
Thank you.
Yeah. Over to you, judges.
Did you apply any mathematical modeling because you have concluded saying that it followed the zero order and the Higuchi model? Did you apply any mathematical modeling for your studies?
No, ma'am. This was used. Factorial design was used. Hello.
Hello.
Yes.
Factorial design was used for what? For understanding your release profile?
No, for formulation.
Correct. So in your in vitro and ex vivo bottom,
Okay.
you have concluded saying that it followed zero order release and followed Higuchi model.
Yes.
So did you apply any mathematical equation or kinetic models here?
Release kinetics.
You have applied?
Release kinetic.
You have applied the release kinetic models?
Yes. Graph was applied.
Okay.
Yeah, ma'am.
So for Higuchi, what is on x-axis and y-axis?
Log percent C cumulative release and log release.
Okay. Thank you.
We can go ahead.
Yeah, ma'am. PPG 22, are you there? PPG 20 and 21 are having some issues, so we'll go ahead with PPG 22. PPG 22, are you there? PPG 22? PPG 23, are you there? PPG 22, are you there?
Yes, ma'am.
Yeah. Yes, you can start.
Yes, ma'am. Just give me a second. Hello.
Yes, you're audible.
Hello. Am I audible?
Yes, you are.
Good afternoon, everyone. This is Nabila Begum from St. Paul's College of Pharmacy. Here, my research topic is design and development of value-added nutraceutical functional food for the benefit of diabetic population. Here, the word nutraceutical is coined by D. Fleece in 1989. And our main aim is to formulate the anti-diabetic cookie for the diabetic patients, which is of natural origin and have no side effects when compared to the conventional synthetic drugs. Here we have formulated the cookie with the use of herbal ingredients and the insulin plant extract. So for the first step in the formulation of this cookie is the extraction of the leaf extract, that is the insulin plant, that is Costus ignis. So we have collected the insulin plant leaves and we have shed dried it, and we have go to the oxalate extraction, and after getting the final extract, we have formulated the cookie by the use of various herbal ingredients like pumpkin seeds, flax seeds, watermelon seeds, sunflower seeds, coconut powder, and sucrose as artificial sugar. Here, we have not used the wheat flour and sugar, which we have replaced it with the artificial sugar, that is sucrose, and all the functionalized ingredients. So the wheat flour is replaced with all the functionalized ingredients such as pumpkin seeds, flax seeds flour, sunflower seed flour, and after collecting all the ingredients, we have formulated into it cookies. After selecting the various composition, the best composition is selected based on palatability and sensory evaluation. And after selection of final
Your time is up. Over to you, judges.
Okay, so I have a question. When you have prepared cookies, what was the temperature condition in the oven? And is there any effect of that temperature condition in these ingredients?
No, madam. The temperature is minimum temperature, that is 100 degrees centigrade, 100 degree Fahrenheit in the oven. I have baked it for only just 5 minutes, and the result is that the cookies was prepared so nicely. There is no effect of temperature on all the ingredients. All the ingredients are stable.
Have you got any reference which indicates that 100 degrees Celsius is okay with the antioxidants? There is no damage with the antioxidant?
No, madam. I have searched a lot, but they have prepared the cookie, but no one have mentioned the temperature. Actually, this nutraceutical is not properly regulated. This is all upcoming source, and it is not regulated properly. So,
okay. One more question. Is there any reference which indicates that sucrose is not having any side effects with the diabetes, or it is not responsible for increasing the glucose and all that, because it is also come under the category of sugars only, no?
Yes, madam. Ma'am, my mistaken, it is not the sucrose, it is actually sucralose.
And why you are saying this is a insulin plant?
Because insulin plant having anti-diabetic activity. From the recent traditionally it is used as anti-diabetic for the diabetic patients. So it is having all the anti-diabetic effect.
There are multiple plants which is having the anti-diabetic effect. Why you are mentioning that this is an insulin plant? Why you are saying that it, as a insulin plant? Again and again, I have heard in your presentation. Is there any traditional reference which is mentioning as a common name as an insulin plant for this?
Scientifically, it is named as Costus igneus, madam. Based on the clinical trial, it is proved that it is having higher antioxidant activities and all the phytochemicals which is exhibiting the anti-diabetic properties which can control the blood sugar level in the diabetic rat models.
Okay.
Okay, we'll move forward.
Okay, ma'am.
23, are you there? PPG 23, are you there? PPG 24. PPG 24. Okay. PPG 25.
Hello, ma'am.
Yes, you're audible.
Yeah. Can I share my screen, ma'am?
No, we'll share.
Okay, ma'am.
Yes, you can start.
So, good afternoon, everyone. So, my presentation topic is generation of 3D printed multifunctional customized drug delivery system and its into and in vivo evolution. So, coming to the introduction part. So, hot melt extrusion and 3D printing technology is a cutting-edge technology in the field of pharmaceutical sector for the preparation of personalized drug delivery system. So, here, coming to the materials part, here I use norfloxacin as a model drug and polydone as a polymer matrix for hot melt extrusion, and PEGDA, that is polyethylene glycol diacrylate, as a photo curable resin for the stereolithography 3D printing. So coming to my method part, so the picture depicted the entire formulation design and experimental design of my work. So here, before going to hot melt extrusion, we use some mathematical modeling for the optimization of hot melt extrusion parameter. After that, speronization, and by using that speronization, we prepared some micro pellets of size 1.5 mm, and which confirmed by using master sizer as well as same analysis. After that, by using stereolithography 3D printing technology, we prepared capsule size of 2.8 mm, diameter and length of 9 mm. So and the entire formulation was evaluated by using DSC, XRD, ATR, and in vitro and in vivo analysis. So coming to the result and discussion part. So first, to optimize the hot melt extrusion parameter, we use mathematical modeling, that is Fleury-Against theory and Hansen solubility parameter approach. So according to the Fleury-Against theory and Hansen solubility parameter, we confirm that we were going to know the solubility and miscibility of polydone SR and norfloxacin drug. So we found a good solubility of norfloxacin in polydone SR. After that, the hot melt extrusion was used to prepare norfloxacin roded filament, and the filament was characterized by using DSC, XRD, and ATR analysis. So the DSC and XRD result shows
PPG 25, your time is up.
Okay.
Over to you, judges.
What is your final formulation? Can you tell me?
So, ma'am, my final formulation is a microparticles or micropellet loaded capsule, ma'am.
Okay. And you have prepared it only using Colidone SR?
Yes, ma'am, only Colidone SR, ma'am. Actually, Colidone SR is which gives a sustained release behavior, property. So I use, ma'am, Colidone SR because norfloxacin has a very short half-life, as well as the marketed formulation norfloxacin, which contain 200 mg of tablet, which we need to take thrice a day, ma'am.
So this picture that you have depicted is of your formulation?
Ma'am, that cap means the picture has two parts, ma'am. First cap and means only capsule, which is empty capsule, and second is a microparticles or micropellet loaded capsule, ma'am.
Okay.
What is the size of capsule?
Hello, ma'am.
What is the size of this capsule?
Hello.
Hello.
Ma'am, your voice is breaking, ma'am.
What is the size of this capsule?
So, ma'am, the diameter of that capsule is 2.8 mm, and the length of that capsule is, ma'am, 9 mm.
Okay.
Okay, I have few questions.
Yes, ma'am.
First of all, congratulations, you have tried exploring new technology for your project. So
Thank you,
just
ma'am.
out of curiosity, just to understand, when you were using this 3D printing technology, especially
Yes.
stereolithography,
Yes, ma'am.
how did you screen this particular polymer? Like, like what I understand, it should have that ability to be used in this technology. So what property of
Yes, ma'am. Actually,
yeah.
Yeah, ma'am. No, ma'am. Actually, Polydone SR we use for the preparation of hot melt extruded filament, ma'am. But for the 3D printing technology, we use PEGDA, that is polyethylene glycol diacrylate, as a photopolymer, ma'am. Actually, stereolithography 3D printing works on photopolymer.
Your voice is breaking. Yeah.
Hello, ma'am.
Yes, yes.
Yeah, my voice is clear, ma'am.
Yes, yes. Please.
So, ma'am, actually, Polydone SR here we use for the preparation of hot melt extruded filament, ma'am. But for the preparation of 3D printed capsule, we use PEGDA as a photopolymer, ma'am. That is polyethylene glycol diacrylate. But polyethylene glycol diacrylate is not a biodegradable polymer, ma'am.
Right.
So, prepared customized resin in our lab, that is incorporating some hydrophilic group like in PEGDA. So here we use PEGDA, polyethylene glycol, and NaCl, that is salt here. The salt acts as a corrosion, ma'am. When we put that capsule in buffer, ma'am, at that time the salt dissolved in that buffer immediately, and it will create some pores, so that our drug release from that capsule, and due to osmotic pressure there is a bursting of that capsule body and shell, ma'am.
Okay, okay. That's the reason, because in the diagram you have not mentioned anything. So I was more curious to understand
Yes,
how
ma'am.
did it fit in that particular this. And
Yeah.
in terms of HME, did it
Yes, ma'am.
extrude out without any plasticizer? I mean, just
Yes.
drug and polymer, did it extrude smoothly?
Yes, ma'am. Sometimes, ma'am, drug itself act as a plasticizer, ma'am.
Okay, okay. What is a TG? What is your TG of your polymer?
So, ma'am, actually the TG of my polymer is 40 to 45 degrees Celsius, ma'am.
Okay.
And the TG of our entire mixture, that is drug plus polymer, it's 40 degrees Celsius, ma'am.
Okay, okay.
Yeah.
Would love to know more about you, about your project. Maybe I'll get in touch with you maybe after the presentation.
Okay, ma'am. Sure, ma'am.
Yeah.
Ma'am, what is your name, ma'am?
Sarita Shetty.
Okay, ma'am. Thank you, ma'am.
Thanks.
Yeah. Thank you.
Okay, moving ahead. PPG 26, are you there?
Yes, ma'am.
Yeah, please note, before going ahead, please note that you are not allowed to say your name, or not allowed to reveal anything related to your college, your affiliation. If you reveal anything, then you can be disqualified.
Okay, ma'am.
You can only say the poster number.
Yeah, ma'am.
Yeah, you can start.
Okay, ma'am. Good afternoon, everyone present here. The topic of my presentation is plant phytosome, a monogenetic approach for better therapeutics. Herbal medicine now being considered as a potential source for anti-cancer agent and are widely used due to availability of the material, affordability, and their therapeutic activity. The phytochemicals obtained from medicinal plants play a crucial role in the treatment of cancer. Mostly plant secondary metabolites and their derivatives have been applied to combat cancer. The aim of this study was to formulate phytosomes containing licorice and gabul extract with good physiochemical properties so it can improve the stability and bioavailability. In experimental, firstly, phytochemical screening of both the extract and then characterization of both the hydroalkalic extract done by TLC and UV. The formulation of the phytosomes were made by solvent induction methods, and then characterization of phytosomes were done by particle size, zeta potential, entrapment efficiency, in vitro dissolution studies in order to ensure the physical stability of the phytosomes. Then qualitative analysis of the phytosomes were done by HPLC, and in vitro evaluation of these phytosomes were done by anti-cancer activity of phytosomes on MCF7 cell line. So this test compound like licorice and guggul, both extract will exhibit significant cytotoxic potential properties with IC50 concentration 22.53 μgml and 73.4 g μgml for guggul. So at the end, it was concluded that the licorice have more cytotoxic as compared to guggul. So it can be used as a anti-cancer agent in further study will be carried out. Thank you.
Yeah, thank you. Over to you, judges.
Yes, why was selected licorice and Google for this phytosome formulation?
Ma'am, actually in marketed, there was one tablet which contained 8 herbal ingredients which were used as anti-cancer agents, which contain other CC ingredients other than licorice and guggul. So I have chosen this licorice and guggul and I work on these two extract. I have made that phytosome. After that, making phytosome and checking this anti-cancer activity, now I will make the tablet of these individual extract of this phytosome, like licorice tablet individual and then guggul tablet. Then I will compare these two individual tablet ka result with their marketed formulation. So I will compare that individual will give more result as compared to this one marketed one.
Okay.
Can we go ahead, ma'am?
Yes, please.
Thank you, ma'am.
PPG 27.
Yes, am I audible?
Yes, you're audible.
Okay. So, good afternoon, everyone. In this poster, we have discussed the interactions of LMTM, that is glucomethylthionine bishydromethane sulfonate, with an acetylcholine esterase inhibitor in a tau transgenic mouse model using L1 female mice. LMTM is intended to reduce the formation of tau protein tangles in the brain. In a study conducted by Riddle et al., they have examined the effect of either LMTM alone or chronic rivastigmine prior to the treatment, expressing short tau fragments that constitute the tangle filaments of Alzheimer's disease. LMTM given alone increased the acetylcholine levels in hippocampus, glutamate release from the synaptosomal preparations, synaptophysin levels in multiple brain regions, and the mitochondrial complex. It has also showed a reduction in tau pathology, partially restored the choline acetyl transferase immunoreactivity, and reversed deficits in the spatial learning. Chronic pre-treatment with rivastigmine was found to reduce or eliminate almost all the effects of LMTM. The long-term pre-treatment of LMTM with a symptomatic drug causes a wide range of alterations in the brain. These alterations can be attributed to various neurotransmitter systems and cellular compartments at multiple levels of brain function. Hence, there is no single locus for this negative interaction. In phase 3 clinical trial, participants receiving a dose of 100 mg twice a day as their only Alzheimer's therapy scored significantly better on their cognitive and functional tests rather than a group taking lower dose or in combination with other symptomatic drugs. Participants receiving a monotherapy of LMTM had also better cognitive scores. I would like to conclude by saying that the driving force behind the study is that an unproven assumption that symptomatic treatments do not interfere with the treatments that target the underlying pathology because the modes of action are different. And in this present study, they have proved that this assumption is incorrect. Thank you.
Over to you, judges.
This is a review article.
No, ma'am, it is a research article that they have shown how does LMTM shows a negative interaction when it is given with a established symptomatic drug. For example, rivastigmine that is already used in the treatment of Alzheimer's. So if this new LMTM drug, if it is given in combination, it shows a negative interaction. And hence, if we are using LMTM for the treatment, it is preferred in monotherapy.
You have done this work, or is it a research paper which you have referred?
No, ma'am, I have referred to this research paper.
And it is called a review paper, no? The review poster.
Okay, okay. Sorry, ma'am. Like, oh, that is my bad.
Just one suggestion, the content which you have included, no, it is not readable at all. So in future, please try to incorporate more number of figures and reduce the text and increase the text size also, because it is very difficult to read and understand.
Okay, ma'am.
Shall we move ahead?
Yes, ma'am. PPG 28. 28, are you there?
Yes, I am.
Yeah.
Yeah, the poster. Yes. Hello. Okay. Thank you. Good afternoon to everyone present here today. We are Group PPG 28, and our topic for presentation is artificial intelligence in drug design and development, where we would like to talk about some applications of AI in designing drugs. So, what is AI? Artificial intelligence or technology, where it can mimic the human ability to think and make decisions. It can collect information from a surrounding environment and continuously improve itself to make better informed decisions. Some points I would like to elaborate from this poster are, now in QACR, which is one of the tools used in molecular modeling, this process can be even furthermore semi-automated by inputting desired requirements and allowing the AI feature to find the lead molecules instead of the scientist needing to spend his valuable time in front of the screen. Moving towards clinical trials, where there's vast unstructured data collected, which the clinicians have to go through. In such cases, AI can filter out relevant information and present it in a more organized manner. Similarly, in drug repurposing, it can identify new biological targets for existing drugs and vice versa. The International Uppsala Monitoring Center for ADR reporting, all the information can be updated in real time and integrated so the database is always up to date, all with the help of AI. At the same time, machine learning, which is interchangeably used with AI, can identify repeating patterns and algorithms and make future predictions of such adverse drug reporting. Now, all companies we know and organizations have different modes of working in the healthcare industry. Hence, artificial intelligence can be either outsourced to a third party or the platform can be customized to and personalized to better suit the organizational needs. To conclude, artificial intelligence can prove to be a helpful tool in the field of medicine by avoiding human errors, assessing large databases efficiently, and speeding up the process of drug discovery. Thank you.
Yeah, over to you, judges. Over to you, judges.
No question from my side.
Even I don't have any questions.
Okay, moving forward. PPG 28, are you there?
I was PPG 28. Hello?
Yes, PPG.
Yeah. 28 is done.
PPG 29. PPG 29, are you there? PPG 30.
Yes, I'm there.
Okay. Please note this is PPG 30.
Okay.
Yes, you can start.
Okay, our topic is tears as a biomarker for the early diagnosis of Alzheimer's disease. So, as we know, Alzheimer's disease is a neurodegenerative disorder which starts slowly and progressively worsens. The major pathological hallmarks of AD include the hyperphosphorylated tau proteins and deposits of amyloid beta. So, the current diagnostic techniques which are used for AD is the blood test, cerebrospinal fluid test, and various neuroimaging techniques such as PET, MRI, and CT scans. So it has been observed that neurodegenerative changes due to AD are also associated with morphological and structural changes in the eye, or rather the entire visual system. So it has been hypothesized that these changes could also result in alterations in the tear composition or the tear profile in AD patients. So a few of the recent studies they demonstrated, which were conducted on AD patients as well as subjects which have AD Alzheimer's familiarity, it was observed that retinal plaques were observed, as well as amyloid beta 42 residues were observed in the tear samples in subjects with Alzheimer's familiarity. And in another study, it was observed that the tears of AD patients they showed altered protein concentration compared to that of control group. So to conclude, the current diagnostic techniques are used. They are invasive, costly, and with seldom possibility of early diagnosis. Also, they mostly try to rule out other causes such as hemorrhage, which could also show symptoms similar to that of Alzheimer's. Hence, tear analysis could have a predictive role in early diagnosis of AD. Thank you.
Over to you, judges.
So, have you found enough literature to support this hypothesis that tears can be, or is it only one article that you have?
Ma'am, there were many review articles, but research articles I found latest. It was published in 2021. So research articles I've not found many research articles, but there have been many review articles on this topic.
Okay. So, which protein is altered? At least have they mentioned that? What proteins?
Yes, ma'am. There were 4 major proteins. It was lysozyme C, lacretin, dermcidin, and ma'am, another one protein. There were 4 proteins.
Okay. So, are these 4 proteins normally present in tears, or it is only in Alzheimer's that they are present, or their concentration is
Ma'am,
altered?
they are present in tears, but their concentration was altered in AD patients and compared to the control groups.
Okay.
There was significant difference in the concentration.
All right.
We'll go ahead. PPG 29, are you there? PPG 29. You are not audible. PPG 29, you are not audible. PPG 31, are you there?
Yes, ma'am.
Yeah, okay. We'll go ahead with PPG 31. PPG 31, you can start.
Good afternoon, everyone. My topic for presentation is nutraceutical in pharmaceutical research. So nutraceuticals are basically a food or food supplements which provide medical and health benefit, including prevention and treatment of diseases. It is a bridging gap between the food and medicine, which is used to combat major diseases. So we are studying the role of nutraceutical in COVID-19, which shows antiviral by viral inactivation, cell response, immune system regulation, and cytokine storm modulation. Our study compound is quercetin, which inhibit the production of which shows action by inhibiting the production of NLRP3 inflazome, which is regulated by NRF2, where the cell response is induced by activation of NRF2 and following gene regulation downstream.
The activated NRF2 dissociate from its complex with KEP1 and migrate from cytoplasm to nucleus, within which it interact with the antioxidant response element, sequencing producing gene, regulation promoting antioxidant response, and reducing the inflammatory cascade. That shows indirect immune system modulation through enhancement of immune performance and concurrent decrease of prophylogistic cytokinase.
Okay, over to you, judges.
What is NLRP3?
Node-like receptor protein.
What is its role?
It inhibits the production of inflammation.
Okay, so the drug which you have mentioned, the quercetin, resveratrol, and GCG, it's EGCG, right? It's not GCG.
Yes, yes.
So they are acting via this NLRP3?
Yes, ma'am. Yes.
Okay.
We have chosen this quercetin because, according to the expression studies, the quercetin alters the expression of 98 genes out of the 33 genes, human genes that are responsible for encoding the protein that is target of SARS-CoV-2.
Okay.
We'll go ahead.
Thank you, ma'am.
PPG 32, are you there?
Yes, I'm present.
Yes, you can start.
Okay. First of all, good afternoon, everyone. This is PPG 32. So my idea for this poster presentation is how a gut malfunction could lead to CNS disorders and affecting the brain and its link. That is a link between a gut leak leading to a anxious mind. So background is we found it is being found that existence of anxiety in major in major GI disorders is very prominently seen, like in IBS disorder it was found to be as 44%, in IBD it was found to be as 20, then in diabetes it was found to be as 28. So the main link between the gut and the brain is the gut-brain axis, wherein the gut as well as the brain works in a synchronized manner for both its proper functioning. Any malfunction in this could lead to disorders both in gut as well as in brain. So the intestinal barrier is one of the factor which could act as a trigger for this malfunctioning, and the barrier mainly involves the IAP, mucus layer, epithelial layer, and the antimicrobial peptide. And the factors which could lead to the malfunctioning of the intestinal barrier included the LPS, that is lipopolysaccharide, a disturbed gut microbes, diet, drugs, the drugs like antibiotics, NSAIDs, environmental stress. So the possible mechanism for this intestinal barrier dysfunction induced anxiety was found to be like due to intestinal barrier dysfunction caused by the above factors, luminal contents are being entered into the systemic circulation, leading to the activated immune system. This further activates the vagal nerve, which activates the HPA axis in the CNS or the brain, and then due to the activated hypothalamus, which releases the ACTH from the kidney, there is an elevated level of cortisol in the blood, which leads to anxiety. So this is the pathway or the mechanism through which the intestinal barrier dysfunction could lead to anxiety induction. So the
PPG
drugs.
32, your time is up.
Okay.
Forward to you, judges.
So is it a review study or is it your work?
It is my hypothesis study, ma'am. This is my idea upon which I'll be working. So the main drug for this condition would be either monotherapy or combination therapy, wherein I could add upon the drugs which would act upon the intestinal barrier protectors, means the epithelial layer protectors, as well as some novel anxiolytics.
Okay, okay.
All right. And okay, you have just proposed this hypothesis on which you will work on. I just want to understand from you what would be the factors that you would be considering: pathogens, disturbed microbes, or only you will be including stress?
No, ma'am, my induction method would be a drug.
Okay.
Because pathogen and microbe, I don't think in this COVID condition I would be able to properly execute the thing. And first of all, there is no such protocol, first of all, like this for this intestinal barrier dysfunction induced anxiety. So first, as a first study, I'll go with the drug, and I'm trying for NSAIDs. Means through
Okay.
NSAIDs, I'll induce the gut barrier dysfunction.
All right. Okay. Thank you.
Thank you, everyone.
Moving ahead, PPG 33, are you there?
Hello. Yes, I'm there.
Yeah.
Okay, good afternoon, everyone. So, our topic for today's presentation is marine source against cancer, particularly cyanobacteria. According to global statistics, there is increase in mortality rate due to cancer, and available treatments have various limitations. There is a need of development of new anti-cancer agents from natural source. Even though various natural sources already have been explored for the anti-cancer therapy, resistance is the important and common problem in them, which makes marine sources novel and novel approach for cancer treatment. So, marine population represents reservoirs of novel bioactive metabolites with diverse group of chemical structure. Here we present cyanobacteria, also known as blue-green algae, which is source of more than 400 novel anti-cancer agents. We have listed few of the important derivatives derived from cyanobacteria. For example, calothrixins, boraphycin, cryptophycin, etc., which have been proved for the anti-tumor activity on various cancer cell lines. Important advantage would be that cyanobacteria, the on research on cyanobacteria would be high discovery rate, which is more than 95% for novel compounds as compared to other traditional marine sources available. The only limitation with marine sources would be the stability problem, which can be overcome by nanocarrier approach or nanoformulation approach. Hence, marine sources can be very effective alternative for existing chemotherapy agents for cancer. Thank you.
Yes, over to you, judges.
No questions from me.
Thank you, ma'am.
Okay, we'll go ahead. PPG 34, are you there? PPG 35, are you there? PPG 34. PPG 35, are you there? PPG 36, are you there?
Yes, present. PPG 36.
Okay, we'll go ahead with PPG 36.
Okay, can I start?
Yes, PPG, judges, please note this is PPG 36. Yeah, you can start.
Okay, good afternoon, everyone. Our topic for today's poster presentation is evaluating the content uniformity of immunomodulatory preparations with simple UV spectrophotometric method. As we all know, in COVID pandemic, along with the allopathic treatment, people have been using the herbal preparations possessing some immunomodulatory properties. So our goal is to evaluate the uniformity of eugenol, which is the main phytoconstituent present in tulsi, that is Ocimum sanctum. Moving towards the experimental details, first of all, we have prepared the 1000 ppm solution of standard eugenol. Then the dilutions were made by the methanol to obtain the standard solution in the range of 5 to 30 μg per ml. Then the absorbance of the resultant solution was determined at the lambda max of 280 nanometer. The absorbance, all the 5 preparation, that is from 5 to 30 ppm, was given in this table. Then we have for calibration curve, we plotted the concentration in ppm versus the absorbance, so that we got this calibration curve. From this calibration curve, we got the regression equation, that is y equals to 0.0235x plus 0.0065, and the correlation coefficient value, that is r square value, is 0.9961, which was highly significant. So the results, the Jolly Tulsi drops, that is our first preparation, contains 0.243% of eugenol. Then the Dabur Tulsi drops contains 0.639% weight by volume of eugenol, whereas the Jandu Tulsi drops contains 3.058% weight by volume of eugenol. So we have concluded that all the immunomodulatory preparations showed variable content of eugenol. That's it from my side. Thank you.
Yeah,
Don't
over to
mind.
you.
Yeah, I don't know if this is right to actually put the brand names like this, because these companies can actually take an objection if they are come to know that someone has done this. So, have you got any permission from them?
No, ma'am, actually, we are not aware of this fact.
Yeah, so maybe you can simply say brand A, brand B, brand C, rather than actually revealing the names, because this can act. I mean, though this is a very private presentation, we are within an organization, it is not public, but then, yes, henceforth, when you are presenting such brand names, you should be careful. People can actually take an objection. Yeah?
Okay. Sorry, ma'am, but I will take care of it next time,
Yes.
definitely.
One more thing I just wanted to add in with respect to same concern, that Jandu and Dabur is also a good company, and there is a huge difference in the concentration of
Yes.
the eugenol. So whatever the formulation parameters, is it same or different? Because this much difference should not be there.
Ma'am, actually, they have mentioned only the direction indication, that is how much drops we can consume in a glass of water or a cup of tea, like that.
Yeah, so there are many factors which are affecting the concentration of the ingredients. Okay, so whenever you are comparing, you should check all these parameters, because this is giving a wrong interpretations that two good companies with a huge difference in the quality. Okay,
Yeah, ma'am.
this is not the right comparison, I must say.
Okay.
And then you will never be able to publish something like this.
Oh, okay, ma'am.
Yes, shall we move ahead?
Yes, ma'am. PPG 37, are you there?
Yes.
Yeah, we'll go ahead with PPG 37. You can start.
Hello.
PPG
Am I
37.
audible?
Yes, you are audible.
So today's my presentation topic is bioanalytical HPTLC method for the simultaneous estimation of the empagliflozin and the atorvastatin in human plasma. So atorvastatin is a statin derivative which is used to treat the CVDs and the hyperlipidemia, which is a most common comorbid condition in patient having the diabetes mellitus, and empagliflozin is a gliflozin derivative which is commonly used to treat the diabetes.
These most are commonly used drug in this comorbid condition, and these are the highly dose dependent. Those are monitoring the plasma concentration is a very important. There are analytical methods are available for estimation of the empagliflozin and atorvastatin in biological metrics, but no method is reported yet to the simultaneous estimation in human plasma. So in present work, the extraction method is carried out using the plasma protein precipitation method by spiking the known aliquot of the drug in the plasma, and after extraction we have to collect the supernatant clear layer, and we have to spot it on the chromatographic plate. In this, the validation parameters which are performed as linearity, precision, accuracy, stability studies, recovery studies, carryover, and robustness. In this, we have to first compare the uh, blank plasma to check the interference at the RF of the drug. So the optimized mobile phase consists of the toluene ethyl acetate, methanol, and formic acid in the ratio of 12:5:3 and 0.5.
Okay, moving on. Ma'am, over to you, judges.
5:2. So the analytical method was developed for the simultaneous
Time is
estimation
up.
of
PPG
the
37, your time is up. Over to you, judges.
No questions from my side.
No questions from my side as well.
Okay, we'll go ahead. PPG 38.
Yes, am I audible?
Yes, you're audible.
Yes.
Yes, you can start.
Can I present?
It's already presented. You can start.
There's a change in the like the PDF. Wrong
Yeah,
PDF
sure.
was shared.
Yeah, sure. You can share.
Oh, thank you. Is my screen visible? Hello.
Yes, it is visible now.
Good afternoon, everyone. Today my topic is enhancement in solubility of drug using in silico tools. So for introduction, drug discovery process is a huge process and takes approximately around 12 to 15 years and lots of investment, that's in crores. So the drug under study is tadalafil, which is a class 2 BCS class 2 drug which has low solubility. It is used in the case of erectile dysfunction. The traditional method of solubility enhancement, which is done on trial and error basis, is a tedious process when compared to the in silico process. In this work, we have focused on enhancing the solubility using the in silico tools. Following is the method used. We have procured the SMILE from the PubChem and used in the Swiss ADME software, and it gave the results. It gives results on various parameters, but we have only focused on these three parameters, which are main. So first is the first parameter, which is in the result, is aqueous solubility. The aqueous solubility, it gives, according to three models, we have considered the silicose IT model. The silicose IT model considers the fragmentational solubility of the structure, and the enhanced solubility of the conjugate range from 7.29 to minus 4.36, and the actual solubility of the drug was minus 5.52. It was the score according to the silicose IT model, and we have screened approximately 50 conjugates. From them, 42% gave enhanced solubility and 58% gave reduced solubility. Second parameter which we have considered is the gastrointestinal absorption. Most of the conjugates had high gastrointestinal absorption, but 9 of them had low gastrointestinal absorption because they had increased that high molecular weight and structure. Third parameter was drug likeliness. In the drug likeliness, we have considered the Lipinski rule of 5, which contains molecular weight, hydrogen bond acceptor and donator, topological surface area, log P, and log P and one more parameter. From that, 29 conjugates only had 1 or 2 violations, and the lead likeliness of the conjugates was very high, which had only 1 or 2 violations. So for conclusion, the drug tadalafil the solubility of the drug tadalafil was increased, and the results were promising. So
Your time is up, PPG 38. Your time is up.
Just last points, last few 2 to 3 sentences. Can I?
It's not allowed. Sorry. Over to you, judges.
Thank you.
No questions.
No questions.
No questions.
Okay, moving ahead.
Thank you.
PPG 29, are you there?
Hello?
Yes, you're audible.
Yeah.
Please go to PPG 29. She had some technical issues.
Hello?
Yeah, PPG 29, you're audible. Just
yeah.
Yes, you can start.
Yeah. Hello everyone. Today's topic of my presentation is the role of herbal drug in COVID-19. The aim of this study to develop a new drug delivery by inhalation against the SARS-CoV by using a herbal drug like tulsi, curcumin, or gilo. Tulsi contain having the antimicrobial activity due to its containing the active constitution like eugenol or ursolic acid, etc. Also, curcumin having the antioxidant and cardioprotective activity, while the gilo having the main is a immunobooster. The herbal drug find wide use in the many medical application with less side effect. The purpose of work is made use the herbal combination containing curcumin, tulsi, and gilo, having less side effect by inhalation mode, like as some device like we use like as nebulizer, dry powder inhaler, spacer, meter dose inhaler that we can use. And finally, we conclude that the herbal nature compound with antiviral activity that causes the inhibition of the COVID host. And here we have provided the mechanistic best supporting and investigating approach for the herbal drug to be incorporated in inhaler.
Are you done, PPG?
Hello? Yeah.
So have you developed this formulation or is it just a theory, hypothesis?
No, ma'am, it's just theory. Okay.
So how do you propose to use these tulsi, giloy, curcumin in what form? As a formulation or extracts or crude drugs?
Yes, ma'am. I found I cross across some article like tulsi and curcumin gilo. We can extract them first and then drug incorporated into the nanoparticle, and then it's give by the inhalation routes.
Is there any reports showing that it can be given in the inhalation form?
No, no, ma'am. Actually, I can't across them.
Can we go ahead, ma'am?
Yes.
Thank you.
With this, we are done with the poster presentation competition. Yeah. I'm very grateful to the judges for their valuable time and support, without which this event would not have been possible. Thank you all. Thank you to all the participants and delegates. You have been very enthusiastic and cooperative for this event. It was really amazing to watch all the participants in their competitive spirit. It was really amazing to watch all the participants in their competitive spirit answering Q&A for their projects. Dear judges, I request you to please speak a few words and provide your valuable feedback for our participants. We would love to know your overall experience for this event. Yes, ma'am. Swati ma'am.
Thank you so much for giving this opportunity to judge this event. I am extremely thankful to BNCP and the management for inviting me here. And it was wonderful to see so many enthusiastic participants and everyone presenting with so much of zeal. It was really nice being here. However, a few suggestions for some of the participants. They can be more precise when they are presenting a poster. They can put up more figures and less of written matter, and especially when it is online, probably we can have it more descriptive in the sense, more illustrative with so many figures, and that would have been better. And it'll be really difficult for us. There have been how many, almost 40 participants to decide a winner amongst this. I think it'll take us some time. We'll need to probably discuss amongst ourselves and come up with a winner, and I'm sure we are going to find it very difficult to decide on a winner. And somehow I think the others should not be discouraged. Those who will not be winning, they should not be discouraged. They should still keep presenting with same enthusiasm. Thank you so much. Best of luck to all the participants.
Thank you, ma'am. Sarita ma'am.
Yeah, thank you so much. I congratulate each and every participants for doing a wonderful presentation. Presenting is something what I feel is something, everybody needs to learn over the years. So naturally, you all have given your best. There were a few wonderful topics which even we could learn from your presentations. So it was nice being here. I thank Munira ma'am and all the organizers, the team members to give us this opportunity to evaluate your work, and wishing you all the best. And thanks once again. It was nice being a part of this event. Thank you.
Thank you, ma'am. Manisha ma'am.
It was a good experience, and we saw the enthusiasm in all the participants. Yes, some of the topics were very novel, and I learned something new from those topics. Again, I'm thankful to entire team of SRC with such a good coordination, and principal madam and management for this type of events where students get motivation. Thank you all.
Thank you so much, ma'am. Thank you, judges. I would like to present certificate of appreciation to our judges. Thank you all.
Thank you once again.
Thank you, ma'am. Thank you all for your kind attention and participation. We may now have a short break of 30 minutes. I request you all to please join the same link for poster presentation of UG category after the break. I, your host, Akshita Geet, signing off. Thank you.
Good job, dear. Yeah.
Thank you, ma'am.
Thank you so much. And how would you like to know the result? What do? We mail that, fill that sheet and send it to you, whatever has been mailed to us by Dr. Praveed Kale?
Yes, ma'am. Just forward it to Lokesh, sir.
Lokesh, sir? Okay, so we'll fill up the marks in that and forward it to Lokesh, sir.
Yes.
Okay.
Thank you.
Thank you.
Thank you, ma'am.
Thank you so much. Can we leave?
Yes, ma'am.
Okay. Thank you. God, thank you so much. Please give this to sir and thank sir also. I'll give it to him.
Yes, ma'am.
I tried to leave the meeting. It's not leaving.
Good afternoon, everyone. As it is rightly said by Dr. APJ Abdul Kalam, creativity and imagination of the human mind would always be superior to any computer. I, Nikita Pase, your host for the UG poster presentation, welcome you all to the second Student Research Congress organized by SVKMs Dr. Bhanubhen Nanavati College of Pharmacy, co-hosted by University of Mumbai. Now we begin with the UG poster presentation, and our eminent judges for today are I request judges to please switch on their cameras.
Is this fine, Peter?
Yes, ma'am.
Yeah.
So now we begin with the UG poster presentation, and our eminent judges for today are Dr. Clara Fernandez, Associate Professor, Department of Pharmaceutics at Bombay College of Pharmacy, Mumbai. She has done her bachelor's in pharmacy from SNDT University as well as master's and doctoral degree from ICT University of Mumbai. She has more than 7 years of research experience, 20 publications including research papers, review articles and book chapters in journals and publishing house of repute. One granted Indian patent with 2 filed patents to her credit, and recipient of award research grants amounting to approximately 1 crore rupees. She is a recipient of Grand Challenge Canada Grant, stars in Global Health, Nutritional Supplement, Enriched Water Purifier as an investigator in 2017. Ma'am was also a finalist of GYTI Award 2019. She has won second prize for poster presentation for collaborative research work titled Fabrication and Characterization of Camellia sinensis based wound dressing films for low supporting wounds at one-day national seminar. Ma'am has also won Best Poster Award, and she serves as Associate Editor of Controlled Release Society CRS newsletter to a publication of CRS Indian chapter from 2008 to February 2012. Ma'am is currently a member of editorial board of APTI Women's Newsletter. Ma'am would like to welcome you with a floral bouquet.
Thank you very much for the honor. Thank you.
And our second judge is Dr. Radhika Raheja, ma'am, Assistant Professor, Department of Pharmaceutical Chemistry from SVKMs Dr. Bhanubhen Nanavati College of Pharmacy, Mumbai. She has done her PhD in Pharmaceutical Chemistry from SNDT Women's University, B.Pharm and M.Pharm from Bombay College of Pharmacy, University of Mumbai. She has academic years of 23 years and have 4 publications to her credit. She has been coordinator for NBA during the 2016-17 NBA accreditation for MET Institute of Pharmacy. She was a member of a local inquiry committee appointed by University of Mumbai for the continuation of recognition for PhD degree in the subject of pharmaceutical analysis at the Principal K.M. Kundanani College of Pharmacy. In MET Institute of Pharmacy, ma'am was also responsible for setting up the academic timetables. Ma'am has also been instrumental in organizing orientation programs on various occasions for syllabus framing and discussion. Ma'am has also worked as a paper setter, examiner, and moderator for organic chemistry and pharmaceutical analysis for both SYB Pharm as well as for final year B Pharm. Ma'am has also guided M.Pharm students and have also evaluated the M.Pharm thesis. Ma'am is the life member of Indian Association of Cancer Research, Maharashtra State Pharmacy Council, and Indian Pharmaceutical Association. Ma'am, would like to welcome you with a floral bouquet.
Thank you so much. So thank you both the judges for sparing your valuable time. Thank you, Dr. Clara. We know that it's a kind of a quite hectic time. Students' exams are near, a lot of activity at Bombay College of Pharmacy. In spite of that, you accepted our invite and you are with us today. And I'm sure your inputs and your comments will be quite helpful to all the UG students with your research inclination. I'm sure you will be able to show UG students a path to move on research activities. So thank you very much. Thank you,
It's my
Bhagwati.
pleasure. It's my pleasure.
Thanks, Doctor. And thanks, Bhagwati, for joining in. Yeah, go ahead, Nikita.
Yes, ma'am. I am grateful to receive this opportunity to introduce our judges to the audience and extend a very warm welcome. Before moving forward, I request participants to please rename themselves as that poster presentation code. Giving out any personal information like name, affiliation is strictly prohibited during presentation. Each presenting author will be given a time of 5 minutes, 2 minutes for presenting the poster and 3 minutes for question and answer session by our judges. Only one author will be allowed to present and answer the Q&A session. All the very best to our participants. So judges, with your permission, can we have our first poster presenter?
Please go ahead.
Yes. So PUG 1 with their titles, Diabetes Reversal via Gut. You may now proceed.
Hello, am I audible?
Yes,
Hello.
yes, you are audible. Go ahead.
Okay, so our poster, so we have to wait for the poster to present. Okay, so may I proceed, ma'am?
Yes, you may proceed.
Okay, so our poster as it presents, diabetes reversal via gut. The co-host of this poster is me and my partner. So we decided to give this disease because as we know that the Indians DNA is very having a very high risk factor for the diabetes 2, specifically for diabetes 2 patients. So and as we go from people to people, they visit the physicians, and the physicians normally advise them to do exercise, diet, and watch over their lifestyles and all that. And then they give them one medicine and two medicine, and then after the two medicines, they eventually comes to the insulin shot. So after each day of insulin shot, the body becomes resistant to insulin, which often leads. So for the treatment is that all the doctors are concentrating on control of glucose levels rather than we should think about something which would reverse the therapy. So we present to you the Revita device. This device is specifically used to improve the glucose control and reduce the insulin usage. The insulin resistance as being the driving effect for diabetes 2 as we speak, for the insulin resistance might actually lead to the high levels of insulin, which might lead to the diabetic hypoglycemia patients. To reverse the effects, the Revita device is used to resurface the gut, which is not at all related to pancreas
the gut helps in the releasing prolonged hormones when a food comes in contact with them, and these hormones which goes through the pancreas and release insulin. So due to the abnormality of these hormone releases, we have this metabolic disease like diabetes 2. As we continue from one step to the other.
One, your time is over.
Okay.
Over to you, judges.
Hello. Thank you for your presentation. Actually, I think you were not able to complete your the presentation completely, right? So could you please again tell me what exactly are you trying to communicate via your poster? Which technology are you trying to propose?
Means this technology is what it does, that there is a 45-minute surgery via endoscope, where they insert the endoscope through our mouth, and then they resurface our gut by providing the ablation at a particular temperature. So while doing this ablation, it reverses it alters the changes which might improve our metabolic activity. Since as the diabetes 2, we are having we do our work while sitting. We don't eat healthy things. So so our body don't get adapted to the digesting system. So as any problems in the digestive activity, which lead to any other number of metabolic diseases. So our idea for this is that there is a possibility, however, our medical science is very advanced, that we might be seeing the diseases in the wrong way or in the wrong organ.
So just only one thing, by ablation, you're damaging the intestine. Am I right?
Yeah.
So isn't that lead to another issue completely? Like to resolve one issue, and as you rightly said that we do not have much insight about the body. Still, we are in a discovery stage. And if you are saying that I'm using some ablation technology to burn or whatever, to scar or whatever for the intestine, won't that impact the other functions of your intestine?
Yeah. So means according to this device, they have done the survey, and among the survey, it not reverse the disease, but at least it improves the disease. It improves the insulin resistance. And they've tried this on a number of their applicants, and out of this, 2/3 of the applicants they didn't needed the insulin shots.
So increasing the insulin resistance or decreasing it?
Decreasing the insulin resistance.
Does it have anything to do with the manipulation of insulin resistance, or is it just helping the patient to have normal glucose levels?
The objective of this device to help them to not at all use the insulin and reverse the disease. But if it's still going on, the 10-year studies it's still going on. But the plus side of this device is that it at least improves the dependence of insulin on our body, the synthetic insulin that we use on our body.
Okay. The references that you have visited, what does it tell you about the frequency of this? Like, is this a one-time phenomena, or the patient has to repeatedly undergo this therapy or whatever, perform this ablation?
The survey which we read, according to the survey, they've examined the patient for 18 months, and out of these 18-month patients, 2/3 of them doesn't require insulin. 1/3 of them require the insulin. So it's a one time, mostly one time.
No, you didn't get the question which what ma'am has asked. Ma'am is asking is that for this ablation to work out, how frequently a particular patient has to visit a doctor. Okay, that is something which I think Radhika ma'am asked.
Correct. That's what I asked. How many times does this ablation have to be done?
Once only.
Only once, and that is sufficient for the patient to have steady blood sugar levels.
Yeah, not steady but improved at least. You can see that till now.
So how do you define improved?
I define improvements that and the point of the driving disease for diabetes 2 is the insulin resistance. Okay, and insulin resistance. And if we talk about diabetes, we talk about the increase and decrease in the glucose blood sugar level of the human being. So when the blood sugar level is increase or decrease due to the pathophysiology happening in our gut, the hormone release like incretins and all that. So if we somehow revert the changes which are causing the symptoms of the diabetes, then we can eventually prevent this effects of the diabetes and thereby reverse the disease.
What is the normal glucose level? When do you say the person has got diabetes? What are the levels in the blood which say that the person has got diabetes?
70 to
testing
131.
Postprandial, and we do the fasting glucose and everything, right? So how would
Yes, ma'am.
So what would be the normal levels according to, in that context I'm asking the question, how do you define this particular therapy is working? You'll be assessing in terms of glucose,
Okay,
right?
Yes, ma'am. So if we do this hydrothermal ablation, so the incretin and the various hormones which are produced by the gut, the intestine, the part of the intestine which we are focused is the duodenum, connected to pancreas as well as the liver. So various hormones are secreted and absorbed by the intestine. So this hydrothermal ablation can eventually lead to decrease in the blood sugar, which is postprandial, that is the absorption of the glucose after having food. And also, as the postprandial sugar level gets reduced, so the fasting sugar also gets reduced.
Radhika ma'am, I'm done with the questions.
Yeah, I think that's what we can go for the next poster. Thank you, boys. Good.
Yeah, thank you. All the best. Thank you.
Thank you, PUG01. Next we have is PUG02. PUG02, are you there? You can unmute yourself.
Yeah, am I audible?
Yes, now you're audible. Yes, please go ahead.
So a very warm afternoon to everyone present here. The topic for my poster presentation is redefining menstrual taboo with innovative hygienic products. Even though there has been very much awareness regarding menstruation, but it is still considered as a taboo in many parts of our country. Looking at the current scenario, we can see that out of the total 54% of menstruating population, 18% of females are still using unhygienic approach, while remaining 36% females are using sanitary napkins. But the question is, are these sanitary napkins really helpful? Because the conventional sanitary napkins being used are composed of 90% of plastic, which are having several environmental hazards as well as health hazards. In order to curb these harmful effects, there have come many advanced and innovative sanitary products like anionic pads. These help in balancing the pH as well as the hormonal levels. Then next is nano pads. These are based upon far infrared technology and they help in reducing the pain and uncomfortness. Then next is biodegradable antimicrobial pads, which possess the antimicrobial property. Then reusable menstrual cups, menstrual discs, and reusable pads. So these are eco-friendly as well as economic. Looking at the cost of the sanitary products being used in a lifetime, we can see that menstrual cups and discs are comparatively more cost effective than the normal sanitary pads and tampons being used. Moving forward to the next generation products like Loon Cups, MyFlow. So these products are basically designed on smart technology, and other than just holding the blood, they also give us the data regarding the bodily conditions. So in order to conclude, I would like to say that by adopting skin-friendly and eco-friendly product, it will be beneficial for our health as well as for the environment. And taking the right action today can help to bring the future of period products one step closer to reality. Thank you.
Thank you for a very interesting topic. Definitely a very interesting and very need of the hour. My query is regarding this Loon Cup. So you mentioned that it helps to give some prognosis about a particular patient's health or something. So how does it work?
So basically, the Loon Cup has three parts: the sensor, battery, and the antenna. And the sensor will give you the amount of like the how much volume of blood is collected. Then like from the color of the blood, it will give like is there any problem, disease related problems in your body. And it will also give you like the indication on your mobile phone, like whether the estimated time of your next coming periods.
Okay.
It will also help in seeing the basal body temperature condition. Average should be about 36.7%, and about the volume, it will detect the daily volume and hourly volumetrics. So for daily volumetrics, it will detect the diagnosis of the abnormal menstrual cycle, and hourly metrics, it will help in the bleeding, to detect the bleeding pattern, whether it is a heavy flow or little medium flow, etc.
Okay. This particular Loon Cup is designed by which company? Is it like under patent?
It is a company's name only, Loon Cup, and but it is not being used currently. Like it is being used, but the FDA approval is not yet given, so it is likely to be a future product.
Okay.
Yeah.
Another interesting product which you're showing out here is NanoPad.
Yeah.
So how does this particular, because you said it reduces pain and discomfort. So how does this work?
So nanopads are basically designed with the far infrared technology. So as we all know that far infrared technology helps in reducing the pain. So these pads emit far infrared waves, and therefore it will the waves resonate with our cellular functions, and they help in reducing the pain and uncomfortness.
Okay, so which is the in women you have different conditions. Okay, and every woman does not have a similar experience during the menstrual cycle. So if I were to ask you which particular consumer is being targeted by this particular product, any specific female related problems or something like that? Have you heard of something called people having this issues with fibroids? People who are having this kind of, I can't get a word now, where they have, Radhika ma'am can help me out here, but the people who have got this, intense pain during periods. So is this product specifically meant for them, or it's just a general category product?
Actually, during a survey for these nanopads, like from the available information, they had stated that by the use of the nanopads, the women who don't feel much pain during the period, for them it is like a normal experience. But for those who are having such troubles, there is reduction in the level of the pain.
I was coming to that point because every person has different experience. So in that context, yes. Another important thing which I saw is something called reusable menstrual cups, and a lot of I think advertisements going in this area. So you being as Indians, we are not so comfortable with exploring newer things. So how do you think will you convince a person to explore this option, reusable menstrual cups? And how it will be very easy for Indians to adapt to this particular aspect. It's cost effective, no doubt, but I think experience is something which you are not used to.
Yeah, for this, like every time a change is required. So I think from shifting from the synthetic, like we can convince people that the synthetic pads are very much harmful for their health as well as environment, but health is like people will feel something personally, and therefore by using menstrual cups, like it is it in the beginning it might be a bit uncomfortable, but once we get used to it, like especially if the youth gets used to it, then it can be used in future on a very wide scale. And also the looking at the sustainability, like these menstrual cups, they if used properly and preserved properly, they can be used up till 10 years, but we can consider like even if we use it for 1 to 2 years, it will be very much cost effective.
My last question then for you.
Yeah.
Now, considering that we are having a society, where there's a lot of sexual awareness, okay? Again, there is a possibility that obviously it's not really spoken a lot in Indian community per se, but there are people who might have STDs or they might have some kind of, bacterial infections. So amongst the products which you have proposed out here, which one do you think would be a viable option for such individuals who are having issues with STD or any kind of, female hygiene issues?
Okay, so the firstly, it is like one can use for antimicrobial pads, which will show the antimicrobial properties and it will kill the bacteria. And another interesting product is anionic pads. In anionic pads, whenever the bodily fluids get in contact with the pads, they release anions, and therefore they help in producing the oxygen, which will kill the anaerobic bacteria present. So even anionic pads can be used.
Okay, my question is, how do you say that these are eco-friendly? What is the material of these reusable menstrual cups or the loon cups?
So these both products are made from the silicone, medical grade silicone.
So does that make it eco-friendly?
It will make, because like we don't have to use it like for a they can be used for a longer time, therefore the disposal rate is being reduced.
So it is more of reducing waste than the safety of using of this material, right?
Yeah, but even the loon cups are giving you the other indications also about the body. So
Right. Now that is my next question. So if there are sensors, etc., for whatever, like for nano pads also you said IR sensors, and for loon cups also you said sensors. Disposal of these sensors, does it come under eco-friendly, the actual use of the term eco-friendly?
Nano pads are not having sensors as such. They are having different ions, like which are already available, like they are present in the environment. And regarding to the loon cups, these
But
are
you said that IR radiation helps to reduce the pain and all, right?
It is based upon FIR, but these radiations are given by the ions itself. Like it is the presence of a silver nano chip or something.
So silver nano, so there's a nano chip, right?
Yeah.
So is the disposal of these materials that are used for the making of sensors, does that make it eco-friendly?
It will but it is not used in a very much higher concentration, right?
But your normal pads do not have these components, right? In fact, whenever you dispose them, you are adding to the non-biodegradable waste, isn't it?
Yeah. Ma'am, I have to read about it then.
Correct.
Yeah.
Okay, then amongst the health hazards, what is RTI?
It is reproductive tract infection.
Acha. So how do these products help in reducing reproductive tract infections?
So basically, in comparison, if we see like the synthetic, this was in comparison with the synthetic products. So
Okay.
the plastic, it contains dioxin, plastic pads. So they contain dioxin, which is a carcinogenic. Then they contain various fragrance and deodorants, and these are synthetic in nature. So they cause the reaction, and also to give the pad a fresh look, like the whiter color, they are bleached with petrochemicals, which causes reproductive and urinary tract infections. And in these advanced products, the use of such products is like they are not used. So therefore, it will not cause reproductive tract infections.
Okay. Okay. Out of all these products, which are the ones that you find are seeming to be more acceptable by the female population? Are there any statistics available?
Like if we see, ma'am, I don't think it is visible in the market trends. If we see, then until now, majority of the people are using, most of the people from the advanced products are using menstrual cups, and even the anionic pads are also being used. But I'm not having a particular statistics about it, but I have heard from many, like people are opting for anionic pads.
Acha. Okay. Which brand available in India?
There is a P-Safe one is also available, and anionic pads, like there are many patented pads, so not a any specific one.
Patented is fine, but then it is selling in the market, right?
Yeah, there is one lady anionic pad available.
Achha. Okay. Okay, nice. This was a good informative poster. Thank you.
Thank you.
Thank you, PUG 2. Now we may proceed with PUG 3. PUG number 3, are you there? PUG 4, PUG number 4, are you present? Okay, so then we move forward with PUG 5. PUG number 5.
Yes.
Yes. So judges, please note we are moving forward with PUG number 5. Yes, you may go ahead, PUG 5.
Yes, just a minute. I'll start now.
Yes.
Good afternoon, everyone. Our topic for today's poster presentation is Artificial Intelligence, Innovations in Acne. Acne vulgaris is a common chronic skin disease involving the formation of comedones, papules, pustules, nodules, and cysts as a result of obstruction and inflammation of pilosebaceous units. A patient's perception of acne severity doesn't always align with the clinical assessment. For this reason, an artificial intelligence algorithm was developed for smartphones to evaluate the facial acne severity with the help of the European GEA scale. GEA stands for Global Evaluation Acne Scale that ranges from 0 to 5. It was used to identify different types of acne lesions and post-inflammatory hyperpigmentation. Data was collected from numerous countries like France, South Africa, China, and India. The suitability of the GEA scale was majorly studied in Black Africans and Asian population, as the problem of PIHP is more important in them than in Caucasians. This data was then segregated into different GEA grades. They were tagged as lesions or PIHP, which were then used to develop the algorithm. The fourth version of the algorithm was tested through clinical evaluation in acne patients. The expertise of the dermatologist given by face-to-face evaluation and that given by AIA were compared and later improved accordingly. The main limitation of the technology is the fact that the algorithm has been built on defined aims and variables. In order to consider all circumstances, the images were taken on different operating systems using visible light exposure. AIA has the potential to accurately diagnose the patient's acne and aid dermatologists and other medical practitioners to choose the most suitable treatment options. Thank you.
Thank you. So this data which was collected, was it done with the intention for only understanding the role of AI, or was it used for understanding the impact of a particular segment of medications on acne?
No, the data was collected with the aim to develop an algorithm, the artificial intelligence algorithm.
Okay. So, according to the survey, uh, was it able to distinguish between the age groups also? Like even sometimes you have acnes which are very old, depending upon the hormonal imbalances. Some could be because of poor habits or whatever. So you can distinguish between the stages of these acnes.
No, ma'am. It is just segregating the acne severity into different grades according to GEA scale.
Okay.
But why the need of artificial intelligence for this? Meaning, can a physician's diagnosis go wrong? What is the need?
Ma'am, the need over here is that rather than going to different dermatologists and getting different opinions, the AI uses a particular global set standard for evaluation. And it has been trained with various populations, so it is accurate. So you can get a proper result, which you may or may not get from other sources.
I think I'll extend what Radhika ma'am is saying. When you're talking about AI based, it is just not for evaluation. I think it is always this data is used for to understand the efficacy of any product. Okay, that's how the companies explore AI data. Because at the end of the day, when a doctor is using the medications, when the company is trying to market a product, okay, they use this data in along with the data what they observe when they do the studies. Okay, so I guess. This is going to help us understand that yes, by AI I can distinguish, but as ma'am said, you have to have a doctor on board to assess the outcome of a therapy. It cannot, it doesn't really take away that particular insight. Am I right, Radhika ma'am?
Yes, in fact, what I was interested in knowing was um, if a physician, a dermatologist misdiagnoses, let's say papules for cysts, okay, suppose he does that misdiagnosis, does the treatment differ? The treatment of nodules, is it different from treatment of papules? What is different in the treatment of these different forms of acne?
Ma'am, the treatment will vary will vary depending on the cause, or like the causes can be different, so the treatment will also vary accordingly.
So how many types of medication? See, what I'm trying to say is, if I'm replacing the judgment of a man by a machine, does that change the way the treatment was supposed to be directed? Because this is just a superficial diagnosis, isn't it? Your AI is only helping you for superficial diagnosis. If you have more than one reasons for acne, if it could be hormonal, it could be any kind of microbial, any kind of microbial infection leading to the same acne, your AI is not going to be capable of telling you that this is because of so and so microorganism and it is not because of hormonal balance, isn't it? It's only going to tell you what grade. So how does it assist in the physician taking the right decision for treatment? Was there any literature available for that?
No, ma'am.
Oh.
Ma'am, over here we are just trying to develop a system to segregate on the severity, which may help in diagnosis and checking the severity. And it has nothing to do with the treatment options and interfering with the dermatologist's job.
No, but the final thing has to be treatment, isn't it? If you're investing so much of money into designing a software that can help you to make predictions, it has to be put to some use, right?
How do we suppose this data would be useful? If you say this for diagnosis, I'm so sorry, I'm interrupting you. It is just for diagnosis, as what you're saying. How would you really harness this data? For what purpose would you use this data? Because as ma'am rightly said, with experience, you kind of assess this is a particular condition, this is an individual who's experiencing either mild or moderate, acne outbreaks. So how would you take this AI and make the whole concept of treating a patient more effective?
Ma'am, the system is developed on what a dermatologist has said. When the system was developed, it was fed with data collected and evaluated by dermatologists. So when the system was developed, like in artificial intelligence algorithm, when it was developed, what happens is you collect data, the pictures are graded. You first give the system labeled data. This picture is considered as grade 1, this picture is considered as grade 2. That is according to what a dermatologist has given. Later, when there is clinical evaluation, you give pictures and the system, as per what it knows, it tries to classify. So the system has been developed based on dermatologist evaluation. Over here, we are trying to just help in diagnosis.
Okay. Can I just give a suggestion out here? In COVID times, you must have realized that most of us could not visit doctors, right? Most it was teleconsultation. So do you think what is being proposed on this poster, will it help in actually, assisting doctors much better? Do you think so?
Yes, ma'am, because there is also this feature of localization, which uses a location and tries to identify the impact of external factors and occurrence, so you can help in diagnosis better, like other factors like pollution and climatic conditions.
Okay. So before I stop my question, again, I would like to ask you that you've done a very good presentation and definitely this is a way forward. Okay. How would you try and see AI? What is your thought process? How would AI help in changing the entire healthcare sector? What would be the impact of AI on healthcare sector, according to you?
Ma'am, as you said, during this COVID time, there's a lot of telediagnosis and online diagnosis and consultation with doctors. So AI can help in the diagnosis process, like this is developed for acne. Systems can be developed for other diseases for better evaluation and diagnosis.
Okay, I'm done. Radhika ma'am, if you have any questions.
Yeah, no, even I'm done. Thank you. Nice concept.
Thank you, PUG5. Now we move forward with our next poster presentation, PUG6. PUG6, are you present? PUG6? Okay, so then we move forward with PUG7.
Yes.
Are you there? Okay, yes. Judges, please note we are moving forward with PUG7. Yes, you may start.
Dreams and psychotherapeutic model of virtual reality in treatment of positive symptoms of schizophrenia are observed to be similar as that to the dreams, and the mechanism has been similar. Allow me to explain how. As we dream, the reprocessing of upsetting memories is initiated in a safe, stress-free environment of dreams, thereby allowing a safe outlet for emotions. Similarly, immersive VR creates computer-generated world and allows a tight control over stimulus presented. It is used to create situations that cannot be achieved in real-life situations by altering variables such as density, population, justice, density, population, and psychological factors. Persecutory delusions is found in 50 to 70% of patients suffering from schizophrenia, and the treatment of immersive virtual reality is used here. This treatment suggests entering feared situations virtually, where safety can be relearned by cognitive behavioral therapy after reduction in influential factors that keep on maintaining persecutory delusions, which are stress, worry, anxiety, etc. This has led to a significant reduction in persistent persecutory delusions. The effectiveness of psychotherapy is backed up by meta analysis conducted in the recent years, experimental design of which is as stated on the screen. Cognitive behavioral therapy is a part of psychotherapy that involves cognitive restructuring and enhancement of patient coping strategies. A set of 17 randomized controlled trials on efficacy of cognitive intervention targeting positive studies stated and
POG
shown.
is now 7. Your time is up.
No problem.
Judges, over to you.
Okay. So what you're proposing now, is it in practice or recent practice, or it was there for many years?
No,
How
ma'am, it
old is
is
technology?
coming on in recent practice. Yeah. So, firstly, meta-analysis were conducted only for the psychotherapy. For example, this is called as the Feeling Safe program, where the patients are given cognitive behavioral therapy and they are made to re-enter the situations they were fearing because of delusions or hallucinations. Now, this can be improved with the use of VR, because with the use of VR, we can create situations that cannot be created in real life. For example, we can alter variables like population density or suppose ethnicity if they fear a particular race. So that can be created and safely the patient can be shifted into that zone where they know that nothing of this is real and they can try fighting their thought process and yeah, that's it.
Okay. So I'm sorry, I couldn't get much of a poster, so I'm going to ask you. So was there any particular uh, any hospitals who have done this study or something where they have found that the patient was improving or the requirement of medications was reducing, anything of that sort?
Yes, in NHS, this study was carried out in UK in NHS, and they saw that when they combined pharmacotherapy along with the VR treatment, they saw that there were reductions in the hallucination rates, and there were, a stable decrease of persistent positive symptoms.
And that is for how many months were they observing this
They were doing
reduction?
this study for they were analyzing it on 3 gaps, which is like 0 months, 6 months, and 12 months. So they checked it all for on 6 months as well and 12 months as well. But one limitation was found that they, as when they followed up, the effects were seen to be reduced.
Okay. Because that's another thing which your mind kind of conditions itself to a new thing. Second thing, how frequently was this patient exposed to virtual reality? How frequently were they exposed during that entire treatment period? Was it once a day, twice a day?
It was weekly. Like they were provided weekly sessions of therapy. Like they were said that all of these things are not real things and they are just thoughts. They were conditioned into. They were provided with evidences that they are just hallucinating, and why they were hallucinating, they were made them understood. And after that, they were sent into virtual reality where they could go and face their fears and relearn safety measures.
So another thing which I kind of see, virtual reality is something which can also affect your behavior otherwise also. So the one who is, if you keep on conditioning somebody saying that it's not real, it's not real, and in reality something happens, it's like cry wolf. So was there any such impact on their normal behavior when people were exposed to virtual reality? They could have faced one of their fears, but what happened in other aspects of their life? Was it very impacted by using virtual reality? Any data on
There
that?
are, okay, so these studies are very few in number. That is the limitation, but they were very encouraging because positive results were seen. Now we need to carry on more studies as such so that we can understand if they are having any such impacts on their real life or not. This was basically done to reduce the load on the patient's relatives and the community they were in, because with increasing rate of patients, not everyone can be treated in the hospitals. So the burden was put onto the relatives and the community around them. So this was done in that aspect. Now to know further, like what more adversities can be caused, we need to find out.
Your experimental design talks about doing the study in adolescence. Is
Yeah.
was this so? But when you talk about schizophrenia or something, it
Mhm.
can manifest itself even in the later part of your life.
Right. This was done to understand if there was a genetic basis to that. So the twin studies were carried out, and a concordance rate of 0.49 was found in monozygotic twins, and a concordance rate of 0.17 was found in dizygotic twins. So
Mm.
this showed that there is a part of genetics, like genetic uh, value, but then environmental factors also play role, because even the like even the yeah the sorry
Was the gender considered out here? Gender?
gender considerations, yeah, it was found mainly in a little increased quantity in males than in females because of the estrogen levels.
Estrogen levels.
Yeah,
Male or female?
female. Like
Okay.
because females have estrogen levels,
Which is
it
a
ends
male
up.
initially. It's what? Okay, it's
Yeah.
female. And again, because it's adolescence, you're going to puberty, everything is, your hormonal imbalances are there. So, do you think that when they had taken a particular section, so were there hormonal analysis done earlier before they took up?
No.
Okay. Anyway, thank you, Radhika ma'am.
Yeah, I was just trying to think of what was the conclusion, meaning uh, how did this help the person to come to almost normalcy?
There was no almost normalcy, but there was some reduction in the persistent positive symptoms. Right. So if they were experiencing very severe hallucinations, they were brought down to a reduced level. There was no normalcy found, but they suggested that the use of VR, it eased the psychotherapy. Like instead of just telling them, and they were really sent onto that feared situation with the use of VR.
Okay. How many papers did you find talking about this?
Regarding psychotherapy alone, I find out a lot of papers, like they were dealing with the efficacy. Like suppose the meta-analysis, it includes 21 sets, right? So if like more than 100 papers were found. So these are 21 sets. The analysis was done on 21 sets of different types. Suppose the graph, it shows if high, I mean, the graph, it shows which all studies were taken in the meta-analysis and how they were done. And in the use of VR, like VR is recently discovering, so it is very new. There are not a lot of research papers on VR, but there are a lot of papers on efficacy of psychotherapy.
Because this is almost like customizing to every individual, right? The treatment will not be, it cannot be a group experiment the way we have clinical trials.
Right.
So the person on whom this experiment is being done is a standard as well as a test, because you cannot compare one individual with another.
Yes.
So, what were the criteria that were used by the physicians or the people performing these experiments to assess that this is making a difference? Is it only?
Criteria? Yeah. That
Is it?
was used as, okay, I'm
Go
sorry.
on.
Male and female that were experiencing more than 60% of hallucinations from the age of 16 to 50, all of them were taken, and the cognitive symptoms, they did not have to be like they should be able to read and write, to note down their experiences of how they felt when they were in VR or what did they analyze. So patients with not a lot of cognitive impairment but higher rates of hallucinations and delusions were taken into this study. And the mean age group was 40, 42.
No, what I meant is, on what basis did the physician or the person conducting this experiment conclude that this is making a difference?
Okay. It was their It
Because
was
the
reduction. Yeah,
Ah.
it was reduction in hallucination rates, like, or the severity of it.
So it was only a qualitative scale, no quantitative analysis.
No.
Okay, because in that case, if the subject is not reporting correctly, there is nothing to, there's no tool to understand whether this is really making a difference or not making a difference, right? So all the papers have been only talking about qualitative analysis. Is that so?
I have preferred 4 to 5 papers for this, like extensive study of 4 to 5 papers that were relating all of these. I have not yet, went through what other papers said. So what I found is that they talked about the, quality of life, how they improve the quality of life, or how the symptoms were reduced to an extent that they can, that along with the drugs, this can aid them. That's all.
Okay, so it's purely a
Qualitative.
qualitative analysis.
Yeah.
Okay, fine. That's it. Nice, good concept. Thank you.
Thank you, ma'am.
Thank you, PUG 7. Is PUG 6 there? So we can have your presentation now. PUG 6. PUG 6, now you are the co-host. You can unmute yourself.
Yes, I'm PUG 6.
Yes. Yes, you may proceed.
Yeah. Basically, my formulation is oral antifungal jelly, which is used to treat a fungal infection caused by yeast in esophagus or mouth. When excess quantity of yeast multiplication occurs inside our body, this type of fungal infection is called candidiasis. So this is the basic rough idea about this poster. And this was a project that was carried out by my MPharm student for about 3 months, in which I assisted her throughout. Basically, in market, there is no such formulation tried as semi-solid molded gelatin or pectin jelly containing a medicament. Especially for antifungal treatment, mostly syrups, tablets, and dosage forms are available. So considering the disadvantage of this marketed preparation and for increasing patient compliance and more therapeutic activity, this type of dosage form was basically formulated. Various clinical trials and studies were carried out before passing this jelly to the market. And there is no such jelly-like formulation currently present, and hence is more advantageous. This is a gelatin and pectin jelly in combination. And basically, the advantages of this jelly is that it can be used anytime, any place. The treatment can started or ended. It might end up being especially reasonable for foundational conveyance of medications which are vulnerable to digestion in the gut or in the liver. As the conveyance preferred route of medication through the oral mucosa, it can possibly defeat the issues of brief activity and varieties in medication discharge and maintenance time. The procedure was first we have to weigh all the ingredients, then we have to make a sugar syrup up to 6.67 grams and make it to 100 like the basic IP formulation. Then we have to steadily blend the gelling agent in it. When gelling agent totally breaks down, stabilizer and citrus syrup should be added to it to improve the non-abrasiveness and improve the pH. After boiling the solution, some preservatives and
your time is up. Judges, you may proceed for the question.
Was this work done by you?
Ma'am, I'll carry, I'll give to Suyash, who is my partner, who was also there. Just a second.
No, I actually only introduced your work. You mentioned done by MPharm students, so I couldn't get it. So.
Yeah, ma'am, can I continue, please?
Yeah, please go ahead.
Yeah. The project was started by an MPharm student, and as we are BPharm graduates, we are right now completing the project at that time. So we assisted him throughout the work because he often due to COVID pandemic and all, he had to move. He had to.
I got it. Thank you.
Yeah, yeah. So we
Okay.
assist him throughout the doing the experiments.
Okay, thank you very much.
So
Yeah.
this is
So
a
you,
research work. It's not a review.
No. It's a research work. We formulated a new type of jelly because
Okay,
currently
good.
in the market there are multiple types of preparations which are available, right, for antifungal properties. Because especially the things that are used currently are more in the form of syrups or in liquids, right? But the simple disadvantage of using these kinds of liquid formulations is that it is quick. Like if you take cough syrup for example, right? It when you drink it, it moves down your throat through the esophagus down to the stomach quickly. And even if it is thick, it doesn't. It is not there for extended period of time. But in that case, if we have a fungal infection in the mouth, okay, if in that case if we use a semi-solid preparation, it is going to dissolve slowly, right? So what it is going to do, it is going to give us an extended period of time more effect. Okay, so as it doesn't go down quickly, it is going to give us more effect for more period of time.
okay. Thank you again. It was Suyash, right? Suyash who spoke, right?
Yes, ma'am, I'm Suyash. And do you want me to explain the type of test
No,
that we
no, no,
carried
no,
out?
no, no. Because we don't have much time, so we'll have to just go ahead with the question. So the
No
first
worries, no
thing,
worries.
what I want to ask you is that when you were working with this drug, was this drug water soluble or water insoluble?
Ma'am, it was water soluble. Okay, we used it. We added it to the syrup, as we mentioned by Surbi earlier. She was my partner throughout the project. We dissolved it in the simple syrup, and there is the method of preparation, the poster that we have presented.
Okay,
We
but
added.
is this fluconazole water soluble?
Yes, ma'am. It was soluble, and it we prepared it with blending of jelly agents, and we made it semi-solid, so
Okay.
that it will sublimate when we put it in the mouth and give an extended period of time. And as mentioned, the advantages by Surbi, if it is over there for more period of time and it doesn't have any like a doctor doesn't need to review you while we are taking. For example, if there are intravenous preparations.
Okay. So let me ask you this question, and afterwards, if at all, you need to answer, just clarify this. So you're saying that you're looking at a product which stays in the mouth. How many hours or how many days you're looking at this product to stay in your mouth?
It is going to approximately stay over there, like from start to end of the jelly sublimation for around 30 minutes maximum.
Okay, so
Under
30
any condition.
minutes.
It won't
Okay.
exceed that
And
under any condition.
as per your thought process, as a team thought process, this
Yeah.
product is supposed to give you a localized therapy or it's going to give you systemic therapy?
No, it is mainly focused only on the local action. Okay, because as it is going to sublimate down your esophagus, it is going to provide localized action. But if we think about it from another perspective, it is at the end of the day going to move towards your stomach, right? So it just might provide action, but it won't be as effective as we are thinking about it locally. So it just might show action, but we haven't studied about the action it has provided, the antifungal action that it is going to provide to the stomach. We are mainly focused on the local action.
Okay.
Yeah, we carried out
And
the studies for IR for the excipient and drug stability, and they all came back good. So there weren't any issues over there.
how
And
much
we,
drug did you load in one jelly?
ma'am, can you please repeat the question?
How much drug did you load in that one jelly, whatever you've shown in the picture?
Yeah, ma'am, we tried multiple permutations and combinations for it, and depending on the type of action, we added up the drugs.
Well, again, I'm asking what is the dose? You can't do permutation combination when you're proposing
Yeah.
an antifungal or antimicrobial thing, right?
So you are asking for the exact dosage in a single jelly that we are going to use for the marketed preparation.
I'm asking when you have done this preparation, whatever
Yeah.
showing the pink color jelly thing you're showing on the
Yeah.
how much drug was there in that particular jelly, and on what basis did you decide this much drug I'm going to load in the jelly?
The 50 mg of fluconazole was added to it in total in a single jelly's uh, unit for the market preparation, right?
Okay. And what is the MIC of this particular fluconazole?
Ma'am, can you please elaborate on
What is the MIC? Okay.
Ma'am, I'm
Min-
an undergraduate student, so can you please like
no,
give the
it's not an undergraduate. That's what I'm asking you. It's not an undergraduate. Minimum inhibitory concentration.
minimum concentration in the jelly?
Inhibitory concentration required to kill the fungi.
Ma'am, we carried out the microbial processes, right? Like in our microbiology practicals, we carry out the antifungal studies for the same kind of thing. We carried out studies for fluconazole 2 of our jelly. For Candida albicans, the bacteria strains that was used, and the zone of inhibition that we found out was around 21.6, and the zone of error was plus minus 0.01. And ma'am, can I start the camera? I'll show you one of the pictures that we have of
Yeah, you
the
can start the
antifungal study.
camera. And this is my last question. Anyway, then Radhika ma'am will take over. Okay.
Yes, ma'am. Can I just flip the camera?
Yeah,
I'll show you the image.
yeah. Okay.
Yeah. Okay. Yeah. Can you see this? I don't
Yeah,
know
yes.
if, yeah. Okay.
Okay.
Yeah, I think that's visible. Yeah.
Okay.
Yeah, that was the zone of inhibition that we found.
Okay. Thank you, Suyash.
Yeah, that's more, I think, visible. Yeah.
Yeah.
Okay. Do you have any more questions, ma'am?
One important thing is, if this is
Yeah.
an MPharm project, it is in the wrong category. This is a UG thing. So an MPharm work should not have been presented here.
Ma'am, we assisted him throughout the project because like he started it at the start of his MPharm this, but due to lockdown he went over
No
to his
problem.
village. Right?
No
So we had
problem.
to cover
Actually,
everything right from the start. So like we had to.
That is fine, but it should have rather gone in the MPharm series, right? Not in the BPharm.
Okay,
Though
sure.
you
We'll take
assisted
that
him.
into consideration and we'll let them. Yeah.
Yeah, and even you
We
should
didn't
not
have
have
any
put
idea.
their names. You should not have put their names on the poster, right? It's all supposed to be unbiased analysis.
Yes, sure, ma'am. We'll take that into consideration next time. We are sorry for that.
Okay. Which year are you studying in?
Currently, I'm in the final year, and due to COVID pandemic, almost like the last 2 years were in lockdown. So we like came into colleges. We have just studied the basics and started doing practical work, and then everything fell back down.
Okay. If you say that fluconazole is water soluble, will this stay in the mouth for a longer time if it is water soluble?
Ma'am, the fluconazole has been distributed throughout the jelly, right? So the jelly mainly it starts sublimizing like within half a minute if you put it in the mouth. As I mentioned earlier, it stays there for a maximum of 30 minutes under any condition, right? So what we aimed at is when it is in solid form, there are no chances of any degradation, right? So what we aimed at was when it is in solid form, it is going to sublimate slowly. So as it is in solid form, it is going to stay inside the jelly and not like move, like go down the esophagus right away. It is going to go there in a slow and paced manner. So that was what we are targeting right there.
How does that compare with an oral fluconazole formulation? Efficacy wise?
Ma'am, as I mentioned earlier, we are getting prolonged effect, right? So what it does is, I think I mentioned the example of getting a syrup, a cough syrup earlier, right? So as we all know, a cough syrup is good. Like when you drink it, we feel good for maybe for 10 to 15 minutes because it just goes down the throat and it is doing action. And in a similar way, if we take a fluconazole liquid formulation, right, it is going to go down your throat for the same period of time, but it is not going to show an extended effect. Like the fluconazole, it needs some time to carry out its antifungal action. Yeah. Am I audible, ma'am?
Yes. Yes. Go on.
Yeah. So what we aimed at is that we have to get it more time because it is going to sublimate slowly. So as it has more time for exposure to your throat, it is ultimately going to show more action because it has like continuous hammering hone wala hai on your throat by the fluconazole against the fungus that is over there. Yeah, so it is going to get more time for to do its action if compared with the liquid formulation, because it is just going to go down in a single gulp. Even if we add thickeners for liquid formulations, right, it is not going to stay there for as long as so much time as the jelly is going to stay over there.
Okay.
Yeah.
Okay, good work. Thank you.
Thank you, ma'am. Thank you, ma'am. I hope I satisfied your queries.
Yeah, yeah.
Yeah.
All
Have
the
a good
best
day,
for
ma'am.
your future.
Thank you.
Judges, sorry for the interruption, but we have many more abstract lineup, almost 11 posters. So I request you, if we can, I'm really sorry for this interruption, but if we can move a little fast. Thank you very much.
Yes, sir. Yes.
Thank you, PUG 6. Now we move forward with our next poster presentation, PUG 9. PUG 8, sorry. We move forward with the poster PUG 8. PUG 8, are you there?
Yes, ma'am.
Yes, you may proceed.
So I am going to start my presentation. So good evening, everyone. Today I'm going to present on the topic artificial intelligence and cardiovascular disease, an unexpected alliance. Cardiovascular disease remains the leading cause of death despite the advancement in technology for diagnosis and treatment. Each year, an estimated 32% of the people worldwide and around 45% of the people in India die to cardiovascular disease, with one-third of a population above the age of 70. The risk factor includes unhealthy diet, obesity, use of alcohol, and tobacco. To improve and optimize cardiovascular outcomes, artificial intelligence and machine learning can be of a great use. Artificial intelligence is the ability to stimulate human intelligence in a computer system, and machine learning is a branch of artificial intelligence that focuses on the use of data and algorithm to evaluate human learning and gradually improving its accuracy. Deep learning is a type of a machine learning that mimics the operation of a human brain and includes a class of algorithm called neural network. This type of a learning is mainly used for cardiovascular diseases. Our primary focus is on application of artificial intelligence-based devices for cardiovascular health management, like ECG data acquisition hardware that can detect changes in the heart rate and conditions like sinus rhythm, trachyarrhythmia, and bradyarrhythmia. The key component in the system are user, hardware device, cloud services, and doctor. The second device is an Apple smartwatch, which measures unusual heart rate and can detect cardiac arrhythmias. Both of the device work on the principle of deep learning. The other application of AI is in cardiovascular disease include echocardiography, magnetic resonance imaging, cardiotomography. The limitation and the scope of artificial intelligence are listed in the poster. Thank you.
Thank you for the poster. It's very well informed poster. Appreciate that. What is the cost of this particular technology? I mean, if I were to compare with the cost of visiting a doctor, how do you think AI can reduce the cost?
Ma'am, when we manufacture this product or this hardware, it will be a one-time expense. Then afterwards, it can be used many times. We do not have to connect with the doctor every time, and the device which we have, we can use it right from our end, and the data it will be sent to a doctor, and it will be evaluated through it, and the result will be provided to both as a doctor as well as the user.
How instant would be the information process? Because what happens now is that though we have 2D echo and all these things, by the time it goes to a doctor, you spend one full day in getting the information or coming to a proper diagnosis. So using this AI watch, and the person could be anywhere in the world, okay, and if the data has been reaching the doctor, how fast would everything happen?
Ma'am, it is based on the new network, neural network. It is a very fast communication because it uses the internet access, and the data you get from your devices are directly connected to the doctor which you are connecting with, and the device hardware can be generated and used from all over around the world through the use of internet.
So do you think what we generally face issues with internet? So would that similar issues impact the outcome of this particular technology?
Ma'am, first we use the device and the data during the time we record the data. There is no need of any connection. For just we have to send the result to the doctor. Only that time we need some amount of a data to send the data to the doctor. And I think it is very easy to use it, and not lot of data is needed, only just the report of the patient is needed.
Thank you very much. Radhika ma'am?
Ma'am, any more questions?
Yes, I have one question. The question is, how are these machines calibrated? How can you be sure that the performance that you see today is equal to the performance that you see, say, 7 years from now?
So, ma'am, we take an example of Apple smartwatch. They have done this in 2022. They have done the clinical trials. They took 600 subjects and they performed the clinical trials on them, and they showed that the efficiency was very quite accurate, and yeah, ma'am, that's it.
So there's no upgradation of the software or something needed for better accuracy, anything of that kind? Like you have updates in your mobile, right?
Yeah, ma'am. Like Apple provides its own app for watches, so it gets regularly upgraded. It enables new interfaces to the Apple Watch, and it provides a good it gives a proper informative kind of feedback to the doctor as well.
Okay, are these also, or can these also be directly connected to hospital setups so that the
Yes, ma'am.
information from the patient directly
Yes, ma'am.
reaches
It
the
can be
hospital?
connected. Yes, ma'am. It can be connected.
Okay, that feature is there, or is it an integral part of the of the usage of this device?
Ma'am, it can be done through a software. Like sometimes the hospital does not uses any application, so they use different kinds of software to provide both kind of uh, the information that patient has given to the doctor.
Yes, somebody from the group wants to say something, I think. Somebody's put up in the chat. Anyone from the three of you wants to say something?
Yes, ma'am.
Okay, go on, go on. Go on. Probably he'll have to be unmuted, looks like. Nikita? Okay, anyway, one more question. Is this categorized as a medical device in regulatory scenario?
Yes, ma'am. The Apple Watch has been approved by FDA and it has been regulated. Apple has done a collaboration which stands for Medical University and it has been approved by FDA.
Okay, okay. Okay, thank you.
Thank you, ma'am.
Thank you, PUG 8. It's a kindly reminder. Participants, please note that only the presenting author can present the poster and answer the questions. So now we move forward to the next poster number, PUG 9. So PUG 9, are you present?
Yes, I'm there.
Yes.
I'll start.
Yes, we are just presenting your poster. Yes, PUG 8, you may proceed.
Good afternoon, respected judges and one and all present here. Our topic for the poster is applications of 3D printing in drug delivery. The 3D printing as an emerging technology and has enabled cascade of innovations and applications in diverse fields, including pharmaceuticals for drug delivery. Various methods such as fused deposition modeling, the most explored one, and stereolithography are used for 3D printing. Applications of 3D printing include the first one is microneedles. In order to enhance drug delivery with no pain, 3D printers are used to prepare accurate and precise size microneedles. Solid microneedles can be coated with API, which are then deposited upon application. Hollow microneedles can be used by adding an API after puncture. The second one is implants, which is more sophisticated drug release characteristics have been achieved by designing 3D printing based implants to have complex micro and macro structures in a single device and for multiple APIs. The third one is oral delivery system. It includes oral tablets whose drug release can be modified by modifying surface area to volume ratio by creating tablets in shapes, oral capsules using various polymers, buccal wafers, and personalized mouth guards that are printed and loaded. Polypill, which is a single tablet that includes combination of several drugs. The last one is topical drug delivery system. 3D printed face mask has been used for management of third-degree facial burns. Johnson & Johnson has also introduced the use of 3D printing in the production of customizable cosmetic face masks. Thereby, we conclude that 3D printing offers an excellent opportunity to allow pharmaceuticals to ease into the age of personalized medicine. Even though this technology shows promising results in the application of drug delivery, it is still in the developing stages with several areas that are being explored. Thank you.
Judges, we may now proceed with the question.
Okay, nice presentation. Besides these four things that you mentioned here, the microneedles, implants, and topical and oral drug delivery, is there any other system that... Oh, sorry, I was on mute. Any other drug delivery system being explored for 3D printing?
Yes, ma'am. So, like, there are transdermal patches which have been explored, and ma'am, so basically, I had read this article in which they had said about bioactive patches, which is based on pectin for treatment of ulcers and wounds, which is done by 3D bioprinting. So basically, like the main thing over there was that cotton bandages. They cause like dehydration of wounds, so because like they absorb moisture from the wound, so like the healing process is hampered. So therefore, like 3D printing technology, it helps to develop like freestanding transparent films which disintegrate upon the contact with the aqueous media. So this was like one of the applications that I read about.
Okay. Any specific diseases for which this technology is found to be useful?
So like ma'am, particularly implants that is useful for like treatment of osteosarcoma. So like basically the like the chemotherapeutic agent, which is cytoxan, it is like basically like delivered into the body by implants for treatment of osteosarcoma. And also anti-epileptic, like there was this like the first FDA approved tablet, which was Pritam. So that was used for that is basically an anti-epileptic which was used for treatment of epilepsy.
And what is the uh, duration of this? Meaning, the implant that is there, for how long does it have to stay in the system and deliver the drug?
So ma'am, like the implants by 3D printing, we can produce biodegradable implants as well. So like the normal implants, there are some type of implants which like you insert it into your body and then you have to go to the doctor to get it removed. But then like the advantage of these biodegradable implants are that they just like degrade in your body and it just gets excreted. And like in case of dental implants, that can be in the body for like 4 to 6 months.
4 to 6 months.
Yeah, like I mean, because that's the average time to heal.
Okay. Dr. Clara?
Yes, thank you, Dr. Radhika. When it comes to 3D printing, do you think it's going to help us have more personalized medicines?
Yes, ma'am. Like it'll definitely help because like you can make a particular drug deliver. Like suppose if you take an example of microneedle, so depending upon like several factors, you can like basically the needle size, the length of the needle, the tip angle of the needle that could be adjusted by drug delivery system depending upon how like the patient's need. So definitely like 3D printing, it can be used for personalizing medicine.
Which are the key process parameters one needs to keep in mind while exploring 3D printing? Like you must have done in your practicals, right? We have different mixing, different stages of developing formulations, and we have to be very careful while mixing all the steps. So out here, which are the key parameters one needs to be really focusing on?
So ma'am, like basically 3D printing is like more of like a software, like a technology based thing. So like, I mean, the main thing over here is that the software or the technology which is used for 3D printing, that should be like properly taken care of, like, you know, approved by some authority, some regulatory authority. So that's the key parameter. And I mean, obviously, if like you're like the size of the needle or something which you would be like you know putting into the software or the technology, like you'll have to make sure that it is correct because that could like those errors could be there. And yeah,
What about the mixing part of it? Because you have mentioned somewhere the polypill. So in polypills, you have different drugs which are coming into picture. So how would you ensure that the dosage, whatever you're printing, it has the right amount of drug? How do you assess that? And what kind of polymers can you print using this particular technology? Do you have any limitations on a particular polymer only can be printed, or you can use any other excipient whichever is being used regularly for our formulations? Do you have any restrictions?
so ma'am, like there are several methods of 3D printing, like fuse deposition modeling, stereolithographic, which is SLA, and SLS, which is selective laser sintering. So like all the three like methods have different applications, like different API and excipients or polymers or whatever, like could be printed with different methods. Like some of the polymers are like PLA, which is polylactic acid, PVA, which is polyvinyl alcohol. Like these could be printed by 3D printing. Like specifically these two polymers are used in mouth guards.
Thank you very much.
Thank you, ma'am.
I'm done, Radhika, ma'am.
Yeah, we can go to the next poster. Thank you.
Thank you, ma'am.
Thank you, PUG 9. Next poster presentation of PUG 10. So can we?
Yes.
Yes. Just a moment, we are presenting your poster.
Yes.
Yes, good afternoon all. My topic is on formulation of novel polymer based on buccal mucosal drug delivery system for nicotine replacement therapy. So, nicotine, we have been seen that all around 8 million cases are there because of nicotine deaths. So, the purpose of choosing the buccal route of administration, because buccal route of administration give oral bioavailability, which is good, and it avoids the first pass metabolism. So it is basically a more suitable route of drug delivery in such cases. We have performed this project on 9 batches from F1 to F9, and we found the F7 result to be ideal and match. For this project, we followed the solvent casting method, which is based on polymers, and we use polymers such as polythene glycol and hydroxy HPMC. And for analyzing the result, we use the design expert software, and also the spectrographic techniques such as FTIR and XRD. And the results were as this transparent patches we can see in the diagram. Thank you.
Judges, we may now proceed with the question.
Is this a research project, Miss?
Yes, ma'am. It is the ongoing project, sorry. This project is now on clinical trials. We are conducting our clinical trial for this project now.
You are in which year of your studies?
Final year B.Pharm.
So, is this project being done by MPharm students or is it being done by you?
Ma'am, I and my colleagues from final year have done this project.
As a part of your SEM 8 or how is it like?
Ma'am, actually due to COVID, we have been at home from 2 years. So we have started our SEM 8 project from SEM 7, made of SEM 7. So we have been doing this project in SEM 7.
Okay, and you're even planning to take it up for clinical trials?
Yes.
Okay. What are the advantages of buccal delivery system?
Ma'am, it has better oral bioavailability than oral route. It prevents the first pass metabolism, so we can ensure that all better drug absorption is there. So we have chosen this route for the nicotine replacement therapy.
Okay, any other advantages of going via the buccal delivery system for nicotine?
Ma'am, as it is easy to use also, and as nicotine has various side effects, it gives pleasant sensation to the brain, and also it affects some nervous neurotransmitter. So it is better if we directly absorb the drug in the mucous membrane, so we can avoid that side effects.
Is FTIR a good way of assessing interaction between drug and polymer?
Ma'am, it is good way, but XRD, we have seen the XRD result appropriate than FTIR.
No, but XRD, are these tools useful for assessing drug formulation interactions? FTIR or XRD, are they good tools?
Yes, ma'am, they are.
Okay, over to you, Dr. Clara.
First and foremost, have you done this work all by yourself in the last few days?
Sorry, ma'am, I couldn't hear you properly.
No, I said, have you done this work in the last few days or something? When did you do this work?
Yes, ma'am, in last few months.
Okay. Why is your drug content so low? What is the possible reason to have 67.3%, 38%?
Actually, ma'am, nicotine show the drug dependency effect on the body. So we have studied different doses, as you can see on the on the third diagram, nicotine level chart. We have studied different dosage form and saw different side effects on the body. So we studied that light side effects are seen only in 6 mg nicotine drug. So we decided to take the 6 mg drug as a result.
Okay. See, there's a difference in drug loading and drug content. What you are showing out is drug content, right? It's like an acid, right? So that doesn't matter how much you take in. Huh? You've got 67.3%. So I wanted to know why have you got so low over here? That's it.
Ma'am, can you repeat the question?
The question is that you have mentioned in drug content F7 is 67.38%, right? It doesn't matter how much you take 6 mg or 24 mg, whatever, it doesn't matter. The amount of drug present in your patches is 67%. So if I've taken around say 10 mg of my drug initially, you are saying I only have got 6.73 mg. So I wanted to know why you got this less drug content, nothing else. Okay. What is the relevance of XRD?
Ma'am, XRD is a technique of X-ray radiation. It helps in crystallographic structure to study the crystallographic structure.
That is single X-ray. This is something to do with regards to formulation, right? So why am I studying XRD of a formulation? What is the objective? Okay, no problem. Radhika, I'm done.
Okay, thank you. We can go to the next poster.
Thank you, ma'am.
Thank you, PUG 10. Next poster presentation we have of PUG 11. PUG 11, are you present?
Yes.
Yes, we are presenting your poster. Yes, you may proceed. PUG 11.
Yes. Good afternoon, one and all. My topic for today is 3D printed implants for local chemotherapy in osteosarcoma. Osteosarcoma is a rare malignant tumor of the skeleton originating from primitive mesenchymal bone-forming cells with treatment options like chemotherapy, which are effective, but the adverse effects of systemic high doses cannot be neglected. The implantable drug-loaded carriers for local chemotherapy can be used for controlled release and local administration, resulting in reduced systemic exposure to chemotherapy drugs. Fabrication of 3D printed drug-loaded implants can provide personalization, multiple drug-loading capacity, rapid and precise physical production, and cost efficiency. Moving on to the experimental details, poly-L-lactic acid or PLLA is an FDA-approved biodegradable biomaterial used for fabricating the implant prototypes, which were loaded with the chemotherapy drugs mentioned in the poster. In vivo pharmacokinetic studies showed peak concentration of all the 4 drugs after approximately 2 weeks and sustained release of the PLLA drug carriers for more than 12 weeks. The drug concentration around the site of implants was also found to be higher than in the whole blood, implying the presence of fewer adverse effects in normal non-cancerous tissues. Immunohistochemistry results showed lower levels of expression of osteosarcoma biomarkers in the experiment group treated with PLLA drug-loaded implants, implying that local chemotherapy with the assistance of 3D printed drug-loaded PLLA implants is much more effective than traditional chemotherapy. A limitation is the fact that the efficacy of this technique is yet to be tested in humans and is likely to be dependent and limited by the chemotherapy agents used. However, this technology has the potential of personalized local chemotherapy in the treatment of osteosarcoma. Thank you.
Is this a review?
Yes, ma'am, it's a review.
Okay, so how many papers did you read through for presenting this poster?
Ma'am, I read through 3 papers.
Okay, and all the 3 were from different labs?
Yes, ma'am.
So this graphs, the graphs that you have presented are from one of these, or it's
No,
from
ma'am,
the
the
same
graph
trend?
is from one of these. The testing was done in one. The rest showed effects of the rest showed possible future scopes.
Okay, okay. So the conclusions that came from all the 3 papers were on similar lines?
Yes, ma'am. The whole point is to get local chemotherapy to reduce systemic effects and reduce adverse drug reactions.
Okay. Any comparisons made of the use of this technology for delivery versus, say, targeted drug delivery or nanoparticles or something? Any comparisons made?
The graph over here, the bar graph, shows a comparison between the implants and normal therapy.
Implants and normal therapy, but no other targeted deliveries or any other things where the treatment is going to be localized?
No, ma'am.
Okay. Okay. And how it's still in the experimental stage? Are the papers very recent?
Yes, ma'am, the papers are recent. The papers are from 2019, 2020, the year, and some are from 2021 too. This technology is not just get tested in humans, it's just in the animal testing phase.
Okay, okay. That's it from me. Dr. Clara?
Okay, thank you. Did they give any idea as to how was this delivery system uh, designed using 3D printing, uh, overcoming the issues associated with drug, like aqueous solubility issues? Any mention of that in the paper?
Ma'am, we have used PLLA, which is a biodegradable material. So it's an FDA approved material. So it will dissolve itself in the where it's implanted. So there's no toxicity as such.
No, I'm asking about the drug because these drugs do have issues with water solubility. So how is this technology helping in overcoming that issue? All these drugs are not water soluble, right?
Yes,
They're
ma'am.
BCS class 2 or BCS class 4 drugs. Okay. I hope are you familiar with BCS class? If you're not, they're not water soluble. So
Yes,
by using this technology, how have they shown that they were able to overcome this drawback? Because when we're talking about, as ma'am mentioned, nano formulations, we use so many other excipients. So how has this technology helped in overcoming that limitation? Or did they have in addition to PLA, was there any other component used, other excipient used?
No, ma'am. There was a solvent used for adding the implant, DCM solvent, for making the implant. Other than that, no other solvent or excipient was used.
Okay. And what was the entire process? If you could just give just a brief idea how they went about doing this entire implant.
Okay. They fabricated the 3D print the PLLA implants, and then the drugs were added by soaking the drug mixture in the implants. There was a solvent DCM in which the drugs were dissolved, and they were and the solvent was vaporized on exposure to air, Further, it was decided in desiccator. Then there was in vitro and in vivo biosafety assessment of the implants by itself for checking the biodegradability and the cytotoxicity. Then there was in vivo pharmacokinetic studies, which tested the drug release kinetics and the therapeutic efficiency.
Okay, so this is only one thing. This is something out of your. You've not done the formulation, but I would just like to ask you one question. Whatever methodology you've explained to me at this point of time, do you think if I use a methodology on a large scale, will I get content uniformity every time? Because what you mentioned is that they have done the implant and they've allowed the implant to soak in a drug solution. Am I right? That's what you mentioned. So tell me whether I would have a content uniformity if I make 10 implants.
Ma'am, if I can speak like beyond this, there are other 3D printing techniques which can be used for adding the drugs to the implants. You can over here they've used stereolithography. Other than that, you can use fused deposition modeling and the use of hot melt extrusion to add the drugs.
Okay. So coming back to my question, do you think by using this technology, okay, this is not your work either ways, you're just explaining certain new concept. So I'm asking from a third person perspective, do you think this technology which they have described is going to assure you that every implant will have the right amount of drug in the right quantity? There'll be no changes. Just asking you as a pharmacist.
Ma'am, because they're soaking, so maybe not, but then there could be some changes made in the manufacturing.
I got the answer. Thank you very much. I got the answer. Thank you very much. Radhika ma'am will go ahead. Yeah, thank you very much. All the best. Bye-bye.
Thank you, ma'am.
Thank you, PUG 11. Now we move forward with our next poster presentation, PUG number 12.
Yeah, I'm 13, I think. If I'm not wrong, right?
Okay, PUG number 13. We move forward with PUG number 13. Judges, please note we are moving forward with the 13 number. Yes, you may go ahead.
Yeah, 1 second. I'll just share my video. Yeah. My video is on, right? Okay, see. Right. My topic is on sirtuins. So sirtuins are a family of compounds which actually is a deacetylases, and it is activated when the cell is under stress and it prevents epigenomic damage. So this paper actually deals with reversing aging by restoring useful epigenetic information. And it has been extensively documented that upregulation of these proteins called sirtuins actually increases lifespan of various organisms by an average of 50%. So although there are some controversies that exist to the degree of benefits, as cancers tend to exploit certain genes' genomic preservation property to enhance their cancerous nature. But nevertheless, there was a similar conundrum with Yamanaka factors. So using mouse and human models, they demonstrated here that Yamanaka factors, excluding c-Myc because it's a cancerous oncogene, it can actually restore the aging signature without resetting the cell identity. So when activated by a tightly controlled TET system, this results in restoring of the aging signature. The expression system with the OSK on, as in the middle you can see beside the Miser twins image, that actually results in 15-fold greater neurite area. So they also demonstrated that this system can actually reverse aging caused by physical damage when they did a nerve crush, as present on the left-hand side nerve crush image, right-hand side nerve crush image, sorry. Also the model can also reverse damage caused by age-based diseases such as glaucoma. So the vision was not just restored, the mice showed similar visual dexterity as younger mice, and their mRNA expression levels, expression profiles, and the DNA methylation patterns were reset to younger levels. So when the OSK is specifically switched on and in conjunction with SERTs, which is their histone deacetylase and other demethylases, they actually prevent aging. And a combination of these factors, only when they are present does this happen. When either one of them are knocked down, they cannot prevent the aging. So upregulation of SERTs by NMN, metformin, resveratrol can actually slow aging and further assist in organ regeneration, cancer prevention, reversing aging. So this is the paper which I am presenting.
Okay. Thank you. I can see in the references you have the oldest being 2011. So it's almost
Yeah.
a decade-old research.
Yeah. Yeah.
So any drugs in the market which are acting on this particular target?
Yeah, so that paper specifically is actually telling about the activity of SOTIs and general review paper on the activity of SOTIs. But this paper is actually recent in 2020. But nevertheless, answering your question, SOTIs actually, as I mentioned, NMN, metformin, resveratrol, all are in clinical trials and actually have metformin has already passed clinical trials, and it shows clear correlation that lower blood sugar levels actually always activate SOTIs because they increase the NAD plus levels in the blood, which activates SOTIs as a stress. It's a cellular stressor. So when the cell is under starvation, it activates it in all types of cells. So it's found in mice, humans, yeast, C. elegans, and even yeast. So yeah, those drugs are already present in the market, and there are a lot of clinical trials going on the same.
So any specific company which is working on drugs for this particular target, and if you can just tell us about which stage of clinical trial is that particular drug in?
So some of them are already finished clinical trials, but most of them are in stage 3 because they started about 5 years ago. But I'm not sure. I don't remember the name of the companies, but there are quite a lot of companies. If you actually follow the news on aging, even Jeff Bezos actually invested in a company recently which was specifically doing aging research. So there are a lot of companies which actually are doing it. I do not know the names specifically of them.
Because you mentioned it is useful in glaucoma, which is something a very difficult disease to even tackle to date.
Yeah.
So that is something which is very, very refreshing to hear this, and very encouraging.
Not just sorry to interrupt, but not just glaucoma. So all of the diseases, if you go to see, are actually caused by aging. If you go to as a cellular and molecular level. So if you prevent this, essentially, this is quite fascinating because if you go to see, actually we're going to reach aging escape velocity in 15 years, which actually means like anyone who is under the age of 40 can actually choose how long you want to live, because you can all of this will actually prevent diseases, any sort of aging diseases, cancers, everything can be actually extrapolated from aging disorders. So yeah, a lot of diseases can actually be cured. So there is a lot of potential on this. That's why billionaires are investing in this. But yeah, sorry.
Can I ask you a random question now? Since you're talking about repairs of the body via targeting this particular receptor, if I were to say that. Okay, during COVID-19, you must have heard of many things going really downwards for many people, especially in case of lung fibrosis or something. So do you kind of see the role of these receptors in helping people who are suffering from long-term effects of COVID-19? Do you think it's going to really be meaningful over there, this particular research?
There have actually been studies which actually showed that COVID-19 patients, when SERTs are activated, show a high recovery rate because SERTs inherently it's made by the body. Evolutionarily it is made because it is activated during stress. So anytime the cell is under any sort of stress, the SERTs just pile up the DNA. So it does not allow any transcription, any translation. It preserves everything. So no damage should occur to the cell. But then again, because of the cytokine storm, actually, I'm doing another paper on that. So over there also it clearly shows because of the inflammatory response, it is so tremendous that the cell cannot, no single cell can actually, resist this. It is inevitable death of cells, and it cannot, studies cannot reverse that amount of damage. So it can prevent, but it cannot reverse by itself. So that's the paper itself is mentioned, as I mentioned also. You need both of them, the SOX, the Yamanaka factors without CMEC to reverse the aging profile. You cannot reset it without SERTs and the Yamanaka factors. So
my last question to you is that there's always something called positive stress and negative stress. So anxiety, depression, all these are not a very desirable stress in the body. So do you think the expression would vary in such scenarios? And do you think that a person is going depression because they have slowed down, they're very much they have dementia and everything will coming up later in life. So do you think therein does this particular thing really work over there also? I just one other question after that. Sorry, I'll just stop over there.
Yes,
And any correlation of Ayurvedic formulations with this particular receptors? There is any such data which shows that whatever we are practicing as Indians in our food habits, they really do have more impact on these kind of receptors? That's it from my side.
Very fascinating answers. I will answer all of them. So, first of all, in base of the first was related to the receptors, right? Like in the sense how it can prevent. Sorry, could you repeat that first part? I just.
I asked you because you're talking about stress, right? So I'm talking about negative and positive stress. Yeah.
Yeah. So psychological stress. So there is no research based on psychological stress because it is not very easily measurable. In the sense, there's not psychology in and of itself is very subjective. But nevertheless, stressors in the sense physical stressors have known to actually activate SOTs, which if you go in actually colder places, if you take a bath in cold water or warm water, that actually activates SOTs. Exercising activates SOTs. So exercising is actually a very good method to activate SOTs by itself. So you don't need to take any medicines. Nevertheless, it stands to reason that it's actually shown, one of the research actually showed people with childhood trauma actually show down-regulated SOTs. So that is something related to psychology. So but I have not come across any papers which actually correlates psychological stress to SOTI activation directly. But that is one paper that I mentioned. And so second part, Ayurvedic. So one of the compounds which I mentioned, resveratrol, is actually typically found in wine skins, raisins. But nevertheless, it's in far too small concentrations. But yes, you cannot. It's, as you know, right? Why do we have drugs? Even though there'll be bioactive compounds will be there, which some drugs mimic. Why do we still take drugs? Because it needs to be at that particular concentration. So the concentration that's required is usually 1 gram of resveratrol. 1 gram of metformin also does the same thing. And even 0.5 grams of NMN in a 70 kilogram person, average weight person, will activate those SOTs. But for having that much amount of resveratrol from raisins, you'll have to eat way too much. And I don't think that's feasible. So but it is present in various compounds, such as raisins and grapes. Yeah.
Any information about liver cirrhosis and these proteins?
Liver cirrhosis. So yeah, even by, as I said, right, it is activated by any sort of cellular stressors. So even alcohol, it also increases the ROS generation. Typical liver cirrhosis ROS levels are typically very high. And so it actually prevents. So SOTs are actually activated. So one of the studies which I'm not allowed to mention the institution, but the studies actually see the SOT expression in zones of liver. So the zones of liver also show increased expression. So when you can actually upregulate SOTs to prevent all of these diseases in rat models, it's already been shown. So there is a lot of potential in prevention of these diseases in humans also. By activation of SOTs. So yeah, there is rat model studies, but not in human models. It's not been done, but in rats, yes.
Okay, thank you. We can go ahead. Good work.
Thank you, PUG13. That was really interesting one. So now we move forward with PUG14. PUG14, are you?
Yes.
Yes. Just a moment, we are presenting your poster. Yes.
Okay. So currently medications are developed especially for adult men, which means that all women and children have an excessive prescription for their bodies. This fact underlines the importance of personalized medicine. This statement was actually given by Fred Parati, the CEO of Multiple Lab. And I think we all agree, one size fits all actually does not hold good in treatment. This makes 3D print a powerful tool. 3D print is based on additive manufacturing, which involves the deposition of material in layer upon layer manner so that we can get a solid model. Various methods include such as stereolithography, selective laser sintering, fusion deposition modeling, inkjet printing, etc. Every method differs by means of component, like the way it is formed, what copolymers use, etc. The three mentioned here are most widely used. Of course, the prime motto was tailor-made dosage, but it also provides low cost and fast operating system. since every coin has two sides and so it has its own disadvantages. Applications include more accurate, safer, flexible, and faster disintegrating mini pills for pediatrics and geriatrics. And recently, poly pills has gained popularity. In traditional mechanism, doses cannot be modified. But here, the release profile can be varied by using a computer-aided design software, which transfers the necessary signal to a 3D printer, which then converts the computerized digital model into 2D section, which generates solid layers to build up the required object. So I'm sure 3D print will be a promising in future when installed in hospital and clinics. Due to it, there will be development in chemotherapy and transplantation. In a nutshell, once the challenges faced are tackled, we are towards a personalization. Thank you.
Oh, thank you. So we heard a lot of 3D printing. I think two of them already spoke about it. And you just now made a statement that it can be easily used in hospitals. Am I right?
Yes.
Yeah. So my question to you is that if at all, in any organization for that matter or any country, if people are able to do things on their own, then a particular profession gets impacted.
Yes.
So do you foresee that if we adopt 3D printing and if we make this technology so feasible that anyone can come up with medications and all you require is a doctor, and a doctor can make his or her own medications, will it impact a pharmacist?
Yes, I think it will impact pharmacists, but we can also include this in the pharmacy stores. Like
If it impacts the pharmacy and pharmacy profession, then do you think pharmacy as a profession would stay? Do you think our profession would be required then? If doctors can do, all they have to introduce is one more subject in their coursework, and then they are good. They can just simply do the formulations. Do you think this will impact? Do you think pharmacy profession will be endangered by having 3D printing as a technology?
Not exactly in danger that because we are still on the drug line, like we can give them drugs and everything. This can actually affect some, but I told you there are some disadvantages. Maybe in future we will get to use it.
Okay, Radhika ma'am, please go ahead.
Yeah, I think no questions from my end. Thank you.
Thank you, ma'am.
Thank you, PUG14. Next poster presentation we have of PUG15. PUG15, are you present?
Yes, I'm present.
Yes. Yes, you may go ahead.
Yeah, so my topic is inhaled insulin. Traditionally, insulin is administered subcutaneously, and there are major drawbacks to it. So it's time to switch to its alternatives. And the most promising here is pulmonary route because of the large surface area it has to offer. Now, the goals are definitely to maintain euglycemia, that is normal glucose levels, both prandial and postprandial. Now we'll go to the journey of the particle. So the particle once inhaled has to first counteract the defense mechanism. So it will get engulfed by the macrophages. Some of it will be left behind in the mucociliary clearance. The remainder will get down to the lung and will get dissolved in those alveolar fluids. From there, it will also slightly give a topical effect on the lung tissue. Not to forget, there are also proteolytic enzymes present, which may degrade the insulin up to a certain extent. And after all of this, it then gets systemically absorbed via the blood vessel. And the bioavailability here of the conventionally used inhaled insulin is around 15 to 22%. As evidently seen from the graph, the pharmacokinetic and the pharmacodynamic profile of inhaled insulin is similar to that of subcutaneously administered insulin. Now, there are various applications also of the inhaled insulin. So the first one to be approved by FDA was Exubera. Now, it is a dry powdered inhaler, and its major disadvantage was that it did not take into account the variations due to patients' breathing, which was an important factor. The second one is Afrisa, which showed advancement in the formulation technology. So they came up with a new drug delivery system, which is technosphere microparticles, and this enhanced the bioavailability of the inhaled insulin. And that's its advantage also. And the third one is AREX insulin diabetes management system, which is currently under phase 3 clinical trials. And the special feature about this is that it contains a breath activated microprocessor control unit. And this completely eradicates the patient's breathing variability, which was a major concern. Then there are some factors affecting it also. like aerodynamic diameter. So the ideal size is 3 micrometer. It can range between 1 to 5, but 3 is the ideal one. Surface morphology, it has to be spherical so that the particles are free flowing. Then the pattern of breathing, this needs to be deeply regulated for lung delivery. Formulation, definitely solids are more stable than liquids, but liquids have their own advantages. Like it's used in the AREX IDMS and it has enhanced viability equal to that of the subcutaneously administered insulin. There are some limitations also to it, and that is impaired.
PUG15, your time is up. Over to
Uh,
you.
the limitations we have mentioned out here is that it cannot be used for pediatric and geriatric patients. Am I right? And these are the populations which require a lot of specialized dosage form. Okay.
Yeah, so
And yeah, go ahead. Yeah,
okay,
I think
so
you got my
we could
question.
use
Yeah.
Exubera and Aprisa, but AREX IDMS tells that they can use them. So it's promised, but still it's under trials. And the first two could not be used because with advanced stage there will be changes in the lung structure and function. Like there will be changes in the diffusion, decreased alveolar area. So it can be used, but then it has to be used with caution. So you can just randomly assign it.
So the last one which can be used is saying Novonordics. But out there, what I can see there is a special feature mentions breath activated microprocessor control device. Now, in case of geriatric populations specifically, okay, they generally, as you know, they might have a poor lung function. So, does this particular technology provide the same amount of insulin?
Not for them yet. Not for them yet. Below 5 years, it's not. The technology is not so advanced yet for them to deliver the drug.
So,
Because of their increased breathing rate, they could, in fact, absorb a lot of drug, and that could be dangerous. So it should be avoided in them.
another thing, when you say insulin, you know that our body has insulin receptors. Okay. So, how does this technology actually prevent any non-specific interactions?
So it's also given in addition to the oral hypoglycemic agent, so it will maintain those glucose levels. And it normally, like there have been studies which say that it gives target delivery. So there's no randomization here.
But the studies would have not been done in the actual population, right? They would have
No,
done.
there have been 4 studies which have been done, which include 25, 37, 73, and around 311 patients. And all of them showed it safe and it can be tolerated well. And in fact, they even showed patient compliance for it.
Okay.
They were fine, and there was no such adverse effects.
Okay. Ma'am?
What I have been thinking is how does this formulation withstand people which have known lung diseases of any type?
So it should not be used in them. Like that's a limitation. So if you have COPD or asthma, then it should not be used. Otherwise, there will be decreased absorption. So if it's fine, like with the decreased bioavailability, I'm also giving a hypoglycemic agent and then I'm maintaining my glucose levels, then it's fine. But it should be thought upon before giving.
Yeah. What about people who also smoke? How about those kinds of population?
Yeah, so smoking causes nicotine deposition, and nicotine increases the permeability of the alveolar capillary membrane. And so these particles, being small enough, are able to go inside in increased amounts. And so there is more absorption here, threefold increase, in fact.
So desirable or undesirable?
Undesirable.
Undesirable. Okay. Okay. Thank you.
Thank you, ma'am.
Thank you, PUG15. Next up, we have PUG number 16. PUG16, are you present?
Yes.
Yes, you may proceed.
Good evening, one and all present here. For many years, genes have been the common denominator in determining how we look or what our hair type will be like. Well, thanks to the new technologies, genes are no longer the only criteria in determining how a person looks or what their hair type will be like. We all use shampoos and hair mists on our hair, don't we? But have you ever thought whether these products are actually repairing and strengthening your hair or damaging it even more? Well, let me tell you something. Some of these products are actually damaging your hair rather than repairing or healthifying it. Something similar was observed by a girl named Violet who lives in the extreme weather conditions of the polluted city Delhi. She's just a consumer like us and was jaded by the unhealthy nature of her hair. After doing some research, she got to know that the culprit behind it all was a substance named vegetable. Some companies saw it too, and finally, a company named SVKMs implemented the shift of synthetic silicon to vegetable silicon or vegetamide. Now, vegetamide or vegetable silicon is a plant-derived liquid cuticle being the outer cellular layer of the hair, and hence this needs to be protected. This is where the F layer comes in picture. The F layer or the fatty acid layer acts as a barrier structure and protects the cuticle of the hair from external damages. This barrier structure breaks when there are any chemical damages to the hair. When such a hair sample is treated with vegetamide, it forms a pseudo-cuticle F layer and acts as a natural repairing agent. The structure of vegetamide is very similar to that of the healthy hair. In healthy hair, the natural F layer consists of 18 MEA and protein, which is recreated by vegetamide as pseudo-18 MEA and plant protein. The plant protein is derived either from pea protein or rice protein. Also, the vegetamide structure consists of terminal carboxylic acid connected to C15 to C16 hydrocarbon chains, giving vegetamide its hydrophobic character. As we look at Violet's hair, we notice that they're all frizzy and fuzzy externally as well as totally damaged internally. To have a better understanding about the difference between healthy hair and damaged hair, we can carry out few experiments stated on the poster in the second column. Now, let's look at the repairing effect that vegetamide will provide to Violet's hair after she applies it. To begin with, it repairs the cuticle structure and hair surface by repairing the cuticle lifter. It restores smoothness in hair, which is mainly due to decrease in friction of hair fibers. Hydrophobic molecules do not produce a static electric field, which is exactly what vegetamide does. If the static effect is large, it leads to frizzy and fuzzy hair. The picture on the extreme right of the poster depicts how the static effect is considerably decreased in hair treated with vegetamide. Now, we're going to look at some applications of vegetamide incorporated in hair care products.
Your time is up. Judges, we can continue with the question.
Does this particular vegetable silicone, what you mentioned, does it prevent split ends? If your hair has split ends, does it repair that?
It is mostly, it is not used for split ends. Rather, it will replenish your hair. Basically, if your hair is damaged, it is frizzy, and if it is unhealthy, then it will repair your hair. It will restore the F layer. It won't cut off or it is not a treatment for split ends. Rather, it is just for healthifying your hair, making it more hydrophobic in nature. That is what it is used for.
And what about its washability? Whether it remains on the hair for longer time? Does it make the hair greasy? Does it interfere with your normal hair function?
Ma'am, exactly the point of this poster, synthetic silicone, if we see, it is firstly, it is a little greasy and stays on the hair. Rather, it builds up over the period of time. Whereas if you talk about vegetable silicone or vegetamide, it does not build up on hair. It only selectively absorbs on the areas where it needs repair. And yes, ma'am, that is all about it. So so to answer your question, it does not build up. It is not greasy and it is very easy to use. It is just like a normal shampoo you use, but with a better benefits.
Are there any reports of any form of skin irritation using this particular component?
As of now, according to our research, there hasn't been any problem. In fact, this is much more better than synthetic silicone because it is all natural. And yes, ma'am.
Any marketed products which have this particular component?
Yes, ma'am. So there is a shampoo by Raw, which uses pea protein in them. There is a shampoo by Requil, which uses pea protein in them again, ma'am. So these are the two marketed products.
And the percentage required to achieve this effect?
Sorry, ma'am?
Percentage required, you know?
Ma'am, it is like 50 grams of 50 ml of the product is required. For every 1000 ml of the product, we need 50 ml of vegetamide.
And stability, how is the stability of this particular compound?
Stability as in, ma'am?
If you use it as shampoo, does it undergo any form of breakdown, any form of oxidation, hydrolysis, anything of that sort is mentioned?
No, ma'am. Hydrolysis it cannot because it is hydrophobic in nature. It won't undergo hydrolysis and any sort of it is quite stable that way. So there have been no reports of it being unstable anytime or undergoing oxidation.
Any interaction with any colors?
No, ma'am.
Okay.
It has
I'm
no
done now.
interactions.
I'm done, Radhika ma'am.
Does it has it been reported to cause any kind of hair fall issues as well? The formulation containing vegetable silicon?
No, ma'am. In fact, the exact opposite. It has actually shown hair growth, and in fact, it is even better. It is just like the normal shampoos which we use has synthetic silicon in them. This is much better because it is all natural. It contains natural protein from the plant, so it will actually more improvise the condition of hair. In fact, the normal shampoos that we use have shown to cause hair fall, but not the shampoos containing vegetable.
Okay, what are those top pictures that you've tried to put above applications? What do they depict?
Yes, ma'am. So, like, if as I said in my speech, the girl, we have named the character as Violet, and she is a girl from the very polluted city Delhi. So, as you can see, the first pictogram, her hair is all frizzy, and she's obviously very jaded by the quality of her hair.
Wait. Above applications, there are two pictures there.
Yes, ma'am.
So what are they depicting? Because they're
Yes,
too
ma'am. So,
tiny for us to see.
yes, ma'am. So I'll tell you. So the first picture that you see on the extreme right had the picture with the bubble. It's actually an experimental picture. That depicts that when the contact angle between the water molecule and the hair strand. So as I like the hair would be classified into two categories here. One will be unhealthy hair and the other one will be healthy hair. So if the hair is unhealthy, the contact angle it will make with the water droplet, like you can see in the second picture, it will be less than 90 degree. And if the hair is healthy, it will make a contact angle of more than 90 degree. And about the second picture, or exactly on the same part, the second picture depicts how vegetamine reduces the static effect. Static effect meaning it will reduce the frizziness of the hair. The hair won't repel each other because of the static effect. The frizziness in the hair increases and the hair starts to repel each other, making it look really bad. So the right side of the picture depicts that when a hair is treated with vegetamine for a few days, it the frizziness goes away and it looks really smooth and nice.
Okay, thank you. Nice
Thank
work.
you, ma'am.
Yeah,
Thank
we
you,
can
ma'am.
go ahead.
Thank you, PUG 16. Now we move forward with our next poster presentation, number PUG 17.
Yes.
Yes, you may proceed.
Good evening, all. My topic is e-cigarettes replacement as a first step towards quitting. So, the tobacco use worldwide has become the largest cause of preventable death globally. The pharmacy profession has played its part by introducing nicotine replacement therapies and drugs, but they have found to show their own side effects and lower success rates. Another relatively newer approach for smoking cessation is the use of electronic cigarettes, which are basically battery operated devices that uses a metal resistance coil to heat a nicotine solution instead of burning tobacco. The device was originally designed to resemble a conventional cigarette. It consists of a mouthpiece connected to a tank containing a solution. This solution is called an e-liquid, which contains nicotine, a solvent system, along with flavorants. It also consists of an atomizer, which aerosolizes the e-liquid, and a power source. On comparison with NRTs, e-cigarettes were found to show many advantages such as greater adherence, cost effectiveness, lower irritation of throat and coughing, and also decreased urge to smoke. However, NRTs were found to have the least cardiovascular risk. In an in vitro cytotoxicity assessment, it was found there was a comparison between the two commercially available e-cigarettes and the regular combustible cigarettes, and it was found that the regular cigarettes were found to show a sharp decrease in cellular viability on increased exposure time. However, the two e-cigarette brands did not show any significant cytotoxicity. The disadvantages of e-cigarettes are mostly related to its misuse. Many cigarette users, up to 89% of e-cigarette users,
were found to be dual users of e-cigarettes along with combustible cigarettes instead of using it just as a cessation tool. Therefore, it potentiates the nicotine addiction. Also, due to its recent introduction, it also does not have any long-term clinical trials suggesting that their role in chronic diseases. Due to all these reasons, there is a great variation to which e-cigarettes are supported or restricted worldwide. For example, in India, there is a great
UG
confusion.
17, your time is up. Just we can have the question now.
I think on your poster, it's kind of you have discussed the controversies which I wanted to ask you. Okay, what will be your first after you have done the research? So, do you propose e-cigarettes should be allowed in India?
Yes, ma'am, but they has to be highly regulated. The nicotine content in each e-cigarette should be regulated just like it was done in UK. They have regulated it to 20 mg per ml in each e-cigarette. And also, if it is made instead of a OTC product, if it is made a prescription product, then I think less number of users will be there.
Another important thing you've mentioned, they have used propylene glycol, glycerol, and all the other flavors. So when you're using this particular e-cigarette, what is the impact on the stability of this propylene glycol or glycerol? That is one question. Second thing, sometimes there are certain articles which say propylene glycol is also carcinogenic. So do you think, though we're trying to remove one addiction, but you're also somewhere compromising on the health of an individual? So is this a good option, or you can modify it further so that you can achieve the benefits at the same time, do not make the person more unhealthy?
Yes, ma'am. For the first question, ma'am, the shelf life of one e-cigarette was found to be 2 years. So I think they are relatively more stable. And also, in case of propylene glycol and glycerol, these are excipients that we even use in our pharmaceutical products, and they are by FDA. They are recommended as generally recognized as safe. However, there are no toxicity studies to show their effect via inhalational route.
Radhika, I'm done.
One important advantage of e-cigarette over traditional cigarettes.
Ma'am, along with their health effects, they are also very cost effective. Like they have a long battery life. They contain lithium batteries, which are also rechargeable. And the tank containing the nicotine solution, it can be refilled as many times as you can. So it has cost effectiveness. And also in terms of
you're talking pro tobacco users. Huh? I'm asking you about what about the passive smokers?
Yes, ma'am. It still contains carcinogenic compounds such as carbonyl compounds, nitrosamines, and everything. But a study found that the quantities were up to 50 times lesser than regular cigarettes. So they are definitely not a harm-free alternative, but they are definitely less harmful than conventional cigarettes.
Okay, thank you.
Thank you.
Thank you, PUG 17. Up next, we have PUG 18. PUG 18, are you present?
Yes.
Okay, you may proceed.
Our topic for today is recent trends in hair care products, botanical keratin. We all know that peptides are the most essential when it comes to hair care products because it supplies proteins which are lost from hair by damage. Keratin is the most abundant peptide. Many people are against animal-based products, and hence this creates a huge market value for organic products like botanical keratin. The reason for having chosen botanical keratin is the fact that it is cruelty-free, paraben-free, non-GMO, and allergen-free. When comparing the structure of conventional keratin and botanical keratin, the amino acid composition turns out to be very similar as shown in the poster. Depending on the formulation and the cost bracket, one can choose to use either the liquid form Promois WJ or the powder form Promois WJ SP. The recommended dosage range is 0.5 to 3%, which is an advantage. And you can expect more efficacy even with less dosage. Botanical keratin, due to its low molecular weight, penetrates into the hair and repairs the hair damage from the inside. It fills up into the damaged part of hair surface and repairs while making them silky, stronger, and more manageable. With daily application, it makes the hair healthy and has no adverse effects. Studies show 25% extra moisturizing effect on damaged hair and increases the tensile strength of hair while reducing the hair fall. It finds use in shampoos, conditioners, hairstyling products, hair masks, and any other leave-on and rinse-off hair care products. So to summarize with, this botanical keratin is the best multifunctional ingredient to use in all possible formats of hair care products. It not only repairs but also makes hair more beautiful by giving luster, smoothness, and manageability. Owing to its natural origin, promised performances, safety for daily use, and for all obvious reasons, botanical keratin is a recent trend in hair care and is being used by majority of companies in the global market. Thank you.
How is this botanical keratin extracted? What is the method of extraction?
The botanical keratin is hydrolyzed pea protein.
So what's the process involved?
So the process is not clearly mentioned, but it is dried and then the starch and the fiber is removed, and then here the main thing is we need to extract the amino acids from the pea protein. So that is done.
What is the possible interaction with the other components of the hair shampoo? Are there any interactions known or reported?
No, ma'am. It is stable and it does not have any adverse effects.
Okay. Radhika ma'am.
Were you there for the previous poster, miss? Did you attend the entire session?
Yes, ma'am.
So did you did you hear the vegetable silicone poster?
Yes, ma'am.
So how is this different from a vegetable silicone?
So, ma'am, keratin is derived from it's hydrolyzed pea protein, whereas that is not related to this.
Wasn't that also from pea?
Ma'am, but here we are trying. Yes, ma'am. But here we are trying to compare the advantage and the mechanism of the botanical keratin over the conventional keratin that's being used.
Okay. Again, does this promote hair growth or promotes hair fall, or what does it do? It only prevents hair damage. That's it. That's the only purpose.
No, ma'am. Along with preventing hair damage, it also is shown, like the studies show that it provides 125% extra moisturizing effect, and it increases the tensile strength. So it reduces the hair fall. And not only that, it makes hair beautiful by giving it the luster, smoothness, and the manageability.
Okay. Any effect of hard water or soft water on the use of this vegetable keratin?
Ma'am, it was not mentioned in the literature.
Okay. Okay. Thank you.
Thank you, ma'am.
Thank you, PUG 18. Now we have with us the last poster presentation, PUG number 19. PUG 19, are you present?
Yes, yes.
Can I start?
Yes, you may proceed.
Yeah. So let's talk about upconversion nanoparticles. These nanoparticles basically exhibit a photon upconversion, which means two or more incident photons are absorbed and they are converted into photons with higher energy. There are several components of UCNPs like host matrix, sensitizer, and activator, but we'll be focusing on the luminescent mechanism. Since we usually use lanthanide doped UCNPs, and we know that trivalent lanthanide ions, they have their 4F energy levels filled. So when these trivalent lanthanide ions produce basic upconvergent luminescence when the electrons jump between the 4F energy levels. There are several applications of UCNPs which are listed, but we'll be focusing more on imaging and drug delivery. In the imaging part, the 6 diagrams which are shown, the A, B, and C part, they have quantum dots as a nanoparticle which was injected, and D, E, F has upconversion nanoparticles which was injected. So basically, like sodium yttrium tetrafluoride UCNP, which was doped with orbium, was injected in the DEF mice, and they have reported to have a better contrast, which is clearly visible as compared to the quantum dots, stating that UCNPs are a better alternative for in vivo imaging. And in the drug delivery part, according to the literature survey, UCNP-based drug delivery systems have reported to improve the efficacy while reducing the cell death and several adverse effects. One of the research work is stated wherein UCNPs with doxorubicin, which is a chemotherapeutic medicament, was encapsulated in a starch-based gel and decorated with several other particles, which leads to construction of a UCNP-based drug delivery system, which had superior anti-cancer therapeutic effect with a combination of ferroptosis and apoptosis, but they had few side effects as well. So to conclude, the advantages, the properties of UCNPs, which are like no background autofluorescence or minimal background tissue damage, will keep the UCNPs at the forefront in imaging and drug delivery. We also need to understand that there are certain surface modifications which are required in UCNPs, which can lead to which will lead to improved upconversion efficiency in UCNPs. Thank you.
UD19, your time is up. Over to you, judges.
Am I audible?
Yes, yes.
Hello. Okay. I just had a query. I think you have mentioned in your conclusion anyway, but tell me one thing, when you're talking about drug delivery system, how is the loading done? How do you, can you just give an idea as to how is the drug distributed in these nanoparticles? Is it surface absorption or whether it is inside? How it is done?
Sorry, you're not audible. Can you repeat the question again?
Yeah, I'm asking, like, when it is used in a drug delivery system,
Yes.
how is the drug distributed in the entire nanoparticles?
So
It's in the
basically,
compounds, yeah.
yeah. So it's a 6-step process which I have listed it down. In this one particularly, there was a nanolongan carrier which was made by using UCNPs as a core particle and DOCs, which was coated with the polymer.
Okay. And how different from quantum dots? What is the advantage or disadvantage of these nanoparticles over quantum dots?
First, talking about the luminescent part, it is clearly visible in the imaging that the luminescent part is clearly visible, better and efficient in upconversion as compared to quantum dots. And the this UCNPs were placed 10 millimeters beneath the skin in the rats. This was depicted, and at the same time it was placed similar in ABC, but the rats have in UCNPs have shown minimal background tissue damage as compared to the quantum dots. That is one more advantage of UCNPs over quantum dots. First is luminescent and second is minimal background tissue damage.
Okay, thank you, ma'am.
Why would you want a luminescent nanoparticle?
Because in imaging, ma'am, the luminescence is clearly visible as compared to other nanoparticles. So it was, main idea was to develop a luminescent based nanoparticle and not just a normal nanoparticle.
No, I understand why you would need luminescence for imaging, but why drug delivery?
Yeah. I'll have to do a little more literature survey on that.
Okay. Okay, thank you.
Thank you, PUG19. I am very grateful to our judges for their precious time. I am very grateful to our judges for their precious time and support, without which this event would have not been possible. Thank you to all the participants and delegates. You have been very enthusiastic and cooperative for this event. It was really amazing to watch all participants in such a competitive spirit. Dear judges, I request you to please speak a few words and provide your valuable feedback for our participants. We would love to know your overall experience for this event.
I was really happy with the knowledge gained by the participants during this entire process. Happy to know that you are going beyond your curriculum, stretching your imaginations to understand research papers, which is not easy at UG level. The topics that were picked up by the students were the latest ones. So good that you want to keep yourself updated. And considering your level of knowledge that you've gained so far, I think you've done a good job. But definitely you should read more because there is never an end to learning. So that's it from me.
Thank you, ma'am. Thank you, Radhika, ma'am. Dr. Clara?
I second ma'am completely. I think at this stage to take up such topics which are contemporary and very new to an undergraduate. And I think the very fact that you are able to tackle our questions, it's commendable. And I wish all of you the very best because the very fact that you have a vision to see what is new and interesting happening in the current scenario definitely shows that you have a research bent of mind. So I really commend each one of you, and I wish you all success. And as ma'am said, learning never stops. You have to keep on, upgrading your knowledge and make sure that you aspire to not only participate and win competition, but also gain knowledge and make an impact in society with your research and with your innovations. All the best to everybody. And once again, thank you, organizers, for giving me this opportunity to participate. And definitely, I have learned from the posters, and definitely, it's going to help me to incorporate certain findings in my research. So, thank you very much.
Thank you, Dr. Clara. Thank you, judges, for giving us your valuable time and gracing the event by your presence. I would now like to present you both with a certificate of appreciation.
Thank you.
Thank you. At the end, I would also like to thank technical team and all the volunteers for their constant support. You have been the backbone of this event. With this, the day 2 of the second Student Research Congress comes to an end. Please join us back tomorrow for another day filled with talks, competitions, and award felicitation ceremony. Looking forward to see you all tomorrow. The feedback link is available in the chat box and will be valid for 20 minutes. So I, your host, Nikita Pase, now would like to sign off for the day. Thank you, everyone. Have a wonderful evening.